വെൽക്കം ടു ബയോളജി ട്യൂട്ടർ. ബയോളജി ട്യൂട്ടറിന്റെ പുതിയൊരു വീഡിയോയിലേക്ക് ഏവർക്കും സ്വാഗതം. ഇന്ന് നാം ഡിസ്കസ് ചെയ്യാൻ പോകുന്നത് മോഡൽ ക്വസ്റ്റ്യൻ പേപ്പറാണ്. 100 ക്വസ്റ്റ്യൻസ് ഫ്രം ഫുഡ് ടെക്നോളജി. 100 ക്വസ്റ്റ്യൻ നമ്മൾ ഡിസ്കസ് ചെയ്യാൻ പോകുക. ഇമ്പോർട്ടൻറ്റ് ഫാക്ട്സ്. അൺറാവൽ ദ മിസ്റ്ററി ഓഫ് കൊക്കുമി പെപ്റ്റൈഡ്സ് ആൻഡ് ഉമാമി. കൊക്കുമി പെപ്റ്റൈഡ്സ്, ഉമാമി, അതുപോലെ ഇമ്പോർട്ടൻറ്റ് ഫാക്ട്സ് ഇതിൽ നമ്മൾ ഡിസ്കസ് ചെയ്യും. കംപ്ലീറ്റ് ആയിട്ട് വീഡിയോ കാണുക. 100 ക്വസ്റ്റ്യൻസ് ഉണ്ട്, ഇത് ഉപകാരപ്പെടും. ക്വസ്റ്റ്യൻസ് ആൻസർ ചെയ്ത് എത്ര മാർക്ക് കിട്ടി എന്നുള്ളത് കമൻറ്റ് ചെയ്യുക, അല്ലെങ്കിൽ നിങ്ങൾ എന്ത് ചെയ്യുക, അത് മനസ്സിലാക്കി വയ്ക്കുക. അപ്പോൾ നിങ്ങളൊരു റേഞ്ച് നിങ്ങൾക്ക് മനസ്സിലാവും. 100 ക്വസ്റ്റ്യൻസിൽ എത്ര നിങ്ങൾക്ക് സ്കോർ ചെയ്യാൻ പറ്റി, നിങ്ങളുടെ റേഞ്ച് എത്ര കിട്ടി എന്നുള്ളത്. എക്സാം ക്ലിയർ ആവണം ഉള്ള ആൾക്കാർ എന്ത് ചെയ്യുക, കൃത്യമായിട്ട് ആൻസർ ചെയ്യുക. അതുപോലെ തന്നെ മാർക്ക് കിട്ടിയത് കമൻറ്റ് ചെയ്യുക, അല്ലെങ്കിൽ അതിൻ്റെ എക്സ്പീരിയൻസ് ഒന്ന് കമൻറ്റ് ചെയ്യുക. ഈ രീതിയിലുള്ള ഇനി ക്വസ്റ്റ്യൻസ് വേണമെങ്കിൽ കമൻറ്റ് ചെയ്യുക. അങ്ങനെയാണെങ്കിൽ നെക്സ്റ്റ് ഓരോരോ സെഷൻ അനുസരിച്ച് ക്വസ്റ്റ്യൻസ് ഉണ്ടാക്കിയിട്ട് അപ്ലോഡ് ചെയ്യാം. Unravel the mystery of Kokumi peptides and umami. First question: which pigment is primarily responsible for the red color in cured meats? Myoglobin, that is the image of myoglobin. Second one, hemocyanin. C, nitrosomyoglobin. D, anthocyanin. Here answer, which is the answer here? Here answer is C, nitrosomyoglobin. Second question, which is the most heat stable vitamin? Vitamin A, vitamin D, niacin, vitamin K. Which is the answer? Here answer is C, niacin. Most effective form of niacin is nicotinamide. Third question, which compound causes astringency in tea and unripe fruits? Tannins, pectin, catecholamine, lignin. Here the answer, which is the answer? Here answer is A, tannins. Fourth question, in freeze drying, phase transition occurs directly from dash phase to dash phase. Solid to gas, liquid to gas, liquid to solid, solid to liquid. Here answer is A, solid to gas. Fifth question, high pressure processing HPP can best be described as use of extreme heat to kill pathogens. B, application of isostatic pressure to inactivate microorganisms. C, irradiation of food for sterilization. D, ultrasonic vibration for food preservation. Here answer is B, application of isostatic pressure to inactivate microorganisms. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notification. Sixth question, which bacterium is commonly used in the production of natto, a traditional Japanese fermented food from soya? A, Pseudomonas fluorescens. B, Lactobacillus acidophilus. C, Saccharomyces cerevisiae. D, Bacillus subtilis. Here answer is D, Bacillus subtilis. Seventh question, which compound is most responsible for the spiciness of chili peppers? Capsaicin, B, piperine, C, curcumin, D, allyl isothiocyanate. Capsaicin എന്ന് പറഞ്ഞാൽ ചില്ലിയിലുള്ളതാണ്, പൈപ്പറിൻ എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ പെപ്പറിൽ, കർക്കുമിൻ എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ മഞ്ഞളിൽ, ഇതിലുള്ള ടർമറിക്കിലുള്ളതാണ്. ഹിയർ ആൻസർ ഈസ് എ, capsaicin. AITC. AITC എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ it's a naturally occurring unsaturated isothiocyanate. The colorless oil is responsible for the pungent taste of cruciferous vegetables such as mustard, radish, horseradish and wasabi. അപ്പോൾ അതാണ് എന്താണ് അല്ലൈൽ ഐസോതയോസൈനേറ്റ്. അപ്പോൾ അങ്ങനെ MCQ ചോദിച്ചു കഴിഞ്ഞാൽ അതുകൂടി ആൻസർ ചെയ്യാൻ പറ്റണം. എയ്ത്ത് ക്വസ്റ്റ്യൻ, വിച്ച് അമിനോ ആസിഡ് ഈസ് കൺസിഡേർഡ് എ ലിമിറ്റിംഗ് അമിനോ ആസിഡ് ഇൻ വീറ്റ്? ലൈസിൻ, മെത്തിയോനിൻ, ല്യൂസിൻ, arginine. Which is the answer over here? Here answer is a lysine. Ninth question, which packing material is most effective for vacuum sealed meat products? A, high density polythene HDPE. B, PVC. C, EVOH. D, polystyrene. Here answer is C, ethylene vinyl alcohol, EVOH. 10th question, which gas is commonly used to maintain the freshness of packaged snacks? Packaged snacks freshness keep in gas. Oxygen ano, nitrogen ano, carbon dioxide ano, helium ano. Your answer is B, nitrogen. 11th question. What is the permitted level of arsenic in drinking water as per FSSAI regulations? A 10 ppb, B 20, C 50, D 100. Here answer is a 10 ppb. 12th question. Which microorganism is used in tempeh fermentation? That is soya fermented food and tempeh. Here the answer A. Rhizopus oligosporus, B. Saccharomyces cerevisiae, C. Lactobacillus bulgaricus, D. Aspergillus oryzae. Which is the answer over here? Your answer is A. Rhizopus oligosporus. 13th question. Which protein is responsible for celiac disease in individuals consuming wheat? A. Glutenin, B. Gliadin, C. Casein, D. Albumin. Here the answer over here B. Gliadin. Here the answer gliadin. In the explanation of celiac disease or celiac disease is a chronic immune mediated intestinal disorder in which the body becomes intolerant to gliadin, which is a component of gluten. And individuals with celiac disease exhibit a lifelong intolerance of wheat, barley and rye, all of which contains prolamines. Now, this is mindil vekkanam. The answer is B. Gliadin. 14th question. What is the primary source of aflatoxin contamination in food crops? A. Poor storage conditions, B. Excessive irrigation, C. Soil contamination, D. Pesticide use. But answer is poor storage condition. That is humidity fungal growth. Storage condition clear. The answer A. 15th question. 15. Which fungus is responsible for producing aflatoxins? Aspergillus flavus, Penicillium notatum, Fusarium oxysporum, Trichoderma hertzianum. Answer is Aspergillus flavus. 16th question. Which of the following aflatoxin is most commonly found in food contamination? B1, G2, M1, B2. Here answer is B1. 17. Aflatoxin contamination in food primarily affects which organ in humans? Liver, kidney, heart, lungs. Liver affect liver. 18th question. Which of the following is potential health consequence of chronic aflatoxin exposure? Chronic aflatoxin exposure is skin rashes, liver cancer, respiratory infections, kidney stones. Your answer is liver cancer. B answer, liver cancer. 19th question. Which diagnostic technique is commonly used for detection of aflatoxin in food and feed? Gas GCMS, ELISA, HPLC, both B and C. Right? ELISA, HPLC and the answer. Here answer is D. 20th question. Which crop is most commonly affected by aflatoxin contamination? Wheat, maize, rice, barley. Yet affected crop B answer, maize, B. 21. Which of the following countries has the highest rates of aflatoxin exposure due to poor food safety standards? Here answer is C, Kenya. 22. Aflatoxins are known to be classified as which type of carcinogen by the International Agency for Research on Cancer, IARC? Group 1, carcinogenic to humans. Group 2A, probably carcinogenic to humans. 2B, possibly carcinogenic to humans. Group 3, not classifiable as to its carcinogenicity to humans. Here answer is A, Group 1, carcinogenic to humans. 23rd. Who is the current Chief Executive Officer for FSSAI? Sri Apurva Chandra IAS, Sri Kamala Vardhana Rao IAS, Rita Theosia IAS, Sri Arun Singhal IAS. Here answer is B, Kamala Vardhana Rao IAS. At present, FSSAI chairman. Who is the chairman of FSSAI? Sri Apurva Chandra IAS. Rita Theosia, Sri Arun Singhal, former FSSAI CEOs. Name of former FSSAI CEOs are Rita Theosia and Sri Arun Singhal. 24th question. Which amino acid is responsible for the Maillard reaction in food? Lysine, proline, methionine, serine. Which is the answer over here? Here answer is A, lysine. Here explanation. The first group which gave the most intense Maillard browning Reaction included lysine, glycine, tryptophan, and tyrosine. While alanine, valine, leucine, isoleucine, phenylalanine, proline, methionine, asparagine, and glutamine were intermediate browning producing amino acids. 25th question. What is the main chemical responsible for the pungent flavor in garlic? Allicin, capsaicin, lactic acid, lycopene. Lycopene in tomato, lactic acid in lactic acid bacteria fermented food, capsaicin in chilies, allicin in garlic. Your answer is allicin. 26th. Which type of bond is broken during gelatinization of charge? Glycosidic bond, hydrogen bond, ionic bond, covalent bond. Your answer is B, hydrogen bond. 27. Which vitamin is most sensitive to heat during cooking? D, C, E, K. Your answer is B, vitamin C. 28. What is the primary component of dietary fiber? Cellulose, pectin, starch, hemicellulose. Your answer is A, cellulose. 29th question. Which microorganism is commonly used for yogurt production? Lactobacillus bulgaricus, Aspergillus niger, Penicillium roqueforti, Saccharomyces cerevisiae. Which is the answer? Your answer is A, Lactobacillus bulgaricus. 30th question. What is the D value in microbial inactivation? A. Time to reduce 90% of microorganisms. Time to reduce 50% of microorganisms. C. Temperature required to sterilize food. D. pH which growth is inhibited. Your answer is A. Time to reduce 90% of microorganisms. 31 question. Question number 31. Which microorganism is used in the production of blue cheese? Penicillium roqueforti, Saccharomyces cerevisiae, Lactobacillus casei, Staphylococcus aureus. Here which is the answer? Your answer is Penicillium roqueforti with green blue color to cheese. 32. Which method is used for pasteurization of milk? Heating at 72 degrees Celsius for 15 seconds, heating at 100 degrees Celsius for 1 minute, heating at 121 degrees Celsius for 20 minutes, defreezing at minus 20 degrees Celsius. Your answer is heating 72 degrees Celsius for 15 seconds. A. 33. What is the main advantage of high pressure processing? Enhanced flavor retention, increased shelf life, minimal loss of nutrients, all of the above. Which is the answer? Your answer is the all of the above. 34th question. Which enzyme is responsible for enzymatic browning in fruits? Polyphenol oxidase, amylase, protease, lipase. Here answer is A. Polyphenol oxidase. 35th question. What is the primary purpose of blanching in food processing? To inactivate enzymes, to kill all microorganisms, to reduce water content, to add flavor. Which is the answer? Your answer is A. To inactivate enzymes. 36th question. Freeze drying is an example of which type of drying? A. Spray drying, B. Sublimation drying, C. Conventional drying, D. Infrared drying. Your answer is B. Sublimation drying. 37th question. Which packaging material is most suitable for microwave heating? PET, glass, aluminum foil, polypropylene. Your answer is D. Polypropylene. 38th question. Modified atmosphere packaging MAP primarily involves altering which gases? Oxygen, carbon dioxide and nitrogen, hydrogen, nitrogen and methane, argon, helium and oxygen, methane, carbon dioxide and hydrogen. This is the answer in modified atmosphere packaging. Your answer is A. Oxygen, carbon dioxide and nitrogen are used in MAP, modified atmosphere packaging. 39. Which type of plastic is commonly used for bottled water? PVC, PET, HDPE, LDPE. Your answer is B. PET bottles, PET bottles. 40th question. What is the main function of an oxygen scavenger in food packaging? Prevent moisture loss, reduce oxidation, control microbial growth, maintain temperature. Your answer is B. Reduce oxidation. 41. Which type of packaging uses edible coatings? A. Active packaging, B. Biodegradable packaging, C. Smart packaging, D. Intelligent packaging. Your answer is B. Biodegradable packaging. 42nd question. What is the primary purpose of using emulsifiers in food products? To extend shelf life, to improve texture and stability, to enhance flavor, to preserve nutrients. Your answer is to improve texture and stability. B. 43. Which food additive is used as thickening agent? Xanthan, sorbic acid, sodium benzoate, aspartame. Aspartame is used as artificial flavonoid. Sodium benzoate and sorbic acid is used as preservatives. And xanthan gum here is used as thickening agent. Answer is A. Xanthan gum. 44. What is the function of ascorbic acid as a food additive? Sweetener, antioxidant, emulsifier, coloring agent. Your answer is B. Antioxidant. 45th question. Which food additive is primarily responsible for enhancing flavor? Monosodium glutamate MSG, potassium sorbate, carrageenan, sodium nitrate. Your answer is A. MSG. 46th question. Sodium nitrite is commonly used in processed meat for color preservation and preventing spoilage. B. Sweetening. C. Enhancing texture. D. Increasing shelf life. Your answer is A. Color preservation and preventing spoilage. 47th question. Omega-3 fatty acids are primarily associated with which health benefit? A. Heart health. B. Improved digestion. C. Bone strengthening. D. Better vision. Which is the answer over here? Your answer is A. Heart health. Omega-3 fatty acids are primarily associated with heart health. 48. Which is the main bioactive compound in green tea associated with its health benefits? A. Caffeine. B. Catechins. C. Tannins. D. Phenols. The answer here answer is B. Catechins. 49. Fortified foods are foods enriched with additional nutrients, natural foods with no additives, foods treated with microbial safety, foods with with a longer shelf life. Foods with a longer shelf life. Your answer is A. Food enriched with additional nutrients. Next, kokumi peptides. What are kokumi peptides? Kokumi peptides. Kokumi is a sensation that is activated by peptides that occur naturally in fermented foods. It can also naturally occur in more Western fare like beer, bread, chicken, soup, various peptides such as glutathione and other glutamyl di and tripeptides isolated from cheese, edible beans, soy sauce and yeast extract all have kokumi kokumi sensation എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ kokumi peptides എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ fermented foodsൽ നിന്ന് അതിലുണ്ടാവുന്ന പ്രത്യേകതരം peptides ആണ്. Kokumi peptides. The researchers have determined that calcium receptors are activated by kokumi foods. അതായത് kokumi peptides ഉള്ള ഫുഡ്സ് fermented foods ഉൾപ്പെടെയുള്ള ഫുഡ്സ് എന്ത് ചെയ്യും? കാൽസ്യം റെസപ്റ്റേഴ്സിനെ ആക്ടിവേറ്റ് ചെയ്യും. Calcium receptors are activated with the help of kokumi foods, which magnifies the signals to the brain regarding texture, mouthfeel, complexity and duration of flavors. അതായത് ഈ kokumi peptides കാൽസ്യം റെസപ്റ്റേഴ്സിനെ എന്ത് ചെയ്യും? ആക്ടിവേറ്റ് ചെയ്യും. ആ ആക്ടിവേറ്റ് ചെയ്യുന്ന ഫലമായി എന്ത് ചെയ്യും? സിഗ്നൽസ് നമ്മൾ കഴിക്കുന്ന ഫുഡിന്റെ സിഗ്നൽസ് ആംപ്ലിഫൈ ചെയ്യും, അല്ലെങ്കിൽ എന്ത് ചെയ്യും? മാഗ്നിഫൈ ചെയ്തിട്ട് നമ്മുടെ ബ്രെയിനിൽ ഒരു ഫീൽ ഉണ്ടാക്കും. ടെക്സ്ചർ, മൗത്ത് ഫീൽ, കോംപ്ലക്സിറ്റി ആൻഡ് ഡ്യൂറേഷൻ ഓഫ് ഫ്ലേവറിനെ പറ്റി ഒരു ഐഡിയ, ഒരു സിഗ്നൽ പാസ് ചെയ്യും നമ്മുടെ ബ്രെയിനിലേക്ക്. അതാണ് kokumi peptides. ഇതാണ് ടേസ്റ്റ് ബഡ്സ്. സോർ, umami, salty and sweet buds are located in tongue. You can see in that image, particular image. Kokumi is known more as a flavor modifier and mouthfeel rather than a taste. Right? Flavor modifier and kokumi peptides. Examine question ചോദിക്കാൻ ചാൻസ് ഉണ്ട്. Flavor modifier and mouthfeel ഉണ്ടാക്കുന്ന പ്രത്യേകം peptides ആണ്. When eating chicken soup, that savory, brothy and meaty flavor is thanks to umami. Umami taste. The complexity of flavors and the lingering sensation of body and richness in the mouth attributes to its mouthfeel. അതാണ് umami taste എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ. The basic kokumi is triggered by certain peptides and amino acids in foods that activate calcium receptors on the tongue. The activation of these receptors enhances our perception salt, sweet and umami. അപ്പോൾ ഉമാമി ടേസ്റ്റും അതിൻ്റെ സ്വീറ്റും സാൾട്ടും അതിൻ്റെ പെർസെപ്ഷൻ എൻഹാൻസ് ചെയ്യാൻ വേണ്ടിയിട്ട് കാൽസ്യം റെസപ്റ്റേഴ്സിനെ ആക്ടിവേറ്റ് ചെയ്യുന്നതാണ് ഉമാമി എന്ന് പറയുന്നത്. ആൻഡ് ഓൺ ദ അതർ ഹാൻഡ് വി ടേസ്റ്റ് ഉമാമി വെൻ സ്പെസിഫിക് ടേസ്റ്റ് റെസപ്റ്റേഴ്സ് ഓൺ അവർ ടങ് കം ഇൻ ടു കോണ്ടാക്ട് വിത്ത് ഗ്ലൂട്ടമേറ്റ്സ് ആൻഡ് ന്യൂക്ലിയോടൈഡ്സ്. അതായത് ഉമാമി ടേസ്റ്റ് എപ്പോഴാണ് നമുക്ക് അനുഭവപ്പെടുക? ഗ്ലൂട്ടമേറ്റ്സും ന്യൂക്ലിയോടൈഡ്സും ഉൾപ്പെടെയുള്ള അത് ടേസ്റ്റ് റെസപ്റ്റേഴ്സിന് ബൈൻഡ് ചെയ്യുന്ന സമയത്താണ്. These are many foods with naturally occurring glutamates and nucleotides such as tomatoes, mushrooms, cured cheeses, meats and seafoods. Kokumi and umami. Kokumi and umami work together synergistically to give us a pleasantly enhanced explosion of flavors. They can be used to make foods reduce in salt and fat more palatable. There are many clean label foods and ingredients that deliver both kokumi and umami, such as soy sauce, shrimp paste, and in the commercial or industrial manufacturing world, yeast extract or hydrolyzed vegetable proteins. With the health foods on the rise, incorporating ingredients into foods that provide umami and kokumi can be a vital tool in turning foods from dull to an exceptional culinary experience. Adana kokumi versus and umami. Next, 50th question. Which of the following is a functional food ingredient known for its cholesterol lowering properties? Beta glucan, lactic acid, glutamate, ascorbate. ഏതാണ് ആൻസർ? എ ആണ് ആൻസർ, ബീറ്റ ഗ്ലൂക്കാൻ. ബീറ്റ ഗ്ലൂക്കാൻ എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ അത് എന്താണ്? അത് ഫൈബേഴ്സ്, സൊല്യൂബിൾ ഫൈബേഴ്സ് ആണ്, സൊല്യൂബിൾ ഫൈബേഴ്സ്. 51. Which microorganism is commonly used in beer production? Saccharomyces cerevisiae, Lactobacillus acidophilus, Rhizopus oligosporus, Aspergillus niger. Your answer is a Saccharomyces cerevisiae. 52. Which fermented product is made using Aspergillus oryzae? Soy sauce, yogurt, beer, cheese. Your answer is soy sauce A. 53. The SCOBY used in kombucha production is a combination of bacteria and yeast, mold and yeast, algae and bacteria, fungi and mold. Your answer is A, bacteria and yeast. 54. What is the main principle behind pulsed electric field technology? Electroporation, heat transfer, high pressure, centrifugation. എന്താ അതിൻ്റെ ആൻസർ വരിക? ഹിയർ ആൻസർ എന്താ വെച്ചാൽ എ ആണ്, ഇലക്ട്രോപോറേഷൻ ആണ്. ഇലക്ട്രോപോറേഷൻ ആണ് ഇതിൻ്റെ ആൻസർ. 55. Cold plasma technology is primarily used for food surface decontamination, nutrient enhancement, shelf life extension, packaging design. Cold plasma technology is used for food surface decontamination. Answer is A. 56. Golden rice is genetically modified to produce which nutrient? Vitamin A, iron, protein, omega-3 fatty acids. Your answer is A, vitamin A. 57. Which compound triggers celiac disease? Gluten, casein, lactose, histamine. Your answer is gluten, A. 58. Peanut allergy is example of IgE mediated allergy, non-IgE mediated allergy, food intolerance, enzyme deficiency. Your answer is A, IgE mediated allergy. 59. Histamine intolerance is linked to consumption of fermented foods, fresh fruits, processed grains, low-fat milk. Your answer is A, fermented foods. Histamine intolerance is linked to fermented foods. 60. The big 8 allergens include all except. Big 8 allergens. ഇതിലേതാ എക്സെപ്റ്റ് എന്ന് പറഞ്ഞാൽ റൈസ് ആയിരുന്നു, റൈസ്. ബിഗ്, വാട്ട് ആർ ബിഗ് 8 allergens? Milk, eggs, fish, crustacean shellfish, tree nuts, peanuts, wheat and soya beans are considered as the big 8 allergens. 61. Which of the following is a natural food preservative? Sodium benzoate, nisin, propylene glycol, aspartame. Your answer is B, nisin. Nisin is a natural antimicrobial peptide or bacteriocin produced by strains of Lactococcus lactis subspecies lactis that effectively inhibits gram-positive and gram-negative bacteria and also the outgrowth of spores of bacilli and clostridia. 62. Antioxidants in food prevent oxidative rancidity, loss of texture, microbial growth, color enhancement. Your answer is A, oxidative rancidity. 63. Which synthetic sweetener is 200 times sweeter than sucrose? Sucralose, aspartame, saccharin, stevia. Your answer is B, aspartame. 64. Which food colorant is derived from natural sources? Tartrazine, annatto, brilliant blue, sunset yellow. Your answer is B, annatto. Annatto is an orange-red condiment and food coloring derived from the seeds of the Bixa orellana. This is the plant of Bixa orellana. 65. Cholesterol lowering effects are a hallmark of foods enriched with plant sterols, omega-6 fatty acids, anthocyanins, lignans. Your answer is a plant sterols. 66. Anthocyanins, anthocyanins, known for their antioxidant properties, are primarily found in blueberries and blackberries, green leafy vegetables, whole grains, dairy products. Which is the answer? Your answer is a blueberries and blackberries. 67. Which is an example of functional beverage? Fermented kefir, carbonated soda, orange juice with pulp, bottled water. Your answer is a fermented kefir is known as among these fermented kefir is a functional beverage. 68. Which functional food category do soy based products belong to? Anti-inflammatory foods, phytoestrogen rich foods, energy dense foods, mineral fortified foods. Your answer is B. Phytoestrogen rich foods. 69. Which acid is primarily responsible for the preservation of pickles? Acetic acid, citric acid, tartaric acid, lactic acid. Your answer is A. Acetic acid. 70. Which thickening agent is most commonly used in tomato sauce? Cornstarch, xanthan gum, gelatin, pectin. Here, which is the answer? Your answer is a cornstarch. 71. Which spice contains the compound piperine known for its pungency? Piperine is a compound in black pepper. So here answer is black pepper. Turmeric, cardamom. Here answer is a black pepper. 72. What is the most common binding agent used in restructured fruit snacks? Pectin, gelatin, starch, agar-agar. Your answer is A. Pectin. 73. Which method is most effective in retaining vitamin C during fruit and vegetable preparation? Steaming, boiling, microwaving, frying. Your answer is B or A. Which is the answer? Your answer is A. Steaming. 74. Which technique is widely used to extend the shelf life of minimally processed vegetables? A. Modified atmospheric packaging, B. Irradiation, C. Vacuum sealing, D. Fermentation. Here answer is A. Modified atmospheric packaging, MAP. 75. What is the primary benefit of the individually cooked frozen technique, IQF technique? Prevents enzymatic browning, preserves texture and flavor, reduces freezing time. Your answer is D. All the above. 76. Which microorganism is primarily involved in natural fermentation of pickles? Lactobacillus plantarum, Saccharomyces cerevisiae, Clostridium sporogens, Aspergillus oryzae. Your answer is A. Lactobacillus plantarum. 77. How is marmalade thickness controlled? A. Adjusting pectin levels, varying cooking time, altering sugar content. Your answer is D. All of the above. 78. In kimchi fermentation, which bacterium predominates during the final Staphylococcus lactis, Leuconostoc misandroids, Lactobacillus brevis, Wisella siberia? Your answer is C. Lactobacillus brevis. 79. Which fermentation product is a primary precursor for the umami flavor in soy sauce? Glutamic acid, succinic acid, pyruvic acid, lactic acid. Your answer is A. Glutamic acid. 80th question. Which bacterium produces diacetyl responsible for the butterly flavor in some dairy-based fermented products? A. Lactobacillus casei, Lactococcus lactis, subspecies Lactis biovar diacetyl lactis. C. Bifidobacterium bifidum. D. Enterococcus faecium. Your answer is B. Lactococcus lactis, subspecies Lactis biovar diacetyl lactis. 81. During fermentation of kefir, the kefir grains contains both yeast and mold, yeast and lactic acid bacteria, acetic acid bacteria and molds, propionic acid bacteria and fungi. Which is the answer over here? Your answer is B. Yeast and lactic acid bacteria. 82. Which microorganism is most frequently associated with the spoilage of fermented cucumbers? A. Pseudomonas fluorescens, B. Lactobacillus bushneri, C. Clostridium bifermentans, D. Zygosaccharomyces roxae. Your answer is Lactobacillus bushneri. B. 83. Which metabolite produced during fermentation enhances the shelf life of fermented dairy products? Hydrogen peroxide, lactic acid, carbon dioxide, acetoin. Here answer is a hydrogen peroxide. 84. In wheat flour fortification, which form of iron is preferred due to its high bioavailability? Ferrous sulfate, ferric chloride, ferrous fumarate, ferric citrate. Which is the answer? Your answer is ferrous sulfate. Your answer is A. Ferrous sulfate. 85. Golden rice was genetically engineered to combat vitamin A deficiency by introducing which provitamin? Beta carotene, retinol, ascorbic acid, ergocalciferol. Your answer is A. Beta carotene. 86. What is the most significant challenge in fortifying rice using coating and extrusion techniques? A. Cost. B. Retention of micronutrients after cooking. C. Consumer acceptability. D. Shelf life of fortified kernels. Your answer is B. Retention of micronutrients after cooking. 87. Which fortified nutrient is added to prevent tube defects in newborns? Vitamin D, calcium, folic acid, zinc. Here answer is C. Folic acid. 88. In oil fortification, why is vitamin E often co-added with vitamin A or D? To stabilize the oil, to prevent oxidation of vitamin A or D, to improve taste, to enhance viscosity. Here answer is B. To prevent oxidation of vitamin A or D. 89. Which type of iron is commonly used in rice fortification due to its minimal sensory changes? Ferric pyrophosphate, ferrous sulfate, ferrous ascorbate, ferric chloride. Which is the answer? Your answer is a ferric pyrophosphate. 90. What is the major drawback of calcium fortification in beverages? Increased bitterness, reduced solubility, interaction with vitamin D, accelerated spoilage. Your answer is B. Reduced solubility. 91. In India, which symbol indicates a fortified food product? A. A triangle inside a circle. A. Plus symbol with a green background. An F with a ring. A square with a check mark. Your answer is B. A plus symbol with a green background. This is the image of tongue. You can see particular taste buds of sour, umami, salty, sweet, bitter in particular color. 92. What is the primary role of L-theanine in health drinks? Act as emulsifier, enhances mental relaxation without drowsiness. C. Provides a bitter taste. D. Serves as a preservative. Your answer is B. Enhances mental relaxation without drowsiness. L-theanine is an amino acid. The human body does not produce this compound and it is not essential for humans. Green tea, black tea and certain types of mushroom naturally contain L-theanine. L-theanine may affect the levels of certain chemicals in the brain. The dose makes the poison. Apple seeds contain amygdalin. Pears contain formaldehyde. Potatoes contain solanine. Cucumber contain higher bitter courgettes. All of the food items above contain natural chemicals that are toxic to humans. However, they are usually present in very small amounts, far below the harmful dose. 93. The bioavailability of calcium in fortified health drinks is significantly improved by the addition of vitamin D, magnesium, zinc, phytase. Here answer is A. Vitamin D. 94th question. Which of the following is often used as a natural nootropic ingredient in health drinks? Spirulina, guarana, turmeric, ashwagandha. Here answer is B. Guarana. Nootropic means a substance that enhances cognition and memory. This is guarana plant seeds. 95. Why are omega-3 fatty acids micro-encapsulated in health drinks? To enhance taste, to prevent oxidation and rancidity, to improve solubility in water, to increase the product's viscosity. Your answer is B. To prevent oxidation and rancidity. 96. Which non-dairy milk alternatives is commonly fortified with vitamin B12 in health drinks? A. Soy milk, B. Almond milk, C. Rice milk, D. Coconut milk. Here answer is a soy milk. 97. Which rare fungus is nicknamed black gold and is highly prized in gourmet cuisine? Morel, porcini, truffle, shiitake. Here answer is C. Truffle. 98. The fruit of the miracle berry plant temporarily alters taste perception by making sour foods taste sweet due to the presence of which compound? Miraculin, glycine, maltodextrin, aspartame. Your answer is a miraculin. 99th question. What is the primary ingredient of the Japanese delicacy fugu, which requires careful preparation due to its toxicity? If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. A. Tuna. B. Pufferfish. C. Octopus. D. Eel. Here answer is B. Pufferfish. Last question. 100 question. ഈ ക്വസ്റ്റ്യൻ ആൻസർ ചെയ്തതിനു ശേഷം എത്ര മാർക്ക് കിട്ടിയ ഒരു 80-85 കൂടുതൽ കിട്ടിയ ആൾക്കാർ എന്തായാലും കമൻറ്റ് ചെയ്യുക. കാസു മാർസു എ റയർ ചീസ് ഫ്രം സാർഡീനിയ ഈസ് മെയ്ഡ് യൂണിക് ബൈ ആഡിങ് ഫംഗസ്, ലൈവ് ഇൻസെക്ട് ലാർവേ, ചാർക്കോൾ ആഷ്, herbs and spices. Your answer is B. Live insect larvae. Subject Nutritional Biochemistry and Food Analysis. Please share your thoughts in the comment box and feedbacks also in my comment box. Thank you. All the best for your exam. First question. Which carbohydrate acts as a prebiotic enhancing gut microbiota? A. Glucose. B. Fructose. C. Inulin. D. Starch. Here answer is C. Inulin. Inulin is a type of prebiotic. It's not digested or absorbed in the stomach. It stays in the bowel and helps certain beneficial bacteria to grow. Inulin is a starchy substance found in a wide variety of fruits, vegetables, and herbs, including wheat, onions, bananas, leeks, artichokes, and asparagus. Second question. What is the glycosidic linkage in lactose? Alpha 1,4, beta 1,4, alpha 1,6, beta 1,6. Which is the answer? Here answer is B. Beta 1,4 linkage. This is a chart. Here you can see the linkage of sucrose, lactose, maltose, and trehalose. Study well for your exam. Third question. Which of the following carbohydrates is a reducing sugar? Sucrose, maltose, starch, cellulose. Which is the answer? Here answer is B. Maltose. Reducing sugar. What are reducing sugars? A reducing sugar that serves as a reducing agent due to its free aldehyde or ketone functional groups in its molecular structure. Examples are glucose, fructose, glyceraldehydes, lactose, arabinose, and maltose. Sucrose is a non-reducing sugar because the two monosaccharide units are held together by a glycosidic linkage between C1 of alpha glucose and C2 of beta fructose. Since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Fourth question. What is the storage form of carbohydrates in animals? Amylopectin, cellulose, glycogen, chitin. Which is the answer? Here glycogen. Fifth question. Which is the second most abundant biopolymer on earth after cellulose? Starch, lignin, suberin, chitin. Which is the answer? Here answer is D. Chitin. Chitin is found in insects' body. Sixth. Which carbohydrate does not undergo hydrolysis? Sucrose, maltose, fructose, lactose. Here which is the answer? Here answer is C. Fructose. Disaccharides consist of two monosaccharide units linked together with glycosidic bonds in the alpha or beta orientation. The most important of them are sucrose, lactose, and maltose. Sucrose is the most abundant and consists of a molecule of alpha glucose and beta fructose linked together. A carbohydrate which cannot be hydrolyzed to simpler compound is known as monosaccharide. Seventh question. What is the primary carbohydrate found in honey? Glucose, fructose, maltose, sucrose. Here which is the answer? Here answer is fructose. B. Fructose. Honey is made up of fructose, glucose, and other sugars. Honey is a concentrated carbohydrate solution containing a mix of simple and complex sugars. The two main sugars present in honey are fructose and glucose. Australian honey usually contains 36 to 50% of fructose and 28 to 36% glucose depending on the floral source. Eighth question. Which of these is an example of a heteropolysaccharide? Starch, glycogen, hyaluronic acid, cellulose. Which is the answer? Your answer is C. Hyaluronic acid. It is a polysaccharide that is made up of D-glucuronic acid and N-acetyl glucosamine. It's found in connective tissue and skin. Ninth question. Which carbohydrate is known as fruit sugar? Glucose, sucrose, fructose, lactose. Which is the answer? Here answer is C. Fructose. Fructose is also known as fruit sugar because it primarily occurs naturally in many fruits. It also occurs naturally in other plant foods such as honey, sugar beets, sugarcane, and vegetables. Fructose is the sweetest naturally occurring carbohydrate and is 1.2 to 1.8 times sweeter than sucrose or table sugar. 10th question. Which gas is commonly used to maintain the freshness of packaged snacks? Oxygen, nitrogen, carbon dioxide, helium. Which is the answer? Here answer is nitrogen. Sweetener excipients. Here you can see sweetener and sucrose. The unit of relative sweetness of sucrose is 1 and addendum is 20,000 times sweeter than sucrose. Sucralose is 600 times sweeter than sucrose. Like fructose is 1.3 times sweeter than sucrose. Please understand and study this chart for your exam. Next, 11th question. Which carbohydrate polymer is used in gel formation in jams? Cellulose, pectin, amylose, inulin. Which is the answer? Here answer is B. Pectin. Pectin is the natural class of carbohydrate found in fruit which is responsible for gelling of fruit juice or pulp. 12th question. Which of the following is a ketohexose? Glucose, fructose, galactose, mannose. Which is the answer? Here answer is B. Fructose. Ketohexoses are 6 carbon monosaccharides which include fructose, sucrose, sorbose, and tagatose. 13th question. What is the calorific value of carbohydrates per gram? 2 kilocalorie, 4 kilocalorie, 6 kilocalorie, 9 kilocalorie. Which is the answer? Here answer is B. 4 kilocalorie. The gross calorific value of fats is actually 9.45 kilocalorie per gram. But in exams, it may ask like mention like 9 kilocalorie per gram. For carbohydrate, it is 4.1. Actual value is 4.1. But for exams, usually it will ask 4 kilocalorie per gram. And for proteins, it is 5.65 kilocalorie per gram. 14th question. Which carbohydrate acts as an anticoagulant? Glycogen, heparin, amylose, chitin. Here answer is B. Heparin. Heparin is a highly sulfated glycosaminoglycan or mucopolysaccharide, long unbranched polysaccharides consisting of a repeating disaccharide unit. It is an anticoagulant and is used to treat blood clots in the veins, arteries, and lungs. 15th question. What is the main structural carbohydrate in fungi? Cellulose, chitin, pectin, starch. Which is the answer? Here answer is B. Chitin. 16th question. Which lipid type primarily contributes to the structure of cell membranes? Triglycerols, phospholipids, sterols, waxes. Which is the answer? Here answer is B. Phospholipids. 17. Which enzyme is responsible for breaking down triglycerides into glycerol and fatty acids? Lipase, amylase, protease, cellulase. Which is the answer? Here answer is lipase A. 18th question. What is the iodine value of a lipid indicative of its calorific value, the degree of unsaturation, its melting point, its emulsification capacity? Which is the answer? Here answer is B, the degree of unsaturation. In chemistry, the iodine value IV, it is also known as absorption value, iodine number or iodine index. It is the mass of iodine in grams that is consumed by 100 grams of a chemical substance. Iodine numbers are often used to determine the degree of unsaturation in fats, oils and waxes. 19th question. Which of the following is an example of an essential fatty acid? Oleic acid, phenolic acid, stearic acid, palmitic acid. Here, which is the answer? Here answer is B, linoleic acid. You can see essential fatty acid and non-essential fatty acids. Here two types are linoleic acid and alpha linoleic acids are essential fatty acids. Here non-essential fatty acids include arachidic acid, stearic acid, palmitic acid, etc. 20. Question number 20. Which lipid is a precursor to steroid hormones? Cholesterol, glycerol, lecithin, sphingomyelin. Here answer is A, cholesterol. Cholesterol is the precursor for steroid hormones in the human body. Aside from steroids, cholesterol is crucial in making vitamin D and bile salt. Bile salts are made of bile acids that are conjugated with glycine or taurine. They are produced in the liver directly from cholesterol. Bile salts are important in solubilizing dietary fats in the watery environment of the small intestine. Sphingomyelin is an important constituent of mammalian cell membranes. 21. Which type of lipids are the main component of waxes? Steroids, fatty acid esters, glycerides, phospholipids. Which is the answer? Here answer is B, fatty acid esters. The wax contains mainly fatty esters 80 to 85%, free alcohol 10 to 15%, acids 3 to 6%, and hydrocarbons of 1 to 3%. 22nd. Which of these fatty acids has the lowest melting point? Lauric acid, myristic acid, linoleic acid, palmitic acid. Here answer is C, linolenic acid. In summary, just read this box. Then in summary, the order of fatty acids based on increasing melting point is alpha linoleic acid, oleic acid, stearic acid, palmitolic acid and palmitic acid respectively. 23rd. What is the chemical basis of rancidity in lipids? Hydrolysis of proteins, oxidation of unsaturated fatty acids, polymerization of triglycerides, dehydration of glycerol. Which is the answer? Here answer is B, oxidation of unsaturated fatty acids. 24. Which process converts unsaturated fats into trans fats during food processing? Hydrogenation, emulsification, hydrolysis, dehydration. Which is the answer? Here answer is A, hydrogenation. 25. Which one of the following is a marker for lipid peroxidation in food? TBA value, iodine number, saponification value, acid value. Which is the answer? Here answer is A, TBA value. TBA is one of the most widely used methods of assessing the extent of lipid oxidation in foods. The TBA value is typically expressed in milligrams of MDA equivalents per kilogram of sample as determined by the methods described. The TBA test measures MDA produced due to the oxidation of fatty acids with 3 or more double bonds, and it measures other TBA reactive substances such as 2-alkenols and 2,4-alkendiols. Therefore, TBA is also referred to as T-bars, TBA reactive substances. 26. Which fatty acid is commonly used as a marker for omega-3 index in nutrition? Linoleic acid, EPA, oleic acid, stearic acid. Which is the answer? Here answer is B, EPA. DHA is also a marine omega-3 found in fish. 27. Which fatty acid is most abundant in olive oil? Palmitic acid, stearic acid, oleic acid, linoleic acid. Which is the answer? In olive oil, the answer is C, oleic acid. 28. Before going to 28, we can discuss the major fatty acids in olive oil triglycerols are oleic acids, a mono unsaturated omega-9 fatty acid. It makes up 55 to 83% of olive oil. Linoleic acid C18-2, a polyunsaturated omega-6 fatty acid that makes up about 3.5 to 21% of olive oil. 28. Which of the following is not a complete protein? Soy protein, egg protein, gelatin, whey protein. Which is the answer? Here answer is option C, gelatin. Unlike other animal proteins like chicken, fish, eggs and meat, gelatin doesn't qualify as a complete protein source because it's a missing the essential amino acid tryptophan. Your body can't make essential amino acids, so you have to make sure you are getting enough of them through your diet. 29. Which type of bond stabilizes the alpha helix structure in proteins? Ionic bonds, hydrogen bonds, covalent bonds, van der Waals forces. Which is the answer? Here answer is B, hydrogen bonds. Intramolecular hydrogen bonding. The alpha helix structure for proteins is stabilized by intramolecular hydrogen bonding between CO and NH groups of different peptide bonds. 30. Question number 30. Which amino acid contains sulfur and contributes to disulfide bonds in proteins? A. Glycine, B. Methionine, C. Cysteine, D. Serine. Which is the answer? Here answer is C. Cysteine. Like cysteine, methionine is a sulfur-containing amino acid. The sulfur in methionine can be oxidized but is unable to form disulfide bonds. The R group renders methionine quite hydrophobic. In prokaryotic cells, a methionine residue is essential for the initiation of protein synthesis. Methionine, cysteine, homocysteine, and taurine are the four common sulfur-containing amino acids, but only the first two are incorporated into proteins. 31. Which protein structure is most sensitive to heat? Primary, secondary, tertiary, quaternary. Which is the answer? Here answer is option C, tertiary. 32. What is the typical isoelectric point range of casein? 4.6 to 5, 6 to 7, 8 to 9, 3.5 to 4. Which is the answer? Here answer is 4.6 to 5, option A. 33. Which protein is primarily responsible for the elasticity in wheat dough? Glutenin, albumin, globulin, gliadin. Which is the answer? Here answer is A, glutenin. One group of gluten proteins, the HMM subunits of glutenin, is particularly important in conferring high levels of elasticity. These proteins are present in HMM polymers that are stabilized by disulfide bonds and are considered to form the elastic backbone of gluten. 34. Which of these proteins acts as a molecular motor in muscle contraction? Myoglobin, myosin, albumin, casein. Which is the answer? Here answer is B, myosin. 35. Which protein component plays a major role in protein gel formation in tofu? Soy albumin, B. Glycinin, C. Beta conglycinin, D. Legumin. Which is the answer? Here answer is B. Glycinin. Thermal denaturation of soy proteins is a prerequisite for tofu gel formation. Differential scanning calorimetry of soy milk showed that the denaturation of temperature of glycinin 92 degrees Celsius is about 20 degrees Celsius higher than that of beta conglycinin 71 degrees Celsius. 36. Which test is used to differentiate proteins from amino acids? Ninhydrin test, xanthoprotein test, biuret test, Millen's test. Which is the answer? Your answer is C, biuret test. A small amount of this aqueous solution must be treated with 1% of sodium hydroxide or potassium hydroxide, that is Biuret test. We are explaining Biuret test. To this mixture, a few drops of copper sulfate aqueous must be added. If the solution turns purple upon the addition of copper sulfate, the presence of protein in the analyte is confirmed. 37. Which amino acid cannot be synthesized by the human body and must be consumed in the diet? Glutamine, alanine, leucine, proline. Which is the answer? Your answer is C, leucine. Here you can learn essential or non-essential amino acids. Like essential amino acids. Happy iguanas love leaping mountains proudly through the valley. So happy iguanas love leaping mountains proudly through the valley. That are the essential amino acids. 38. Which of the following fibers is primarily found in oats? Lignin, beta-glucan, chitin, fructooligosaccharides. Which is the answer? Your answer is B, beta-glucan. 39. Which analytical method is commonly used to measure dietary fiber content in foods? Success extraction, AOIC enzymatic gravimetric method, Jalal method, high performance liquid chromatography or HPLC. Which is the answer? Your answer is B, AOIC enzymatic gravimetric method. Question number 40. Which of the following is a naturally occurring source of resistant starch? A. Brown rice, B. Unripe bananas, C. Whole wheat, D. Almonds. Which is the answer? Your answer is B. Unripe bananas. What are resistant starch? Resistant starch is a carbohydrate that resists digestion in the small intestine and ferments in the large intestine. As the fibers ferment, they act as prebiotic and feed the good bacteria in the gut. There are several types of resistant starch. They are classified by their structure or source. More than one type of resistant starch can be present in a single food. Benefits of resistant starch. When starches are digested, they typically break down into glucose. You can see a particular box over here in the right corner of the slide. Study well these all the important points. Please study all these important points and write exam. All the best. 41. What is the primary byproduct of fiber fermentation in the gut? Methane, short chain fatty acids, ethanol, hydrogen gas. Which is the answer? Your answer is B, short chain fatty acids. 42. Which vitamin acts as coenzyme for the enzyme methionine synthase in DNA synthesis? Vitamin B6, B12, B1, vitamin C. Which is the answer? Your answer is B, vitamin B12 or cyanocobalamin. 43. Which vitamin is associated with the maintenance of rhodopsin in the retina? Vitamin D, A, E, K. Which is the answer? Your answer is B, vitamin A. 44. Niacin is synthesized in the body from which amino acid? Tryptophan, lysine, tyrosine, phenylalanine. Which is the answer? Your answer is A, tryptophan. 45. Hypervitaminosis D leads to which condition? Osteomalacia, hypercalcemia, scurvy, beriberi. Which is the answer? Your answer is B, hypercalcemia. 46. Which vitamin is crucial for the carboxylation of clotting factors 2, 7, 9 and 10? Vitamin B6, vitamin K, vitamin D, vitamin E. Which is the answer? Here answer is vitamin K. 47. Which vitamin can prevent spina bifida during pregnancy and taken as a supplement? Vitamin B12, folic acid, vitamin C, vitamin D. Which is the answer? Here answer is B, folic acid. Spina bifida is a condition that affects the spine and is usually apparent at birth. It can happen anywhere along the spine if the neural tube does not form properly or close all the way. The severity of spina bifida can range from mild to severe. 48. Pantothenic acid is a precursor for which essential coenzyme? Coenzyme Q, coenzyme A, NADPH, FMN. Which is the answer? Here answer is coenzyme A, option B. 49. Which vitamin deficiency can lead to peripheral neuropathy? Vitamin B1, B6, vitamin B12, all the above. Which is the answer? Your answer is D, all of the above. 50. The deficiency of vitamin B7 biotin is most commonly caused by consuming excessive amount of raw eggs, alcohol, red meat, coffee. Which is the answer? Your answer is a raw eggs. 51. Which vitamin plays a crucial role in preventing rickets? Vitamin C, vitamin D, vitamin A, vitamin K. Which is the answer? Your answer is B, vitamin D. 52. Which vitamin's deficiency is associated with Wernicke-Korsakoff syndrome? Vitamin B12, vitamin B1, B6, vitamin C. Which is the answer? Here answer is vitamin B1. Sorry, vitamin B1. Option B, vitamin B1. Wernicke-Korsakoff syndrome is an unusual type of memory disorder due to the lack of thiamine requiring immediate treatment. It most often happens in people with alcohol use disorder and malnutrition. 53. Vitamin B9 is chemically known as pyridoxine, folic acid, cobalamin, thiamine. Which is the answer? Your answer is B, folic acid. 54. What is the primary storage form of vitamin D in the human body? Calcitrol, cholecalciferol, ergocalciferol, 25-hydroxy vitamin D. Which is the answer? Your answer is D, 25-hydroxy vitamin D. First, it is converted to calcidiol or 25 OHD and in your liver. This is the storage form of the vitamin. Second, it is converted to calcitrol or 125 or OH2D, mostly in your kidneys, in the kidneys. 55. Which mineral is essential for the function of the antioxidant enzyme glutathione peroxidase? Iron, selenium, zinc, manganese. Which is the answer? Your answer is B, selenium. 56. What is the bioavailable form of iron predominantly found in plant-based foods? Heme ion, non-heme ion, ferritin-bound iron, iron-sulfur clusters. Which is the answer? Your answer is B, non-heme iron. Non-heme iron found in plant sources such as beans, nuts, dark chocolate, legumes, spinach, and fortified grains has about 2/3 of bioavailability of heme iron. 57. Zinc absorption in the human body is enhanced by the presence of which compound? Phytic acid, citric acid, oxalates, phosphate. Which is the answer? Your answer is B, citric acid. 50. Here right side you can see a box. Amino acids such as histidine and methionine and other low molecular weight ions such as EDTA and organic acids, example citric acid, are known to have a positive effect on zinc absorption and have been used for zinc supplements. 58. Which mineral is involved in ATP stabilization and acts as a cofactor for over 300 enzymes? Calcium, magnesium, potassium, sodium. Which is the answer? Your answer is B, magnesium. Magnesium is primarily found within the cell where it acts as a counter ion for the energy-rich ATP and nucleic acids. Magnesium is a cofactor in more than 300 enzymatic reactions. Magnesium critically stabilizes enzymes, including many ATP generating reactions. 59. A deficiency in iodine during pregnancy can lead to which condition in newborns? Anemia, cretinism, beriberi, scurvy. Which is the answer? Your answer is B, cretinism. 60. Which mineral plays a critical role in the clotting cascade by activating prothrombin? Potassium, sodium, calcium, phosphorus. Which is the answer? Your answer is C, calcium. Calcium is the necessary mineral for blood clotting, and the liver produces many clotting factors. Vitamin K is necessary for prothrombin synthesis, and the clot itself is primarily composed of a protein called fibrin. 61. Copper is a cofactor for which of the following enzymes involved in the electron transport chain? Cytochrome oxidase, C oxidase, ATP synthase, succinyl enzyme synthase fumarase, which is the answer. Here answer is a cytochrome C oxidase. 62. Which mineral deficiency is associated with the Keshan disease, a cardiomyopathy condition? Which is the answer? Selenium, magnesium, chromium, zinc. Which is the answer? Here answer is a selenium. Selenium deficiency. This has been named as the Keshan disease due to the geographical prevalence of a specific form of dilated cardiomyopathy in northeast China, in which the soil has a low selenium content. 63. Phytic acid in grains reduces the absorption of which minerals? A. Calcium and magnesium. B. Iron and zinc. C. Sodium and potassium. D. Both A and B. Which is the answer? Your answer is D. Both A and B. 64. Which trace mineral plays a significant role in glucose metabolism by enhancing insulin action? Chromium, selenium, vanadium, zinc. Which is the answer? Your answer is a chromium. Key minerals including magnesium, zinc and chromium are essential for optimal insulin function and glucose metabolism. Magnesium facilitates insulin action, zinc is vital for insulin storage and secretion, and chromium enhances insulin sensitivity. It enhances glucose metabolism by stimulation of insulin action in peripheral tissues. Chromium 3 supplementation benefits patients with comorbid T2DM and glucose intolerance. It also reverses obesity and insulin resistance caused by a high-fat diet. 65. Which mineral is a major constituent of hydroxyapatite, a component of bone? Sodium, potassium, calcium, magnesium. Which is the answer? Here answer is C, calcium. Hydroxyapatite HAP is a mineral composed of mainly of phosphorus and calcium, naturally occurring in human bones and teeth. The compound is one of the apatites. 66. Which among the following is a newly discovered vitamin by ADSMA Research and Publishing located in the small town of Lodha? A. Vitamin H. B. Vitamin MEPIA. C. Vitamin K. D. Zinc. Which is the answer? This is the current affair question. Newly discovered vitamin. It is known as vitamin MEPIA or methyl phosphinic acid. It is known as anti-aging vitamin. 67. Hemochromatosis is caused by excessive accumulation of which mineral? Copper, zinc, iron, selenium. Which is the answer? Your answer is C, iron. 68. Which mineral is critical for maintaining osmotic balance and nerve impulse transmission in cells? Calcium, sodium, phosphorus, magnesium. Which is the answer? Here answer is B, sodium. 69. The Wilson's disease results from a mutation affecting the metabolism of which mineral? Zinc, copper, selenium, manganese. Which is the answer? Your answer is B, copper. Wilson disease is a disorder of copper metabolism that affects men and women. About 1 person in 30,000 has the disorder. Affected people are homogeneous for the mutant recessive gene located on chromosome 13. Wilson disease is a rare genetic condition that occurs when your body accumulates too much copper, especially in the liver and brain. Your body needs a small amount of copper from food to stay healthy, but without treatment. Wilson disease can lead to high copper levels that cause life-threatening organ damage. 70. Which mineral is a structural component of DNA and RNA backbones? Iron, phosphorus, zinc, magnesium. Which is the answer? Your answer is B, phosphorus. 71. Manganese deficiency can impair which physiological process? Protein digestion, bond formation, oxygen transport, blood clotting. Which is the answer? Your answer is B, bond formation. Manganese is an essential trace element that is naturally present in many foods and available as a dietary supplement. Manganese is a cofactor for many enzymes including manganese superoxide dismutase, arginase and pyruvate carboxylase. Through the action of these enzymes, manganese is involved in amino acid, cholesterol, glucose and carbohydrate metabolism, reactive oxygen species scavenging, bond formation, reproduction and immune response. Manganese also plays a role in blood clotting and hemostasis in conjunction with vitamin K. 72. Sodium potassium pumps maintain cellular ion balance. For every 3 sodium ions expelled, how many potassium ions are taken into the cell? Your answer is B, 2. 73. Which adulterant is commonly used in milk to increase the thickness but poses a severe health hazard? Urea, formalin, starch, sodium bicarbonate. Which is the answer? Your answer is a urea. Urea, which is used to make milk white by increasing the non-protein nitrogen content. Formalin, boric acid also used, benzoic acid, and are also used for particular purpose for milk adulteration. Milk urea is an approximate indicator of the amount of crude protein in cow's diet. 74. Methanol yellow used to enhance the color of turmeric powder is harmful because it can cause respiratory disorders, liver damage, cancer, neural disorders. Which is the answer? Your answer is D, neural disorders. 75. Argemone oil is an adulterant in mustard oil. Which toxin is responsible for its harmful effects? Erucic acid, sanguinarine, aflatoxin, patulin. Which is the answer? Your answer is B, sanguinarine. Edible oil is adulterated with argemone oil, which exposes the consumer to sanguinarine and dihydrosanguinarine, the toxic alkaloid causing dropsy. 76. Which adulterant is used to polish grains and can cause kidney failure? Lead chromate, talc, paraffin, potassium bromate. Which is the answer? Your answer is A, lead chromate. Lead chromate may damage the nervous system too. Exposure may cause kidney and brain damage and anemia. Lead poisoning, also known as plumbism and satanism, is a type of metal poisoning caused by lead in the body. 77. The adulterant Sudan dye, often found in chili powder, is prohibited due to its neurotoxicity, carcinogenic properties, cardiotoxicity, mutagenic effects. Which is the answer? Here answer is B, carcinogenic properties. Sudan, CI, solvent yellow 14 or solvent orange or is a monoazo dye with the chemical formula of 1-phenyl-azo 2-naphthol. It can cause tumors in the liver or urinary bladder in mammals and is a potential mutagen and carcinogen for humans. 78. Which adulterant in green vegetables is known to cause cancer? Malachite green, copper sulfate, tartrazine, erythrocin. Which is the answer? Your answer is A, malachite green. 79. What is the chemical formula of the adulterant borax often found in sweets? Na2B4O7 10H2O, Na2CO3, NaClO3, Na3PO4. Which is the answer? Your answer is A, Na2B4O7 10H2O. 80. Which law governs food adulteration in India before FSSAI was established? A. The Essential Commodities Act. B. The Prevention of Food Adulteration Act 1954. C. Consumer Protection Act 1986. D. Food Safety and Standards Act 2006. Which is the answer? Your answer is B. The Prevention of Food Adulteration Act 1954. 81. Which test is used to identify adulteration in tea leaves with iron fillings? A. Floating test. B. Magnet test. C. Staining test. D. Fluorescence test. Which is the answer? Your answer is B. Magnet test. 82. Which amino acid is classified as glucogenic and ketogenic? A. Lysine. B. Leucine. C. Tryptophan. D. Threonine. Which is the answer? Your answer is D. Threonine. Amino acids that break down into carbohydrate precursors are called glucogenic. Those leading to acetyl coenzyme are called ketogenic. Leucine and lysine are the only exclusive ketogenic amino acids. Isoleucine, threonine, phenylalanine, tyrosine, and tryptophan are both glucogenic and ketogenic. 83. The Cori cycle links which two organs for glucose metabolism? Liver and brain, liver and skeletal muscle, kidney and heart, liver and intestine. Which is the answer? Think twice before answering the question. Your answer is B. Liver and skeletal muscle. The Cori cycle or lactate cycle was originally described in 1929 as a pathway that involved the conversion of glycogen to glucose and lactate in the skeletal muscle and the conversion back to glucose and glycogen from lactate in the liver, replenishing glycogen stores. 84. Which molecule is the most reactive form of reactive oxygen species? Hydrogen peroxide, superoxide anion, hydroxyl radical, singlet oxygen. Which is the answer? Your answer is C. Hydroxyl radical. Hydroxyl radical. 85. Which enzymatic antioxidant converts superoxide radicals into hydrogen peroxide? Glutathione peroxidase, superoxide dismutase, catalase, peroxidase. Which is the answer? Your answer is B. Superoxide dismutase SOD. 80. Superoxide dismutase is an important neuroprotective enzyme that converts harmful superoxide radicals to the less toxic hydrogen peroxide. 86. What is the primary role of glutathione in cells? Provide structural support, neutralize hydrogen peroxide, enhance enzymatic activity, regulate gene expression. Which is the answer? Your answer is B. Neutralize hydrogen peroxide. Glutathione peroxidase GPX1 is an intracellular antioxidant enzyme that enzymatically reduces hydrogen peroxide to water to limit its harmful effects. 87. Which vitamin acts as both an antioxidant and a cofactor for enzymes in collagen synthesis? Vitamin A, E, C, K. Which is the answer? Your answer is C. Vitamin C. 88. Which type of free radical is commonly generated during lipid peroxidation? Hydroperoxyl radical, hydroxyl radical, alkoxyl radical, nitric oxide. Which is the answer? Your answer is a hydroperoxyl radical. 89. Which antioxidant is responsible for regenerating vitamin E after it has been oxidized? Vitamin C, glutathione, beta-carotene, selenium. Which is the answer? Here answer is A. Vitamin C. It has been reported that vitamin C can synergistically regenerate vitamin E, resulting in an enhancement of antioxidant activity in membranes, thereby preventing them from being oxidized under free radical oxidative stress more efficiently. 90. Which compound is not classified as non-enzymatic antioxidant? Tocopherol, carotenoids, catalase, flavonoids. Which is the answer? Here answer is C. Catalase. A wide variety of non-enzymatic antioxidants like melatonin, resveratrol, L-ascorbic acid, carnitine, acetyl-N-acetyl, cysteine, cysteamine, quercetin, noveltin, lycopene, acetoside, mogroside, 5-cyanine, and laminarin have been used to supplement culture media. 91. The Fenton reaction involves which metal ion and leads to the production of hydroxyl radicals? A. Copper, B. Zinc, C. Iron, D. Manganese. Which is the answer? Your answer is C. Iron. Fenton's reaction is a named reaction in which hydrogen peroxide is converted into the hydroxyl free radical via a catalytic process. The hydrogen peroxide reactant is usually formed by the mitochondrial oxidative respiration. 92. Which antioxidant enzyme requires selenium as a cofactor? SOD, glutathione peroxidase, catalase, peroxidases. This is the answer. Your answer is B. Glutathione peroxidase. The antioxidant enzyme glutathione peroxidase consists of 4 selenium cofactors. These cofactors catalyze H2O2 and organic hydroperoxides. In animals, 4 different glutathione peroxidase isozymes are present. Glutathione peroxidase 1 works very effectively on hydrogen peroxide. Manganese is the cofactor for catalysis, peroxidases, and one type of superoxide dismutase. Superoxide dismutases are classified into 3 types based on their metal cofactors: iron SOD localized to chloroplast, manganese SOD localized to mitochondria, and third one is last one, copper zinc SOD localized to chloroplast, peroxisomes, and cytosols. 93. What is the primary role of carotenoids in preventing oxidative damage? inhibiting ROS production, quenching singlet oxygen, neutralizing hydroxyl radicals, regenerating regenerating vitamin C. Which is the answer? Here answer is B, quenching singlet oxygen. 94. Which amino acid is a precursor for glutathione synthesis? Arginine, cysteine, lysine, methionine. Which is the answer? Here answer is B, cysteine. Glutathione, the most abundant antioxidant in the cell, and its precursor amino acids: cysteine, glutamate, and glycine. 95. What is the primary function of catalase in the cell? Neutralize hydroxyl radicals, convert hydrogen peroxide to water and oxygen, repair damaged DNA, reduce lipid peroxides. Which is the answer? Here answer is B, convert hydrogen peroxide to water and oxygen. 96. Which lipid soluble antioxidant is embedded in the cellular membrane? Vitamin A, vitamin C, vitamin E, beta carotene. Which is the answer? Here answer is C, vitamin E. 97. Which laboratory assay is commonly used to measure total antioxidant capacity in foods? DPPH assay, ELISA assay, Western blot, Bradford assay. Which is the answer? Here answer is A, DPPH assay. 98. Which process in food preservation relies on antioxidants to prevent spoilage? Pasteurization, irradiation, oxidation inhibition, freeze drying. Which is the answer? Here answer is C, oxidation inhibition. 99. Who coined the term vitamin? Casimir Funk, William Harvey, Karl Landsteiner, Louis Pasteur. Which is the answer? Here answer is A, Casimir Funk. 100 question, last question. If you like this channel, please share and subscribe. Don't forget to click the bell button. Which vitamin acts as a coenzyme for transamination reactions in amino acid metabolism? Vitamin B6, vitamin B12, vitamin K, biotin. Which is the answer? Think twice before answering each and every question. Which is the answer over here? Your answer is A, vitamin B6. Which of the following stain is primarily used to detect bacterial endospores in foodborne pathogens? A, malachite green, B, nigrosin, C, carbol fuchsin, D, methylene blue. Here, which is the answer? Here, answer is A, malachite green. Second question. What is the role of tannic acid in flagella staining? It acts as a mordant to enhance flagella visibility. Option B, it fixes the flagella to the cell wall. Option C, it serves as the primary stain. Option D, it prevents decolorization. Which is the answer? Your answer is option A, it acts as a mordant to enhance flagella visibility. Third question. Which of the following staining technique is most suitable for identifying Mycobacterium species in contaminated milk? A, gram staining. Option B, Zeal-Nielsen staining or acid-fast staining. C, Schaefer-Fulton method or endospore staining. D, Indian ink staining. Which is the answer? Here answer is option B, Zeal-Nielsen staining. The Zeal-Nielsen stain is a special bacteriological stain used to identify acid-fast organisms, mainly mycobacteria. The reagents used are Zeal-Nielsen carbol fuchsin, acid alcohol, and methylene blue. Acid-fast bacteria will be bright red after staining. Fourth question. In food microbiology, which dye is used to visualize viable microorganisms under a fluorescence microscope? A, acridine orange. B, crystal violet. Option C, Sudan black bee. Option D, Congo red. Here, which is the answer? Here, answer is option A, acridine orange. Acridine orange is a nucleic acid binding fluorophore that can permeate viable cell membranes to selectively stain all nucleated cells. It is excited at wavelengths of approximately 502 nanometers when interacted with double-stranded DNA, emitting a green fluorescence with wavelengths of approximately 525 nanometer. Black Sudan B is used for the staining of wide variety of lipids such as phospholipids, sterols, and neutral triglycerides. Black Sudan B is not lipid specific like other Sudan dyes and can also be used for chromosome staining, Golgi apparatus, and leukocyte granules. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Why is Congo red staining used to detect biofilm forming bacteria in food processing environments? A. It stains extracellular polymeric substances EPS. B. It highlights bacterial cytoplasm. C. It binds to lipid granules. D. It detects peptidoglycan. Which is the answer? Your answer is option A. It stains extracellular polymeric substances EPS. Congo red reacts with beta glucans in exopolysaccharide producing black colonies, which helps in distinguishing EPS producer from non-EPS producer. Sixth question. Which staining method distinguishes between viable and non-viable bacterial cells? A. Gram staining. B. DAPI staining. C. Live dead backlight staining. D. Endospore staining. Which is the answer? Your answer is option C. Live dead backlight staining. Live dead backlight bacterial viability kits are easy to use fluorescent staining kits commonly used for assessing the viability of bacterial cells. These kits provide a two-color fluorescence assay of bacterial viability using green and red fluorescent dyes for use with imaging, flow and assays. This is an in vitrogen kit. Seventh question. Which is the most suitable staining technique for diphtheria? Albert staining, Neisser staining, Giemsa staining, Zeil-Nielsen staining. Here which is the answer? Here option is A. Albert staining. Albert's method of staining diphtheria cultures consists of staining a fixed smear for 1 minute, some laboratory stain for 5 minutes with a solution containing toluidine blue and malachite green, washing with water and applying Albert's iodine for 1 minute. The Neisser stain is a microbiological stain for visualizing the polar bodies in the cytoplasm of gram-positive bacteria. This staining is used to identify diphtheria bacteria. Cells are stained with methylene blue and crystal violet in acidic solution and then counterstained with chrysoidin solution. Next question, eighth question. Which stain is preferred for detecting lipid inclusions in bacterial cells found in food samples? Sudan black B, Negrosin, crystal violet, malachite green. Which is the answer? Your answer is a Sudan black B. Ninth question. In fluorescent staining, what is the function of FITC, fluorescein isothiocyanate? A. Stains DNA in bacterial cells. B. Binds to antibodies for specific detection. C. Highlights gram-positive cell walls. D. Detects spore-forming bacteria. Here which is the answer? Here answer is B. Binds to antibodies for specific detection. FITC is widely used to label proteins, antibodies, peptides, hormones, amine-modified oligonucleotides, and other amine-containing molecules with the green fluorescent dye fluorescein. 10th question. Which staining technique is suitable for identifying bacterial capsule production in foodborne pathogens? A. Giemsa staining. Option B. Indianing staining. C. Albert staining. D. Gram staining. Here which is the answer? Here answer is B. Indianing staining. The bacterial capsule is non-ionic. It can be stained with the usual manner. The capsule is stained with a negative staining technique. A small amount of culture is mixed with Indian ink and the resulting smear is stained with a dye low fluorescent methylene blue which penetrates the bacterial cell. 11th question. Why is heat fixation avoided in negative staining? Option A. Distorts the true shape of bacteria. Option B. It decolorizes gram-negative bacteria. C. It binds the stain irreversibly to the slide. D. It enhances cellular autofluorescence. Here answer is A. It distorts the true shape of bacteria. Since a negatively charged dye such as nigrosin is used for negative staining, the dye does not interact with the negatively charged cell due to charge repulsion. No heat fixation or harsh detail staining techniques are involved, so morphology and size are not altered by this protocol. 12th question. Which of the following dyes is used in both gram staining and endospore staining? Safranin. Option B, crystal violet. Option C, carbon fusion. Option D, malachite green. Which is the answer? Here answer is a safranin. 13th question. The phobia of number 13 is known as triskaidekaphobia. Triskaidekaphobia. What is the function of iodine in gram staining? A. It acts as mordant to fix the dye. Option B. It enhances the uptake of the primary stain. Option C. It dissolves lipids in gram-negative bacteria. D. It directly stains the bacterial capsule. Here which is the answer? Here answer is option A. It acts as a mordant to fix the dye. 14th question. Which staining method helps identify metachromatic granules in Corynebacterium species? A. Gram staining. B. Albert staining. C. Capsule staining. D. Endospore staining. Here answer is B. Albert staining. Albert stain is a kind of differential stain used for staining and identifying metachromatic granules. The granules appear purple-black when exposed to Albert stain and against light green cytoplasm. Albert stain only acts to stain metachromatic granules and no other granules in this bacteria. 15th question. What does the use of methylene blue in Loeffler's staining primarily identify? A. Spores of Bacillus species, granules in Corynebacterium diphtheriae, flagella in motile bacteria, biofilms in food pathogens. Which is the answer? Your answer is option B. Granules in Corynebacterium diphtheriae. Loeffler's methylene blue is a metachromatic stain, which means that certain biological materials stain in different color to that of the stain itself, and in case of Corynebacterium diphtheriae, the polyphosphate granules stain a deep blue-black color against the pale blue cytoplasm. 16th question. What is the primary use of Lugol's iodine in microscopic analysis contaminated food samples? Staining starch granules, staining bacterial cell walls. Option C. Staining spores in Bacillus species. D. Option D. Staining extracellular polysaccharides. Which is the answer? Here option is A. Staining starch granules. The iodine in Lugol's solution selectively binds to alpha-1,4 glucans found in polysaccharides such as glycogen. 17th question. What is the primary advantage of fluorescent antibody staining in detecting foodborne pathogens? A. It selectively stains spores without affecting vegetative cells. Option B. It identifies specific bacteria using antigen-antibody interactions. Option C. It provides a clear distinction between gram-positive and gram-negative bacteria. Option D. It detects bacterial biofilms with higher sensitivity than crystal violet. Which is the answer? Here answer is B. It identifies specific bacteria using antigen-antibody interactions. Question number 18. What is the unique characteristics of Leveson staining used in microbiology? It stains bacterial flagella. It highlights lipid inclusion. It differentiates acid-fast and non-acid-fast bacteria. It is used to detect capsules in bacterial cells. Here which is the answer? Here answer is option A. It stains bacterial flagella. The Leveson staining method was used to diagnose Helicobacter pylori infection. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. 19th question. Which stain is combined with lactophenol to visualize fungi in contaminated food samples? A. Cotton blue. B. Crystal violet. C. Sudan black. D. Carbol fusion. Which is the answer? Your answer is option A. Cotton blue. 20. Question number 20. In triple sugar iron agar contamination studies, which staining method is recommended to identify bacterial colonies with sulfur production? gram staining, Indian ink staining, lead acetate staining, methylene blue staining. Here which is the answer? Your answer is option C, lead acetate staining. The purpose of this test is to identify materials that contain sulfur. 21. Which staining technique is used to identify bacterial species that lack a typical cell wall, such as mycoplasma, in food-borne infections? A. Acridine orange staining. B. Dye staining. C. Neisser staining. D. Acid fast staining. Which is the answer? Your answer is option B. Dye staining. Dye staining was first developed as a specific stain to visualize cultivable mycoplasma colonies on agar surfaces, but the stain has subsequently been used to visualize spiroplasma too. 22. Question number 22. In 17th century, a widespread epidemic disease named black bane killed animals and humans. Which is the disease? A. Rabies. Option B. Shigellosis. Option C. Anthrax. Option D. Plague. Which is the answer? Here answer is option C. Anthrax. 23. Question number 23. Which staining technique detects Cryptosporidium oocytes in contaminated water used in food processing? A. Modified acid fast staining. B. Giemsa staining. C. Negative staining. D. Safranin staining. Which is the answer? Here answer is A. Modified acid fast staining. Question number 24. What is the significance of phenol in carbon fusion staining? Option A. It dissolves lipids in bacterial membrane. Option B. It enhances dye penetration into waxy cell walls. Option C. It decolorizes gram-negative bacteria. Option D. It acts as a mordant for the counter stain. Which is the answer? Your answer is option B. It enhances dye penetration into the waxy cell walls. Question number 25. What is the primary reason for using malachite green in spore staining techniques for foodborne pathogens? Option A. It stains the vegetative cell intensely. Option B. It stains spores while excluding vegetative cells. Option C. It enhances fluorescence in bacterial DNA. Option D. It decolorizes non-spore forming cells. Which is the answer? Your answer is option B. It stains spores while excluding vegetative cells. 26. In the presence of heavy metals, which staining technique is often used for the electron microscopic visualization of bacterial structures? Option A. Negative staining. Option B. Acid fast staining. Option C. Gram staining. Option D. Safranin staining. Here which is the answer? Here answer is option A. Negative staining. The most commonly used negative staining reagents are uranyl acetate and uranyl formate. 27. Which type of staining is used to identify bacterial colonies producing biofilms in contaminated food processing environments? Option A. Congo red staining. Option B. Endospore staining. Option C. Capsule staining. Option D. Acridine orange staining. Here which is the answer? Here answer is option A. Congo red staining. To determine the qualitative analysis of biofilm formation, the Congo red agar method was used. Black colored colonies were considered biofilm positive and red pink colored colonies were considered biofilm negative. 28 question. Which staining method is uniquely suited to distinguish between Bacillus cereus and Bacillus subtilis spores in food samples? A. Wid's conclave staining. B. Gram staining. C. Negative staining. D. Dye staining. Which is the answer? Here answer is option A. Wid's conclave staining. 29. Which staining procedure can be adapted to detect E. coli O157H7 in contaminated food samples? A. Immunofluorescence staining. B. Acid fast staining. C. Indian ink staining. D. Endospore staining. Which is the answer? Here answer is option A. Immunofluorescence staining. Bacterial cells in samples were stained with CTC and FITC labeled anti-E. coli O157H7 direct fluorescent antibody. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notification. Question number 30. Which unique feature distinguishes endospore staining with Schaefer-Fulton method from other spore staining techniques? A. Use of heat to drive malachite green into spores. Option B. Application of counterstain to color vegetative cells red. Option C. Selective binding to spores forming bacteria. Option D. Visualization of lipids within the spore coat. Which is the answer? Here answer is option A. Use of heat to drive malachite green into spores. 31. Which staining technique can reveal the glycocalyx layer in biofilm forming bacteria? Ruthenium red staining. Option B. Acid fast staining. Option C. Alcyon blue staining. Option D. Congo red staining. Which is the answer? Your answer is option A. Ruthenium red staining. Ruthenium red has been used to show the ultrastructural details of bacterial glycocalyces. 32. Question number 32. What is the primary counterstain in Wheat's Conklin endospore staining? Safranin, malachite green, crystal violet, carbol fusion. Which is the answer? Your answer is option A. Safranin. 33. Which foodborne pathogen is most likely to cause post-infectious Guillain-Barré syndrome? Salmonella enteritica, Listeria monocytogenes, Campylobacter jejuni, Shigella dysentery. Which is the answer? Your answer is Campylobacter jejuni. One of the most common risk factors for GBS is infection with the bacteria Campylobacter jejuni, which causes gastroenteritis, including nausea, vomiting, and diarrhea. Sometimes surgery will trigger the syndrome, and in very rare cases, certain vaccinations may increase the risk of GBS. Question number 34. What is the primary virulence factor of E. coli O157H7 that leads to hemolytic uremic syndrome or HUS? Option A. Enterotoxins. Option B. Shiga toxins STX1 and STX2. Option C. Lipopolysaccharide. Option D. Heat stable toxin. Here which is the answer? Here answer is option B. Shiga toxin STX1 and STX2. Next question 35. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Which spore forming pathogen is commonly associated with improperly canned foods due to its production of neurotoxins? A. Bacillus cereus. B. Clostridium botulinum. C. Clostridium perfringens. D. Listeria monocytogenes. Here which is the answer? Here answer is option B. Clostridium botulinum. Question number 36. Which of the following pathogens can survive and multiply in refrigerated temperatures, making it a significant hazard in cold storage? A. Clostridium botulinum. B. Salmonella enterica. C. Listeria monocytogenes. D. Campylobacter jejuni. Which is the answer? Here answer is option C. Listeria monocytogenes causes listeriosis. 37. Who is known as the father of microscopy? Anton von Leeuwenhoek, Louis Pasteur, Edward Jenner, or Ehrlich? Your answer is option A. Anton von Leeuwenhoek. 38. Question number 38. What is the target organ of hepatitis A virus in cases of foodborne transmission? Small intestine, liver, kidneys, pancreas. Your option B. Answer is option B. Liver. 39. Which foodborne pathogen or pathogens is or are the leading cause of reactive arthritis, Rieter's syndrome? A. Campylobacter chlamydia. B. Clostridium difficile and E. coli. C. Salmonella, Shigella, and Yersinia. All the above. Which is the answer? Here answer is option D. All the above. 40. What is the infectious dose of norovirus, one of the most common viral foodborne pathogens? 18 to 1000, 100 to 1000, 1000 to 10,000, 10,000 to 1 lakh particles. Here answer is option A, 18 to 100 particles. Noroviruses have an extremely low infectious dose, greater than or equal to 18 viral particles with their low infectious dose of 18 to 1000 virus particles. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Next question 41. Which of the following pathogens produces the heat-stable emetic toxin cereulide? Option A. Clostridium perfringens. Option B. Bacillus cereus. Option C. Staphylococcus aureus. Option D. Vibrio cholerae. Which is the answer? Your answer is option B. Bacillus cereus. Cereulide is a toxin produced by Bacillus cereus during the growth phase in food, particularly starchy foods like rice and pasta. It is resistant to heat and pH variations, causing an emetic syndrome characterized by nausea and vomiting, similar to Staphylococcus aureus related foodborne illness. Question number 42. Which pathogen is known for causing shellfish poisoning through the production of thermostable enterotoxins? Option A. E. coli. Option B. Vibrio parahemolyticus. Option C. Clostridium botulinum. Option D. Shigella dysentery. Which is the answer? Your answer is option B. Vibrio parahemolyticus. VP is a bacterium that likes to live in salt water and is commonly found in estuaries and coastal waters where fish and shellfish live. VP can cause food poisoning symptoms like abdominal pain, diarrhea, vomiting, and sometimes mild fever. The disease is usually mild to moderate. Next question 43. Which foodborne pathogen is most commonly associated with raw or undercooked pork and can cause pseudoappendicitis? Option A. Yersinia enterocolitica. Option B. Clostridium difficile. Option C. Salmonella typhimurium. Option D. Listeria monocytogenes. Which is the answer? Your answer is option A. Yersinia enterocolitica. Yersinia enterocolitica is a bacterial species in the family Enterobacteriales that cause enterocolitis, acute diarrhea, terminal ileitis, mesenteric lymphadenitis, and pseudoappendicitis. But if it spreads systemically, can also result in fatal sepsis. Pseudoappendicitis is often caused by bacterial infection, commonly Yersinia enterocolitica or Campylobacter. 44. What is the primary mode of transmission of Cyclospora cayetansis in foodborne outbreaks? A. Direct contact with infected individuals. B. Contaminated fresh produce. C. Undercooked meat. D. Contaminated shellfish. Which is the answer? Your answer is option B. Contaminated fresh produce. Cyclospora cayetansis is a parasite that has emerged as an important food pathogen during the past 20 years. It causes prolonged and recurrent watery diarrhea in patients who are not properly diagnosed and treated. 45. Which toxin produced by Clostridium perfringens is primarily responsible for food poisoning? Alpha toxin, beta toxin, epsilon toxin, CPE or Clostridium perfringens enterotoxin. Here, answer is option D, CPE. Next question. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. 46. Which of the following pathogens has the ability to form biofilms on food contact surfaces, increasing its resistance to sanitizers? A. Salmonella typhi. B. E. coli O15787. C. Listeria monocytogenes. D. Vibrio cholerae. Which is the answer? Your answer is option C. Listeria monocytogenes. 47. Which of the following is a zoonotic foodborne pathogen transmitted via unpasteurized dairy products? A. Brucella abortus. B. Salmonella enterica. C. Shigella sonni. D. Clostridium botulinum. Which is the answer? Your answer is A. Brucella abortus. 48. Which of the following foodborne pathogens is most likely to cause infection from consumption of unpasteurized apple cider? A. Vibrio cholerae. B. E. coli 15787. C. Campylobacter jejuni. D. Vibrio parahemolyticus. Which is the answer? Your answer is option B. E. coli 15787. The most common pathogens in unpasteurized juice are toxidenic E. coli, Salmonella, hepatitis A, and Cryptosporidium. 49. Question number 49. What distinguishes Clostridium botulinum group 1 from group 2 strains? A. Group 1 strains are proteolytic and group 2 strains are non-proteolytic. Group 1 strains grow at lower temperatures than group 2 strains. C. Group 1 strains produce type E toxin while group 2 strains produce type A toxin. D. Group 1 strains are non-spore forming unlike group 2 strains. Which is the answer? Your answer is option A. Group 1 strains are proteolytic and group 2 strains are non-proteolytic. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. 50. Which pathogens cause foodborne illness through cross-contamination of raw chicken and contaminated cutting boards? A. Campylobacter jejuni. B. Escherichia coli. C. Clostridium perfringens. D. Vibrio vulnificus. Your answer is option A. Campylobacter jejuni. Question number 51. Which toxin of Staphylococcus aureus is associated with rapid onset food poisoning symptoms? A. Alpha toxin. B. Beta toxin. C. Enterotoxin. D. Pandan-valentine leucocidin or PVL. Which is the answer? Your answer is C. Enterotoxin A. Pandan-valentine leucocidin PVL is a cytotoxin produced by Staphylococcus aureus that causes leukocyte destruction and tissue necrosis. Question number 52. Which foodborne pathogen has a unique corkscrew motility due to its polar flagella? A. Helicobacter pylori. B. Campylobacter jejuni. C. Treponema pallidum. D. Vibrio cholerae. Your answer is B. Campylobacter jejuni. Question number 53. What is the primary reason Chronobacter sagazake is a critical concern in powdered infant formula? A. Production of endotoxins. B. Resistance to pasteurization. C. Ability to survive desiccation and low water activity. D. Spore formation. Which is the answer? Your answer is option C. Ability to survive desiccation and low water activity. Chronobacter sagazake or Chronobacter and other species are germs found naturally in the environment. Chronobacter can live in dry foods like powdered infant formula, powdered milk, herbal teas, and starches. It has also been found in contaminated feeding items like breast pump equipments. 54. Which foodborne pathogen employs a type 3 secretion system to inject effector proteins into host cells? A. Shigella fluxneri. B. Listeria monocytogenes. C. Bacillus cereus. D. Vibrio cholerae. Which is the answer? Your answer is option A. Shigella fluxneri. Type 3 secretion systems are highly conserved in a wide variety of plant and animal pathogenic bacteria, including Salmonella, Shigella, Yersinia, Pseudomonas, enteropathogenic E. coli, Erwinia, and Rhizobium. 55. Which Yersinia species is most commonly implicated in foodborne outbreaks due to contaminated dairy or pork products? Yersinia pestis, Yersinia pseudotuberculosis, Yersinia enterocolitica, Yersinia rucerae. Here which is the answer? Here answer is C. Yersinia enterocolitica. 56. Which serotype of E. coli is classified as an enteroaggressive pathogen? O104H4, O157H7, O111H8, O145H28. Which is the answer? Here answer is option A. O104H4. A novel strain of E. coli bacteria caused a serious outbreak in foodborne illness focused in northern Germany in May through June 2011. The illness was characterized by bloody diarrhea with a high frequency of serious complications, including hemolytic uremic syndrome, a condition that requires urgent treatment. It is an emerging E. coli pathogen. 57. Which of the following pathogens can utilize quorum sensing to regulate the production of virulence factors? Salmonella enterica, E. coli, Vibrio cholerae, Clostridium botulinum. Which is the answer? The answer is Vibrio cholerae. Next question. Which foodborne pathogen is known for its high infectivity and ability to cause outbreaks on cruise ships? Norovirus, Salmonella typhi, Campylobacter jejuni, Vibrio cholerae. Which is the answer? Here answer is option A, norovirus. Transmission typically occurs in crowded spaces where particles can float around the air, making cruise ships a relatively easy place for norovirus outbreaks to occur. William Schaffner, a professor of infectious diseases at Vanderbilt University, explained. 59. Which Salmonella serotype is most frequently isolated from poultry-related foodborne outbreaks? A. Typhi, B. Enteritis, C. Cholerasis, D. Typhimurium. Which is the answer? Here answer is option B, enteritis. Among the Salmonella isolate from the broiler sample, Salmonella enteritis was the main dominant serotype, followed by Salmonella Indiana, Salmonella Kentucky, and Salmonella Newport. 60. Question number 60. Which bacterial genus includes species that cause scombroid poisoning by producing histamine in improperly stored fish? Morganella, Shigella, Aeromonas, and Enterobacter. Which is the answer? Your answer is option A, Morganella. 61. Question number 61. Which component in McConkie agar act as a pH indicator? Neutral red, bromothymol blue, phenol red, crystal violet. Which is the answer? Your answer is option A, neutral red. 62. Which selective agent in Baird-Parker agar inhibits non-staphylococcal bacteria? Potassium telluride, lithium chloride, sodium azide, both A and B. Your answer is A, both A and B. Baird-Parker agar is used for the isolation and differentiation of coagulase positive staphylococci in food and pharmaceuticals according to the Baird-Parker method. As a nitrogen source for the organism, casein, peptone, and meat extract are added to this medium. 63. What is the purpose of cysteine in cysteine lactose electrolyte deficient agar? A. Enhances lactose fermentation. B. Maintains low oxidation reduction potential. C. Inhibits gram-positive organisms. D. Acts as an energy source for yeast growth. Which is the answer? Your answer is option B. Maintains low oxidation reduction potential. 64. Why is mannitol salt agar used for Staphylococcus species isolation? D. Mannitol fermentation and high salt concentration. B. Neutral pH and rich nutrients. C. High lactose content. D. Presence of bile salts. Which is the answer? Your answer is A. Mannitol fermentation and high salt concentration. 65. Which medium is used for enumeration of lactic acid bacteria in fermented foods? MRS agar, EMB agar, Sabouraud's dextrose agar, tryptic soy agar. Which is the answer? EMB agar specially for E. coli, Sabouraud's dextrose agar specially designed for fungus, tryptic soy agar commonly used for different types of bacteria, varying range. Here MRS is the answer, option A. Demon Rogas sharp agar, often abbreviated to MRS, is a selective culture medium designed to favor the luxuriant growth of lactobacillus for lab study. Developed in 1960, this medium was named for its inventors John Cornelius Demon, Morrison Rogosa, and Margaret Elizabeth Shaw. 66. What role does sorbitol play in sorbitol McConkie agar? A. Differentiates E. coli O157H7. B. Selects for gram-positive bacteria. C. Acts as a general carbon source. D. Inhibits yeast growth. This is the answer. Your answer is option A. Differentiates E. coli O157H7. McConkie sorbitol agar is a selective differential medium used in the detection of sorbitol negative E. coli such as serotype O157H7. The majority of outbreaks of hemorrhagic colitis have been caused by E. coli serotype O157H7. Question number 67. Which medium is most suitable for the isolation of Campylobacter species? Skyrov agar, TCBS agar, Sabar SDA, EMB agar. Which is the answer? Here answer is option A, Skyrov agar. 68. Which medium is used to test bacterial motility? SIM medium, nutrient agar, XLD agar, MRS agar. Which is the answer? Here answer is option A, SIM medium, sulfide indole motility medium. 69. What is the purpose of reducing agents in thioglycolate medium? A. Create anaerobic conditions. B. Neutralize acids. C. Inhibit gram-positive organisms. D. Stimulate bacterial motility. Which is the answer? Your answer is option A. Create anaerobic conditions. 70. What is the primary use of potato dextrose agar? Fungal isolation, selective growth of gram-positive bacteria, enumeration of coliforms, detection of salmonella. Which is the answer? Your answer is option A. Fungal isolation. 71. Which component in brilliant green agar inhibits gram-positive organisms? Brilliant green dye, bile salt, sodium thiosulfate, peptone. Which is the answer? Your answer is A. Brilliant green dye. 72. What is the role of ferric ammonium citrate in XLD agar? Detect hydrogen sulfide production, provide a nitrogen source, neutralize acids, enhance bacterial motility. Which is the answer? Here answer is option A. Detect hydrogen sulfide production. The addition of sodium thiosulfate, ferric ammonium citrate, and sodium deoxycholate created the more selective medium XLD agar. This medium was found to be satisfactory for the isolation of Shigella and Providencia species, as well as proving to be an effective differential media. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. 73. Which sterilization method uses a combination of pressure and carbon dioxide to achieve microbial inactivation? A. Ethylene oxide sterilization. B. Supercritical CO2 sterilization. C. High pressure processing. D. UVC radiation. Your answer is option B. Supercritical carbon dioxide sterilization. It is the most effective sterilization modality when it comes to penetration. Similar to ETO, SCCO2 is a low temperature sterilization modality with an operating temperature of 35 degrees Celsius. Unlike ETO, SCCO2 does not require an outgassing period of sorry of tens of hours. It requires no outgassing time. Question number 74. In which sterilization technique are Bacillus stereothermophilus spores commonly used as biological indicators? Dry heat sterilization, steam sterilization or moist heat sterilization, ethylene oxide sterilization, gamma irradiation. Your answer is option B, steam sterilization. 75. What is the main mechanism of action for ionizing radiation in sterilization? Protein denaturation, oxidative stress degeneration, DNA strand breaks, membrane disruption. Your option is C, DNA strand breaks. 76. Which of the following techniques is most suitable for sterilizing heat-sensitive nutrient media? Autoclaving, membrane filtration, dry heat sterilization, chemical sterilization. Which is the answer? Your answer is B, membrane filtration. 77. Which microorganism is the standard reference organism for validating sterilization by gamma radiation? Bacillus subtilis, Clostridium botulinum, Deinococcus radiodurans, and Bacillus pumilus? All the above. Your answer is option C, Deinococcus radiodurans and Bacillus pumilus. Deinococcus radiodurans is the most familiar radiation-resistant microorganism due to its rather extreme resistance to DNA damage. When sterilizing medical devices, the radiation dose is entirely dependent on the bioburden of the device. Bacillus pumilus ATCC number 27142 spores were used as reference standard in evaluating of gamma radiation sterilization process due to its high radiation resistance. 78. Which is the least effective sterilization method for spores of Clostridium? Ethylene oxide, UVC radiation, steam sterilization, gamma irradiation. Which is the answer? Your answer is option B, UVC radiation. 79. Which sterilization method generates hydroxyl radicals for microbial inactivation? High pressure processing, cold plasma, supercritical fluid sterilization, microwave sterilization. Which is the answer? Your answer is B, cold plasma. 80. Question number 80. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Which pH condition enhances the effectiveness of chemical sterilants like hypochlorite? Neutral pH, slightly acidic pH, highly acidic pH, slightly alkaline pH. Which is the answer? Your answer is option D, slightly alkaline pH. Question number 81. Which sterilization process is described as surface sterilization without heat or chemicals? ethylene oxide, high pressure processing, cold plasma, ionizing radiation. Which is the answer? Here answer is option C, cold plasma. Question number 82. Which sterilization process relies on the generation of reactive oxygen species to inactivate microorganisms? A. Parasitic acid. B. Cold plasma. C. Ozone treatment. D. UVC radiation. Which is the answer? Your answer is option C. Ozone treatment. 83. Which sterilization technique is most effective for removing bacterial endotoxins? Dry heat sterilization, steam sterilization, gamma irradiation, ethylene oxide. Which is the answer? Your answer is a dry heat sterilization. To depyrogenate glassware, a temperature of 250 degrees Celsius and a holding time of 30 minutes is typically applied to achieve a reduction of endotoxin levels by a factor of 1000. This is the most common type of depyrogenation undertaken in pharmaceutical industry. 84. What is the recommended dose of gamma radiation for sterilization of medical and food contact surfaces? Your answer is option D, 25 kilogray. 85. Which microorganism is most likely to grow in foods preserved with high salt concentrations? A. Vibrio parahemolyticus. B. Staphylococcus aureus. C. Halobacterium salinaro. D. E. coli. Which is the answer? Your answer is B. Staphylococcus aureus. Staphylococcus aureus not only is prevalent in hospital settings but also has survival efficiency in hostile environments. It successfully survives in high saline up to 25% sodium chloride, which is commonly found in canned foods. 86. Which spoilage microorganism is most commonly associated with vacuum packaged seafood? Clostridium botulinum type E, Listeria monocytogenes, Vibrio vulnificus, Pseudomonas fluorescens. Is the answer? Your answer is option A, Clostridium botulinum type E. 87. What is the effect of adding sodium nitrate to processed meats? Reduce lipid oxidation, inhibits Clostridium botulinum spores, C. Enhances water activity, D. Increases pH stability. Which is the answer? Your answer is option B, inhibits Clostridium botulinum spores. 88. Which phase of the bacterial growth curve is most sensitive to antibiotics targeting cell wall synthesis? Lag phase, log phase, tertiary phase, death phase. Which is the answer? Your answer is option B, log phase. Cells in the log phase are preferentially used for industrial application and research work. The log phase is also the stage where bacteria are the most susceptible to the action of disinfectants and common antibiotics that affect protein, DNA, and cell wall synthesis. 89. What is the primary reason for cell death during the death phase of the growth curve? A. Accumulation of toxic metabolic byproducts, nutrient excess, genetic mutations, increased cell density. Which is the answer? Your answer is option A. Accumulation of toxic metabolic byproducts. 90. In a bacterial growth curve, what event occurs during the lag phase? Cell division, RNA synthesis, production of secondary metabolites, maximum growth rate achieved. Which is the answer? Your answer is option B. RNA synthesis. During lag phase, bacteria adapt themselves to growth conditions. It is the period where the individual bacteria are maturing and not yet able to divide. During the lag phase of the bacterial growth cycle, synthesis of RNA, enzymes, and other molecules occurs. RNA synthesis during lag phase is prerequisite for the production of proteins required to equip the bacteria for exponential growth. Question number 91. Which factor most limits bacterial growth during the tertiary phase? pH change, oxygen depletion, competition for nutrients, all of the above. Which is the answer? Your answer is all of the above, D. 92. What determines the slope of the line during the exponential phase? Nutrient availability, nutrient availability. B. Specific growth rate of the organism. C. Initial cell density. D. Generation time. Which is the answer? Your answer is option B. Specific growth rate of the organism. 93. What happens to the metabolic activity of cells during the lag phase? It decreases sharply, it remains dormant, it increases as cells prepare for division, it stops completely. Which is the answer? Your answer is option C. It increases as cells prepare for division, lag phase. There is little no change in number of microbes, but their metabolic activity is high. The microbes adjust to the specific conditions of the growth media and enzymes are induced to metabolize the substrates that are available. 94. In which growth phase is secondary metabolite production most prominent? Lag phase, log phase, stationary phase, death phase. It is the answer. Your answer is option C, stationary phase. Secondary metabolites are synthesized during the end or near the stationary phase of growth and are not involved in cell growth, development or reproduction. 95. What is the term for continuous culture system maintaining cells in the exponential phase? Batch culture, chemostat, fed batch system, turbidostat. Which is the answer? Your answer is option B, chemostat. 96. Which phase is characterized by equal rates of cell growth and death? Lag phase, log phase, stationary phase, death phase. Which is the answer? Here answer is option C, stationary phase. 97. Which scientist first observed and described the phenomenon of dioxic growth? Louis Pasteur, Jacques Monod, Robert Koch, Alexander Fleming. Which is the answer? Here answer is option B, Jacques Monod. 98. What is the primary molecular mechanism responsible for dioxic growth in bacteria? Catabolite repression, osmotic regulation, feedback inhibition, gene amplification. Which is the answer? Here answer is option A, catabolite repression. Catabolite repression allows microorganisms to adapt quickly to a preferred carbon and energy source first. This is usually achieved through inhibition of synthesis of enzymes involved in catabolism of carbon sources other than the preferred one. 99. Which regulatory protein is directly involved in the catabolite repression mechanism during dioxic growth in E. coli? LACL, CRP, FTSZ, SPO0A. Which is the answer? Here answer is B, CRP, CMP receptor protein. 100. Which day is celebrated as the World Microbiome Day 2024? June 27, September 16, July 27, April 8. Which is the answer? Here answer is option A, June 27. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. First question. Which microorganism is responsible for the fermentation of paneer? Option A, Enterococcus species. Option B, yeast. Option C, lactic acid bacteria. Option D, Saccharomyces cerevisiae. Which is the answer? Here answer is option C, lactic acid bacteria. Second question. Which bacterium is primarily responsible for the fermentation of yogurt? A, Lactobacillus acidophilus. B, Lactobacillus delbrueckii subspecies bulgaricus. Option C, Streptococcus thermophilus. D, both B and C. Here answer is D, both B and C. Third question. What is the key microorganism used for the fermentation of kombucha tea? A, Saccharomyces cerevisiae. B, Acetobacter xylenum. C, Lactobacillus plantarum. D, Clostridium acetobutylicum. Which is the answer? Which is the key microorganism used? Here answer is B, Acetobacter xylenum. Fourth question. Which yeast species is primarily involved in the fermentation of idli and dosa batter? A, Saccharomyces cerevisiae. B, Candida albicans. C, Aspergillus oryzae. D, Saccharomyces pastorianus. Your answer is option A, Saccharomyces cerevisiae. Fifth question. Which microorganism is most commonly associated with mesofermentation? A, Penicillium species. B, Aspergillus oryzae. C, Bacillus cereus. D, Enterococcus faecalis. Your answer is option B, Aspergillus oryzae. Sixth question. Which of the following microorganisms is used in the production of Srikhand? A, Lactobacillus acidophilus. B, Lactobacillus bulgaricus. C, Lactobacillus lactis and bifidobacterium. All the above. Which is the answer? Here answer is option D, all of the above. Food Safety Officer book is available with 1000 MCQs from all sections. Contact number 9645093283, 8086205050. Amazon link is available in the description. Seventh question. In the fermentation of bread, which yeast is the most important for leavening? Saccharomyces cerevisiae, Candida albicans, Saccharomyces pastorianus, Lactobacillus rhamnosus. Is the answer? Here answer is a Saccharomyces cerevisiae. Eighth question. Which microorganism is responsible for the fermentation of idli batter? Saccharomyces cerevisiae and Lactobacillus. Lactobacillus acidophilus and Streptococcus thermophilus, Bacillus cereus and Lactobacillus rhamnosus, Bacillus subtilis and Aspergillus oryzae. Here answer is A. Saccharomyces cerevisiae and Lactobacillus. Ninth question. What is the spoilage microorganism commonly associated with improperly fermented paneer? Bacillus cereus, Lactobacillus acidophilus, Escherichia coli, Pseudomonas species. Which is the answer? Here answer is A. Bacillus cereus. 10th question. Which year was designated as the International Year of Millets by the United Nations? 2004, 2023, 2006, 2007. Which is the answer? Your answer is 2023. Food and Agriculture Organization and United Nations has recognized 2023 as IYM, International Year of Millets. 11th question. Bacterial black discoloration of butter is caused by Pseudomonas nigrifolians, Pseudomonas fetida, Penicillium notatum, Pseudomonas aeruginosa. Which is the answer? Your answer is option A. Pseudomonas nigrifolians. 12th question. Black fungal discoloration of butter is caused by Aspergillus niger, Cladosporium herbarum and torula, Penicillium species, Geotrichum candidum. Which is the answer? Your answer is option B. Cladosporium herbarum and torula. 13th question. Brown fungal discoloration of butter is caused by Aspergillus species, Cladosporium herbarum, Penicillium species, Geotrichum candidum. Which is the answer? Your answer is option A. Aspergillus species. 14th question. Green fungal discoloration of butter is caused by Aspergillus species, Cladosporium herbarum, Penicillium species, Geotrichum candidum. Which is the answer? Your option is C. Penicillium species. 15th question. Orange and yellow fungal discoloration of butter is caused by Aspergillus species, Cladosporium herbarum, Penicillium species, Geotrichum candidum. Which is the answer? Your answer is D. Geotrichum candidum. 16th question. Reddish pink fungal discoloration of butter is caused by Aspergillus species, Cladosporium herbarum, Fusarium, Geotrichum candidum. Which is the answer? Your answer is C. Fusarium. 17th question. Pink fungal discoloration of butter is caused by Aspergillus species, Cladosporium herbarum, Rhodotorula species, Geotrichum candidum. Which is the answer? Your answer is C. Rhodotorula species. 18th question. Putrefactive taint of butter is caused by Pseudomonas putrefaciens, coliforms, Flavobacterium malolioris, all the above. Which is the answer? Your answer is all the above. 19th question. Musty taint in cold stored butter is caused by Achromobacter, coliforms, Flavobacterium malolioris, all the above. Which is the answer? Your answer is A. Achromobacter. 20th question. Apple-like aroma in butter is caused by Pseudomonas putrefaciens, coliforms, Flavobacterium lactis, all the above. Which is the answer? Your answer is C. Flavobacterium lactis. 21. Question number 21. Skunk-like flavor in butter is caused by Pseudomonas mephitica, coliforms, Flavobacterium lactis, all the above. Which is the answer? Your answer is A. Pseudomonas mephitica. 22. Fishy taint in butter is caused by Pseudomonas ichthyosnea, coliforms, Flavobacterium lactis, all the above. Which is the answer? Your answer is A. Pseudomonas ichthyosnea. Fishy taint. Fishiness is developed due to the decomposition of lecithin to trimethylamine by microbial activity. 23. An outbreak in USA in 1970 was linked to whipped butter, and to the butter from which the whipped butter was made is due to which microorganism? Option A. Pseudomonas ichthyosnea, B. Coliforms, Staphylococcus aureus, all the above. Which is the answer? Your answer is C. Staphylococcus aureus. The presence of staphylococcal enterotoxin A was demonstrated in both butters. 24. In 1995, an outbreak of enteritis by eating garlic butter in USA affected 30 people due to consumption of garlic butter. Which is the causative organism? Which is the answer here? The answer is Campylobacter jejuni. Campylobacter. In 1995, an outbreak of Campylobacter jejuni enteritis in USA affected 30 people due to consumption of garlic butter, and it could survive in butter without garlic for 13 days at 5 degrees Celsius. 25. Which microorganism is used to produce the blue veins in Roquefort cheese? Penicillium camemberti, Penicillium roqueforti, Aspergillus flavus, Geotrichum candidum. Which is the answer? Your answer is B. Penicillium roqueforti. 26. Which bacterium is commonly responsible for spoilage in cheese by producing gas and cracks? Clostridium triobutyricum, Pseudomonas fluorescens, Bacillus subtilis, Lactobacillus casei. This is the answer. Your answer is a Clostridium triobutyricum. 27. What spoilage microorganism in yogurt produces an off-putting fruity odor? Which spoilage microorganism? A. Saccharomyces cerevisiae, Candida kefir, Geotrichum candidum, Pseudomonas fragae. Your answer is D. Pseudomonas fragae. 28. What is the key fermenting microorganism that leavens idli batter? Saccharomyces cerevisiae, Leuconostoc mesenteroids, Pediococcus acidulacti, Bacillus subtilis. Which is the answer? Your answer is B. Leuconostoc mesenteroids. 29. Heat resistant yeast isolated from fermented pasteurized pineapple juice are Candida pelliculosa and Cloquera apis. B. Saccharomyces cerevisiae and Candida tropicalis. C. Torula and Candida albicans. D. All the above. Which is the answer? Your answer is A. Candida pelliculosa and Cloquera apis. 30. Which among the following yeast can survive greater than 10 of high ethanol concentration? 10% of high ethanol concentration. Schizosaccharomyces pompe, Zygosaccharomyces bailey, Zygosaccharomyces fermentati. D. All the above. Which is the answer? Your answer is option D. All the above. 31. What type of spoilage occurs in ghee due to the activity of lipase producing bacteria? Rancidity, proteolysis, putrefaction, fermentation. A. Rancidity. Choose the microorganisms involved in chakka production from the following options: Lactobacillus lactis subspecies lactis, Lactobacillus lactis variety diacetyl lactis, E. coli. D. Both A and B. Which is the answer? Your answer is D. Both A and B. 33. Which yeast is responsible for leavening bread by producing carbon dioxide? Saccharomyces cerevisiae, Candida milleri, Saccharomyces baianus, Aspergillus oryzae. Which is the answer? Your answer is option A. Saccharomyces cerevisiae. 34. Which spoilage microorganism is associated with the formation of rop in bread? So, ropinus in bread is caused by Bacillus subtilis, Clostridium perfringens, Pseudomonas aeruginosa, Rhizopus stolonifer. Which is the answer? Your answer is A. Bacillus subtilis. Bacillus amyloliquefaciens, Bacillus subtilis, Bacillus licheniformis, Bacillus cereus group, Bacillus fumulus, Bacillus sonorensis, Cytobacillus firmis, Nealius circulans, Panibacillus polymixa, and Pristia megatherium. These all will cause ropinus in bread. 35. What spoilage microorganism is responsible for the mousy taint in wine? Which among the following microorganism is the causative agent of mousy taint? Brettanomyces species, Lactobacillus brevis, Zygosaccharomyces ruxi, Aspergillus flavus. Which is the answer? Your answer is A. Brettanomyces species. Mousery taint is a microbiological fault caused by the spoilage yeast belonging to the genera Brettanomyces and Dacara, and by the lactic acid bacteria like Lactobacillus hilgardae and Lactobacillus brevis. The compound primarily responsible for the taint is 2-acetyl 3,4,5,6 tetrahydropyridine, which is commonly described as mouse cage, mouse urine, popcorn, or corn chip. 36. What microorganisms is responsible for the characteristic aroma of Swiss cheese? Lactobacillus delbrueckii, Propionibacterium frudentrici, Penicillium camemberti, D. Lactococcus lactis. Which is the answer? Your answer is B. Propionibacterium frudentrici. 37. Which fungal species is essential for developing the rind of brie cheese? Penicillium roqueforti, Penicillium camemberti, Aspergillus flavus, Candida albicans. Which is the answer? Your answer is option B. Penicillium camemberti. 38. Which symbiotic relationship is essential in yogurt production? Streptococcus thermophilus and Lactobacillus bulgaricus, Lactobacillus acidophilus and Lactobacillus casei, E. coli and Saccharomyces cerevisiae, Bifidobacterium bifidum and Lactobacillus reuteri. Which is the answer? Your answer is A. Streptococcus thermophilus and Lactobacillus bulgaricus. 39th question. What microbial spoilage in yogurt leads to an uncharacteristic pink coloration? Rhodotorla species, Aspergillus niger, Penicillium chrysogenum, Candida parapsilosis. Which is the answer? Here answer is A. Rhodotorla species. 40th question. Which metabolic compound in green tea has antioxidant properties that inhibit microbial spoilage? Catechins, theanine, flavonoids, tannins. Which is the answer? Here answer is A. Catechins. 41. Causative agent of wine spoilage is Zygosaccharomyces bailey, Saccharomyces cerevisiae, both A and B, none of the above. Your answer is A. Zygosaccharomyces bailey. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these two numbers. Amazon link is available in the description. 42. Which spoilage organism is in wine causes cork taint? Trichoderma longibracciatum, Penicillium expansum, Aspergillus niger, Brettanomyces bruxellensis. Which is the answer? Your answer is the Brettanomyces bruxellensis. 43. Which yeast strain is preferred for fermenting sparkling wine? Saccharomyces baianus, Saccharomyces cerevisiae, Candida krusei, Brettanomyces bruxellensis. This is the answer. Your answer is A. Saccharomyces baianus. 44. Which year was designated as the International Year of Plant Health by United Nations? 2004, 2020, 2006, 2007. It is the answer. 2020 is the answer B. 45. Which year was designated as the International Year of Quinoa by the United Nations? 2004, 2013, 2006, 2007. It is the answer. Your answer is B. 2013. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. There are 100 MCQs. First question. Which food is considered as a natural probiotic? A. Yogurt, B. Bread, C. Pasta, D. Milk. Which is the answer? Your answer is A. Yogurt. Second question. Omega-3 fatty acids are primarily associated with which health benefit? Heart health, improved digestion, bone strengthening, better vision. Which is the answer? Your answer is A. Heart health. Third question. Which of the following is a functional food ingredient known for its cholesterol lowering properties? Beta-glucan, lactic acid, glutamate, ascorbate. Which is the answer? Your answer is A. Beta-glucan. Fourth question. Which fermented product is made using Aspergillus oryzae? Soy sauce, yogurt, beer, cheese. Which is the answer? Your answer is soy sauce. Fifth question. Which method is commonly used to determine protein content in food? Gel method, Soxhlet extraction, C. Titration, D. Gas chromatography. Which is the answer? Your answer is A. Gel method. Sixth question. What is the calorific value of 1 gram of fat? 9 kilocalorie, 4 kilocalorie, 7 kilocalorie, 5 kilocalorie. Which is the answer? Your answer is A. 9 kilocalorie. Seventh question. What is the purpose of the Atwater system? To determine the total calorific value of food, to measure vitamin content, to analyze mineral composition, to test moisture content. What is the purpose of the Atwater system? Your answer is A. To determine the total calorific value of food. Eighth question. Which vitamin is measured using the HPLC technique? Vitamin C, vitamin A, vitamin D, all of the above. Which is the answer? Your answer is the all of the above. Ninth question. Which mineral is commonly deficient in global diets? Iron, potassium, magnesium, zinc. Which is the answer? Your answer is iron A. Deficiencies in iron, vitamin A and iodine are the most common around the world, particularly in children and pregnant women. Low and middle income countries bear the disproportionate burden of micronutrient deficiencies. 10th question. What is the advantage of 3D food printing? A. Customized nutrient profiles, B. Increased production speed, C. Improved microbial safety, D. Reduction in energy consumption. Which is the answer? Your answer is A. Customized nutrient profiles. 3D printed food. 3D printed food allows precise control over ingredients, textures, and shapes, enabling tailored culinary experiences for individuals with specific dietary preferences, restrictions, or cultural requirements. 11th question. What is the key ingredient in lab-grown meat? A. Animal stem cells, soy protein, wheat gluten, synthetic fat. Which is the answer? Here, answer is A. Animal stem cells. 12th question. What is the purpose of nanotechnology in food packaging? Improve barrier properties, detect contamination, enhances shelf life, all of the above. Your answer is D. All of the above. 13th question. What is the primary purpose of BT cotton? Pest resistance, high yield, drought tolerance, enhanced flavor. Your answer is A. Pest resistance. 14th question. Which technique is used for gene editing in GM crops? CRISPR-Cas9, gel electrophoresis, mass spectrometry, D. PCR. Which is the answer? Your answer is A. CRISPR-Cas9. 15th question. What does the term transgenic mean in GM food? Genetically modified foods. A. Gene transferred between different organisms. Naturally modified organisms. Genetically identical organisms. Organisms modified with non-DNA methods. Which is the answer? Here answer is A. Genes transferred between different organisms. 16th question. Lactose intolerance is caused by a deficiency of which enzyme? Lactase, amylase, protease, lipase. Which is the answer? Your answer is A. Lactase. 17th question. What is the main sugar substrate in traditional wine fermentation? Glucose, fructose, sucrose, glycerol. Which is the answer? Your answer is B. Fructose. 18th question. Toxic substance in apple seeds. Anthocyanin, curcumin, amygdalin. All of these. Which is the answer? Your answer is C. Amygdalin. 19th question. Which compound in almond can complicate the digestion of proteins and the uptake of iron? Phytic acid and oxalates. B. Tannins. C. Amygdalin. D. All of this. Which is the answer? Here answer is B. Tannins. 20th question. First commercially grown genetically engineered food is to be granted a license for human consumption. It was developed by the Californian company Calgene in the 1980s. A. Flavor Saver. B. CGN 895642. Both A and B. Lily. Which is the answer? Your answer is C. Both A and B. This flavor saver is known as flavor saver tomato. 21. Which food allergy is most prevalent in children? Milk allergy, fish allergy, wheat allergy, nut allergy. Which is the answer? Your answer is A. Milk allergy. 22. When was the Food Safety and Standards Authority of India established? 2004, 2006, 2008, 2010. Which is the answer? Your answer is C. 2008. FSSAI was established on 5th September 2008 under Food Safety and Standards Act 2006, which was operationalized in year 2006. The FSSAI consists of a chairperson and 22 members. Question number 23. Which act serves as the legislative framework for FSSAI? Prevention of Food Adulteration Act, Food Safety and Standards Act 2006, Food Products Order 1955, Edible Oil Packaging Order 1988. Which is the answer? Your answer is B. Food Safety and Standards Act 2006. 24. What is the primary responsibility of FSSAI? Framing regulations for food safety, conducting food business audits, granting trade licenses, handling food import policies. Your answer is A. Framing regulations for food safety. 25. What is the name of FSSAI's mobile food testing lab? Food on Wheels, Safety on Wheels, Food Safety on Wheel, Food and Safety Mobile. Your answer is C. Food Safety on Wheel. Update on food testing laboratories in India released on 29 July 2022. A total of 224 food testing laboratories recognized by FSSAI grants to states for National Accreditation Board for Testing and Calibration, NABL accreditation, certified reference materials, consumables, and for hiring contractual manpower provided by FSSAI. 224 testing laboratories, including 53 state government laboratories, 145 private laboratories, and 26 other government laboratories for primary testing, and 20 laboratories for testing referral food samples. 26. FSSAI recently launched an awareness campaign in which city in 2024? Mumbai, Chennai, Delhi, Bangalore. Here answer is C, Delhi. 27. What is the FSSAI limit for trans fats in food products? 2%, 5%, 10%, 0%. Here answer is A, 2%. The Food Safety Standard Authority of India announced that all edible refined oils, vanaspati, bakery shortening, margarines, vegetable fat spreads, and mixed fat spreads may only contain 3% or less trans fats by January 2021 and 2% or less trans fats by January 2022. 28. Which FSSAI initiative focuses on reducing trans fat levels in foods? Safe India Drive, Heart Attack Rewind, Trans Fat Free India, Clean Eating India. Is the answer? Your answer is B, Heart Attack Rewind. Heart Attack Rewind. The FSSAI launched a mass median campaign, Heart Attack Rewind, on November 30, 2018 for the elimination of industrially produced trans fat in food supply. It will support FSSAI's global target of eliminating trans fat in India by the year 2022. This is a year ahead of the global target by the World Health Organization for the complete elimination of trans fat. 29. Who appoints the chairperson of FSSAI? Prime Minister, Ministry of Food Processing Industry, Central Government, State Governments. The answer is C, Central Government. The Chief Executive Officer of the Food Authority is an officer not below the rank of Additional Secretary to the Government of India, appointed by the Government of India as the Executive Head of FSSAI. 30. What is FSSAI regulation on organic food? Prohibits all sales, requires specific labeling, mandates 100% organic content, does not regulate organic food. Which is the answer? Your answer is B, requires specific labeling. Labeling on the package of organic food shall convey full and accurate information on the organic status of the product. 31. What is the term for FSSAI certification for organic products? India Organic, FSSAI Organic Standard, Jaivik Bharat, Organic India Seal. Which is the answer? Your answer is C, Jaivik Bharat. Jaivik Bharat, the portal on organic food from India, is a regulatory portal. At the heart of the portal is an Indian Organic Integrity Database jointly developed by FSSAI, APEDA, and PGS India on organic food standards, certification processes, information relating to FBOs. The unified logo is an identity to mark distinguish organic products from non-organic ones, supported with the tagline Jaivik Bharat. 32. Question number 32. What is FSSAI's role in imported food products? Complete exemption from scrutiny, licensing and testing, only labeling inspections, no role in imports. Which is the answer? Your answer is B, licensing and testing. 33. Which lab standard does FSSAI rely on for food testing? NABL, ISO, HACCP, GMP. This is the answer. Your answer is A, NABL. 34. Which program educates school students on food safety? Eat Right School, Safe Food Zone, Blissful Food Program, Food for Thought. This is the answer. Your answer is A, Eat Right School. Eat Right School is a nationwide program to inculcate right eating habits in school children through curricular and extracurricular activities. Question number 35. Which FSSAI initiative aims to combat food wastage? A, Save Food, Share Food. B, Zero Hunger Mission. C, Food for All. D, Hunger Free India. Which is the answer? Your answer is A, Save Food, Share Food. What is IFSA? Known in its full form as Indian Food Sharing Alliance. IFSA has been formed by the Food Safety and Standards Authority of India to help solve India's food waste and hunger crisis by working with various partner organizations, food recovery agencies, and NGOs. Question number 36. In 2024, FSSAI introduced new standards for which food product? Edible oils, milk, packaged water, ready to eat foods. This is the answer. Your answer is C, packaged water. Question number 37. What is the consumer complaint helpline number of FSSAI? A 1911 1556 1900 112 211 1900 11 2100. Which is the answer? Your answer is D 1900 11 2100. 38. Which international agency collaborates with FSSAI on food standards? WHO, FAO, CAC, ISO. Your answer is C. Codex Alimentarius Commission. 39. FSSAI uses which platform for online licensing? FOSTAC, Foodlick Online, FLRS, License Serve. Which is the answer? Your answer is option C. FLRS. FLRS is an online application launched by Food Safety and Standard Authority of India to facilitate food business operators in India to apply for license or registration certificate and track their application during the course of processing. 40. Slogan of World Food Safety Day 2022 is Safe Food, Better Health, Health Through Food, Eat Right, Stay Fit, Food Safety First. Which is the answer? Your answer is option A, Safe Food, Better Health. 41. Question number 41. World Food Safety Day is celebrated on June 7th, July 7th, April 8th, March 21. March 21st. Which is the answer? Your answer is A, June 7th. Theme of World Food Safety Day 2024 is Food Safety, Prepare for the Unexpected. 42. Which of the following organisms can grow in both aerobic and anaerobic conditions, making it a significant food safety concern? A. Clostridium botulinum, B. E. coli, C. Listeria monocytogenes, D. Salmonella typhi. Which is the answer? Your answer is B. E. coli. 43. What is the primary mechanism by which umami flavor is detected? Sweet receptor activation, bitter receptor signaling, glutamate interaction with taste receptors, D. Interaction with fat molecules. Which is the answer? Your answer is C. Glutamate interaction with taste receptors. 44. Which compound is most responsible for the spiciness of chili peppers? Capsaicin, piperine, curcumin, allyl isothiocyanate. Your answer is A. Capsaicin. Next question. 45. Which is the most spiciest chili in the world? The Carolina Reaper, Food Jolokia, Pepper X, none of these. Which is the answer? Your answer is the Carolina Reaper. Food Jolokia is the spiciest chili in India, found in Assam. The Carolina Reaper is officially the world's hottest pepper as ranked by Guinness Book of World Records. Carolina Reaper can top out at 2.2 million SHU. SHU means Scoville Heat Unit. It is a measurement of hotness or spiciness in food. 46. Question number 46. Which term describes the incorporation of plant DNA into bacterial plasmid for food biotechnology applications? CRISPR modification, gene splicing, cloning, recombinant DNA technology. Which is the answer? Your answer is D, rDNA technology. 47. Which is known as the king of poisons? Lithium, arsenic, hemlock, cadmium. This is the answer. Your answer is B, arsenic. Arsenic became a favored method for murder of the Middle Ages and Renaissance, particularly among ruling classes in Italy, allegedly because the symptoms are similar to those of cholera, which was common at that time. Arsenic poison often went undetected. 48. Which spice contains the compound piperine known for its pungency? Black pepper, cloves, turmeric, cardamom. Your answer is A, black pepper. 49. Name the heat and spice perception receptor in human body. TRPV1 receptor, epinephrine B2, both A and B. D, none of these. Your answer is A, TRPV1 receptor. 50. What is the primary function of sugar in jam making? A, preservative, acidifier, thickener, flavor enhancer. Your answer is A, which acts as preservative. 51. What is the optimum pH range for pectin to form a gel in jelly? 2.8 to 3.5, 4 to 5, 6 to 7, 7.5 to 8. Which is the answer? Your answer is A, 2.8 to 3.5. 52. Which citrus fruit is traditionally used in making marmalade? Orange, lemon, grapefruit, lime. Your answer is A, orange. 53. Which natural compound in health drinks is most effective in boosting immunity? Flavonoids, anthocyanins, polyphenols, ascorbic acid. Which is the answer? Your answer is D, ascorbic acid. 54. What is the most common binding agent used in restructured fruit snacks? Pectin, gelatin, starch, agar agar. Your answer is option A, pectin. 55. Which technique is widely used to extend the shelf life of minimally processed vegetables? MAP, irradiation, vacuum sealing, fermentation. Your answer is option A, MAP. 56. What is the primary benefit of IQF technique? Prevents enzymatic browning, preserves texture and flavor, reduces freezing time, all of the above. Your answer is D, all of the above. 57. Which microorganism is primarily involved in natural fermentation of pickles? A. Lactobacillus plantarum, B. Saccharomyces cerevisiae, C. Clostridium sporogens, D. Aspergillus oryzae. Your answer is option A. Lactobacillus plantarum. Question number 58. For what purpose ASTM method is used in spices? Assessing moisture content, measuring volatile oil content, determining spice color value, D. Checking microbial load. Which is the answer? Your answer is C. Determining spice color value. 59. Which component ensures proper setting of jam? Acid, sugar, pectin, all of the above. Your answer is D, all of the above. Which pectin type is used in low sugar lily? High methoxyl pectin, low methoxyl pectin, unmodified pectin, acidic pectin. Which is the answer? Your answer is B, low methoxyl pectin. 61. How is marmalade thickness controlled? Adjusting pectin levels, varying cooking time, altering sugar content. All the above. Your answer is the all of the above. 62. Which factor is most critical in preventing enzymatic browning during the storage of tomato paste? Low pH, high oxygen concentration, low temperature, high sugar concentration. Your answer is C, low temperature. 63. Which packaging material provides the best barrier against moisture and oxygen for tomato based products? Polythene, glass jars, metal cans with epoxy lining, laminated aluminum foil. Your answer is option D, laminated aluminum foil. 64. The occurrence of flat sour spoilage in canned tomato products is caused by which microorganism? Bacillus coagulans, Lactobacillus acidophilus, Clostridium botulinum, Pseudomonas fluorescens. Which is the answer? Your answer is option A, Bacillus coagulans. 65. Which defect is associated with excessive heating during the processing of tomato puree? Caramelization of sugars, phase separation, C. Protein coagulation, D. Color bleaching. Which is the answer? Your answer is A. Caramelization of sugars. 66. Tomato sauce developing a metallic taste during storage is most likely caused by acidic reactions with can lining, over fermentation, high temperature storage, microbial contamination. Which is the answer? Your answer is option A. Acidic reactions with the can lining. 67. Which enzyme is responsible for causing watery separation in improperly stored tomato juice? Lipase, amylase, polygalacturonase, pectinesterase. Which is the answer? Your answer is C. Polygalacturonase is the answer. Option C. 68. Which bacterium is responsible for the production of GABA in fermented foods? Lactobacillus plantarum, Streptococcus thermophilus, Pediococcus acidilactici, D. Lactobacillus rhamnosus. Which is the answer? Your answer is option A. Lactobacillus plantarum. Among GABA producing microorganisms, lactic acid bacteria are commonly used in the production of many fermented foods. Lactobacillus plantarum. It is a new name of Lactobacillus plantarum. Lactobacillus plantarum is the new name of Lactobacillus plantarum. It is a lab species that has a long history of natural occurrence and safe use in wide variety of fermented foods and beverages. 69. Which microorganism is used in the industrial production of tempeh? A. Saccharomyces cerevisiae, B. Rhizopus oligosporus, C. Penicillium camembert, D. Zygosaccharomyces bailey. Which is the answer? Your answer is option B. Rhizopus oligosporus. 70. What role does leuconostoc misandroids play in sourdough fermentation? A. Produces ethanol, B. Initiates heterolactic fermentation, C. Hydrolyzes gluten proteins, D. Enhances moisture retention. Which is the answer? Your answer is option B. Initiates heterolactic fermentation. 71. In vinegar fermentation, acetic acid bacteria oxidize ethanol to lactic acid, carbon dioxide, acetic acid, butyric acid. In vinegar fermentation, this acetic acid bacteria oxidize ethanol to acetic acid. 70. Question number 72. Which bacterium produces diacetyl responsible for the butterly flavor in some dairy-based fermented products? Lactobacillus casei, Lactococcus lactis subspecies, Lactis biobar Diacetyl lactis. C. Bifidobacterium bifidum. D. Enterococcus faecium. Which is the answer? Your answer is option B. Lactococcus lactis subspecies lactis biovar. Diacetyl lactis. Diacetyl lactis. 73. What is the primary carbohydrate fermented in dosa batter? Starch, glucose, fructose, cellulose. Which is the answer? Your answer is option C. Starch. Next question, which of the following is a byproduct of heterolactic fermentation in sourdough? Lactic acid, acetic acid, ethanol, all the above. Your answer is D, all of the above. 75. Food irradiation is a process used to sterilize food by exposing it to radiation. The UV light kills harmful microorganisms and extends the shelf life of food without significantly affecting its taste or nutritional content. Option A, both statement and the reason are true and the reason is the correct explanation for the statement. B, both the statement and the reason are true but the reason is not the correct explanation for the statement. C, the statement is true but the reason is false. Statement is false but the reason is true. Which is the answer? Your answer is option C, the statement is true but the reason is false. Food irradiation typically uses gamma, electron beams or X-rays, not UV light. 76. In high pressure processing, food is subjected to very high pressures to kill microorganisms and pathogens without applying heat. Reason, this process helps maintain the food's natural flavors, nutrients and texture while providing a long shelf life. Here, which is the answer? Your answer is option A. Both the statement and reason are true and the reason is the correct explanation for the statement. 77. Irradiation is a food processing technique that uses high energy radiation to eliminate harmful pathogens and extend shelf life. This method can also help preserve the sensory and nutritional qualities of food, making it an effective alternative to traditional preservation methods. Which is the answer here? Your answer is A. Both statement and the reason are true. 78. Bacterifugation is a technique used to separate bacteria from milk before it is processed, particularly during the pasteurization stage. Reason, it helps in reducing the bacterial load, ensuring that the milk is safer and extends the shelf life of the product. Both are correct. So here answer is A. 79. Thermally processed foods never lost their vitamin content due to the high temperatures used in pasteurization and canning. The loss of vitamins can be minimized by using low temperature or shorter processing time. So here first sentence is not correct. So statement is not correct but reason is correct. So option is D. The statement is false but the reason is true. 80. Aseptic preservation is a process where food is sterilized and then packaged in a sterile container to prevent contamination. This method enables food to be stored for extended periods at room temperature without refrigeration. So here both are correct. Both the statement and the reason are true and the reason is the correct explanation for the statement. Aseptic preservation is a process where the food is sterilized and then packaged in sterile container to prevent contamination. This method enables food to be stored for extended periods. Both are correct. 81. Thermosonication uses high-frequency sound waves to pasteurize food products at lower temperatures. This method accelerates the heating process, reduces nutrient loss, and preserves food sensory properties compared to traditional thermal pasteurization. Both are correct. So here answer is A. 82. Which rare pathogenic bacterium caused botulism outbreaks in a traditional salted fish fermented product in Egypt, and what specific conditions during fermentation promoted its toxic production? Clostridium botulinum anaerobic low acid conditions. B. Listeria monocytogenes aerobic low salt conditions. C. E. coli high temperature fermentation. D. Vibrio cholerae high sugar fermentation. Here answer is option A. Clostridium botulinum anaerobic low acid conditions. 83. Which of the following foodborne pathogens is most commonly associated with the Vibrio cholerae outbreaks in India, particularly during monsoon season? Contaminated seafood, improperly cooked meat, contaminated drinking water, unpasteurized milk. Your answer is option C. Contaminated drinking water. 84. A large outbreak of typhoid fever occurred in a city. What might have been the common source of contamination? street food contaminated with Salmonella typhi, contaminated municipal water supply, improperly stored dairy products, poultry infected with Salmonella enterica. Which is the answer? Your answer is option B, contaminated municipal water. It might have been the common source for contamination. 85. Which of the following pathogens is most likely to cause gastroenteritis in children consuming untreated dairy products in India? E. coli, Shigella and Fluxneri, Campylobacter jejuni, Staphylococcus aureus. Your answer is Campylobacter jejuni. 86. In India, cases of hepatitis A linked to street food are most commonly associated with which type of food preparation? Uncooked vegetables in chats, fermented milk products, waste products, fried snacks like samosas, D. Spicy curries. Your answer is A. Uncooked vegetables in chats. 87. What is the primary reason for the higher prevalence of Ascaris lumbricoides infections in regions consuming unwashed raw vegetables? Use of untreated sewage water for irrigation, lack of refrigeration on harvested crops, high humidity during storage, overuse of chemical fertilizers. Your answer is option A, use of untreated sewage water for irrigation. 88. Why is Vibrio vulnificus poisoning rare but severe among consumers of raw oysters imported to India? Lack of salinity regulation during shipping, low pH of the storage medium, survival of pathogens in the cold chain, cross-contamination during preparation. Which is the answer? Your answer is A, lack of salinity regulation during shipping. 89. In a particular city, an outbreak of Shigella soni incident reported. What might have been the reason for the same to occur? Choose your opinion from the options. Cooked rice, cooked pulses, contaminated ice, pickles. Your answer is option C, contaminated ice. 90. In 1996, the Sakai City E. coli outbreak in Japan linked to contaminated radish sprouts caused over 9,000 illnesses. Which toxin was implicated? Shiga toxin, enterotoxin, botulinum toxin, endotoxin. Your answer is option A, Shiga toxin. 91. What was the primary contaminant in the 2008 Chinese milk scandal that led to widespread kidney damage in children? Cyanuramide, cyanide, aflatoxins, heavy metals. This is answer. Your answer is option A. This question I asked in previous question paper, but here answer is same, but cyanuramide is also known as melamine. Common name for melamine is cyanuramide and the chemical name for melamine is triaminotriazine. 92. Which bacteria caused the 2019 papaya outbreak in United States affecting multiple states? Salmonella ucanta, Listeria monocytogenes, E. coli, Shigella dysentery. This is answer. Your answer is option A, Salmonella enterica serotype Uganda. 93. Which day is celebrated as a World Food Day by the United Nations? October 15th, October 16th, September 28th, November 25. Your answer is option B, October 2016. 94. The United Nations declared 2016 as the International Year of Pulses. What was the main goal of this designation? Promoting sustainable agriculture, reducing global hunger and malnutrition, increasing awareness of legume health benefits, promoting technological innovation in food processing. Which is the answer? Your answer is option C, increasing awareness of legume health benefits. 95. The International Year of Fruits and Vegetables was declared in which year by the United Nations? 2020, 2019, 2021, 2022. Which is the answer? Here answer is option B, 2021. 96. Which of the following is the theme of World Health Day 2021, which focused on food systems? It's time to build a fairer, healthier world for everyone, everywhere. Food for all. Healthy diet for a healthy planet. Nurturing the world's health. Which is the answer? Your answer is option A. It's time to build a fairer, healthier world for everyone, everywhere. Question number 97. World Soil Day is observed on December 5th, December 10th, January 11th, June 7th. Which is the answer? Your answer is option A. December 5th is observed as World Soil Day. World Soil Day 2024 campaign under the theme Caring for Soils, Measure, Monitor, Manage underscores the importance of accurate soil data and information in understanding soil characteristics and supporting informed decision making on sustainable soil management for food security. 98. Which year was declared by the United Nations as the International Year of Potato, focusing on its importance for global food security? 2008. 99. The International Year of Sustainable Tourism for Development, which emphasizes food systems and their impact on global health, was declared in which year? 2017. 100th question, last question. What is the main focus of the Food Safety Training and Certification FOSTAC program? To enhance food safety knowledge among the public, to train food handlers and manufacturers, to promote international food safety standards, to provide financial aid to food industries. This is the answer. Your answer is B, to train food handlers and manufacturers. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Food Safety Officer book is available with 1000 MCQs from all sections. You can directly order from Amazon. Link is available in the description. Thank you. Some of the food festivals, famous food festivals in the world, and FSSAI important schemes, facts and MCQs. First question, what is the significance of using McConkey agar in microbial analysis? It detects Staphylococcus species. It differentiates lactose fermenters from non-fermenters. It is selective for spore-forming bacteria. It detects fungal growth. Which is the answer? Your answer is option B. It differentiates lactose fermenters from non-lactose fermenters. Second question, the pour plate technique is preferred over the spread plate technique because it requires less media, it can detect anaerobic bacteria, it prevents contamination, it is faster. Which is the answer? Your answer is option B. It can detect anaerobic bacteria too. Third question, for which of the following organisms is TCBS agar selective? A. E. coli, B. Vibrio species, C. Salmonella species, D. Listeria species. Your answer is option B. Vibrio species. Fourth question, which method is commonly used for testing coliforms in water? Plate count method, most probable number, for MPN, membrane filtration, staining and microscopy. Which is the answer? Your answer is option B. MPN. Fifth question, in the most probable number method, how many tubes are typically used in each dilution set? 2, 3, 5, 10. How many? 5 are used. C is the answer. Sixth question, which enzyme is used in bioluminescent ATP assays? Luciferase, protease, maltase, invertase. Which is the answer? Your answer is A, luciferase enzyme. Seventh question, which group of microbes is most commonly used as an indicator of water contamination? A. Enterococci, total coliforms, C. Clostridium, D. Listeria monocytogenes. It is the answer. Your answer is B. Total coliforms. Eighth question, the presence of E. coli in water indicates chemical contamination. Presence of fecal contamination. C. Presence of viruses. D. Presence of parasites. This is the answer. Your answer is option B. Presence of fecal contamination. Ninth question, why are Enterococcus species considered indicators of water quality? A. Option A, they are resistant to chlorination. Option B, they survive longer in water than coliforms. C, they indicate marine fecal contamination. Which is the answer? Your answer is all of the above. 3 are correct. So, 10th question, Clostridium perfringens is used as an indicator of recent fecal contamination, historical fecal contamination, water hardness, viral contamination. Here, which is the answer? Your answer is B. Historical fecal contamination. Clostridium perfringens spores have been proposed as indicators of fecal pollution because they are entirely of fecal origin, widely distributed in sewage, and consistently associated with human feces. Next question, 11th question. Which of the following test detects E. coli O157H7 in food? A. Gram staining. Option B. Latex agglutination test. Option C. Triple sugar ion test. Option D. Coagulase test. Which is the answer? Your answer is option B. Latex agglutination test. The polystyrene latex particles provided in the kit are coupled to antibodies against E. coli serotype O157H7. When the latex particles are mixed on the test card with fresh colonies of E. coli O157, the bacteria will bind to the antibody causing the latex particle to agglutinase. It is known as a positive reaction. 12th question, what does the Vox-Prosker test detect? Mixed acid fermentation, production of acetoin, lactose fermentation, sulfur reduction. Your answer is option B, production of acetylene. 13th question, the oxidase test is used to detect the presence of catalase, cytochrome oxidase, coagulase, beta-lactamase, which is the answer. Your answer is option B, cytochrome oxidase. Cytochrome oxidase test is used to identify bacteria that produce cytochrome oxidase, an enzyme of the bacterial electron transport chain. 14th question, which method is used to detect Listeria monocytogenes? XLD agar, PALCAM agar, EMB agar, McCongie agar. Which agar is used here? PanChem agar is used. 15th question. Which molecular technique is most sensitive for detecting pathogen in food? It is known as gold standard. Which is the answer? PCR, ELISA, immunodiffusion, fluorescent microscopy. Your answer is option A, PCR. 16th question. Multi-top MS is a rapid technique for microbial identification, antimicrobial resistant testing, measuring microbial loads, detecting toxin. Which is the answer? Your answer is option A, microbial identification. Microbial identification of bacteria species level in less than a minute. Although this method is about to become the standard of identification of microorganisms in most laboratories, multi-top MS technology is also being introduced in virology assays. 17th question. Which pathogen is often detected using loop-mediated isothermal amplification? E. coli, Salmonella, Vibrio, all of the above is the answer. 18th question. Largest known bacteria in the world? Epilepsium fuselsoni, Thiomargarita magnifica, Vibrio cholerae, Salmonella typhi. Before it was Epilepsium fuselsoni. Now it has been changed and the answer is Thiomargarita magnifica. It is all that is answer B, option B. 19th question. World's largest maple syrup festival was held in 2000 in which city? Milan, Elmira, Ottawa, Dubai. Which is the answer? Your answer is B, Elmira. 20th question. What is the primary focus of World Food Day observed on October 16th? Promoting gourmet cuisine, eradicating hunger and ensuring food security, celebrating local recipes, encouraging vegetarianism. Your answer is option B, eradicating hunger and ensuring food security. 21. What is the purpose of World Bee Day celebrated on May 20th? To promote beekeeping as a hobby, to highlight the role of bees in pollination and food security, to raise awareness about bee stings, to celebrate honey production exclusively. Your answer is option B, to highlight the role of bees in pollination and food security. 22. When is International Coffee Day is observed? September 29, October 1st, July 7th, August 3rd. Which is the answer? Your answer is option B, October 1st. 23rd. World Food Safety Day is celebrated on June 7th, July 11th, May 29th, April 22. Which is the answer? Your answer is option A, June 7th. 24. When is World Vegetarian Day celebrated? October 1st, November 1st, September 5th, August 19th. Which is the answer? Your answer is option A, October 1st. Next question 25. World Water Day is celebrated on March 21st, March 22nd, June 7th, December 5th. Which is the answer? Your answer is option B, March 22nd. 26. International Sushi Day is observed on June 18th, June 19th, May 29th, April 12th. Which is the answer? Your answer is option A, June 18th. 27. What spoilage microorganism in yogurt produces an off-putting fruity odor? Saccharomyces cerevisiae, Candida kefir, Geotrichum cantitum, Pseudomonas fragae. Which is the answer? Your answer is option D, Pseudomonas fragae. 28. Which day is celebrated as National Milk Day in India to honor Dr. Varghese Kudian? June 1st, November 26th, October 16th, August 15th. Your answer is option B. National Milk Day is celebrated on November 26 every year, making the birth anniversary of Dr. Varghese Kudian, who revolutionized India's dairy industry. This day highlights the importance of milk as a healthy beverage and recognizes key contributions to the dairy sector through awards and initiatives. 29. National Nutrition Week is observed in India during July 1st to 7th, September 1st to 7th, August 15th to 21st, October 16th to 22nd. Which is the answer? Your answer is option B, September 1st to 7th. National Nutrition Week is an annual nutrition event of great importance and is observed in the country from 1st to 7th September every year since 1982. It has been started in the year 1982. 30th question. Which Indian festival is known for the preparation of special dishes like gujia and tandai? Diwali, Holi, Pongal, Eid, which is the answer. Here answer is option B, Holi. 31. Which country is the Mecca of street food? Which country is known as the Mecca of street food? Thailand, China, Bali, Sumatra. Which is the answer? Here answer is a Thailand. Question number 32. The highest known concentration of vitamin C of any food is found in kakadu, plum, amla, raspberry, blueberry. Which is the answer? Here answer is option A, kakadu plum. Kakadu plum is an Australian native superfood containing 100 times more vitamin C than oranges. Question number 33. Sweetest fruit in the world? Mango, strawberry, coconut, raspberry. Which is the answer? Your answer is option A, mango. Mango is also known as king of the fruit. 34. The Eat Right India movement was launched by FSSAI in which year? 2015, 18, 2020, 22. Which is the answer? Your answer is option B, 2018. To improve public health in India and combat negative nutritional trends to fight lifestyle diseases, FSSAI launched the Eat Right movement on 10th July 2018. You have to remember the day, 10th July 2018. 35. Which FSSAI initiative is targeted at workplace food safety? Eat Right Campus, Safe Food at Work, Workplace Hygiene Movement, Healthy Office Plan. Which is the answer? Your answer is option A, Eat Right Campus. The Eat Right Campus initiative led by FSSAI aims to promote safe, healthy and sustainable food in campuses such as universities, colleges, workplaces, hospitals, tea estates, etc. across the country. So the answer is Eat Right Campus. 36. The fortification of food program by FSSAI promotes adding nutrients to fresh fruits, staple foods like rice, wheat and salt, soft drinks, canned goods. Which is the answer? Your answer is option B, staple foods like rice, wheat and salt. 37. What is the tagline of the Eat Right India movement? Eat smart, eat safe, healthy India, happy India, Sahi Bhojan Bhaktar Jeevan. D. Nutrition for all. Which is the answer? Your answer is option C, Sahi Bhojan Bhaktar Jeevan. 38. RUCO, Repurpose Used Cooking Oil Initiative by FSSAI focuses on recycling cooking oil for industrial use, reducing the use of palm oil, monitoring oil adulteration, repurposing used oil into biodiesel. Your answer is option D, repurposing used oil into biodiesel. 39th question. Which mobile app was launched by FSSAI to help consumers report food safety violations? Food Safety Connect, Food Watch India, Eat Right app, Safe Food Guide. This is the answer. Your answer is option A, Food Safety Connect. 40th question. Under the Eat Right India campaign, FSSAI promotes the reduction of which three components in food? Sugar, fat and additives, salt, sugar and trans fats, salt, preservatives and flavor enhancers, fats, carbohydrates and proteins. Which is the answer? Here answer is option B, salt, sugar and trans fats. 41. The Jaivik Bharat logo by FSSAI used for certifying organic foods, processed foods, packaged drinking water, fortified foods. Your answer is option A, organic foods. 42. What is the primary aim of the Food Safety Magic Box Initiative? Educating children on food adulteration detection, providing food testing kits to restaurants. C. Promoting organic food in schools. D. Regulating street food vendors. Which is the answer? Here answer is option A, educating children on food adulteration detection. 43. Which of the following is not an FSSAI initiative? Eat Right School, Safe Food Drive, Jaivik Bharat, Poshan Abhiyan. Here answer is option D, Poshan Abhiyan is not an FSSAI initiative. It is government initiative. 44. What does the FOSTAC program by FSSAI stand for? Food Safety and Training Center, Food Safety Training and Certification, Food Standardization Testing Campaign, Food Security and Training Certification. Which is the answer? Here answer is option B, Food Safety Training and Certification. 45. The dark book of FSSAI refers to Guide for Safe and Nutritious Food at Home, Guide for Safe and Nutritious Food at Restaurants, Detect Adulteration with Rapid Test booklet. D. None of the above. Your answer is option D. Detect adulteration with rapid test. 46. The purple book of FSSAI refers to Guide for Safe and Nutritious Food at Home, Guide for Safe and Nutritious Food at Restaurants, on general recommendations on diets for common medical conditions such as diabetes, hypertension, cancer, and so on. None of these. Your answer is option C. 47. The orange book of FSSAI refers to Guide for Safe and Nutritious Food at Home, Guide for Safe and Nutritious Food at Restaurants, Detect Adulteration with Rapid Test, Safe and Nutritious Food at Workplace. Your answer is option D, safe and nutritious food at workplace. 48. The Yellow Book of FSSAI refers to Guide for Safe and Nutritious Food at Home, Guide for Safe and Nutritious Food at Restaurants, to assure safe and nutritious food at school. None of these. Your option is option C. Option C, to assure safe and nutritious food at school. The Yellow Book of the Food Safety and Standards Authority of India is your guide to making such choices to assuring safe and nutritious food at school. To inculcate wholesome food habits at a young age, as habits formed in our early years stay with us for life. Next question, 49th question. Which chemical preservatives are found carcinogen and banned in bread? Potassium bromide, potassium iodide, both A and B, sodium chloride. Which is the answer? Your answer is option C, potassium bromide and potassium iodide. Question number 50. The first genetically engineered crop plant is tomato, tobacco, potato, brinjal. Which is the answer? Your answer is option B, tobacco. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. First question. Which country is the largest producer of rice globally? A. India, B. China, C. Indonesia, D. Vietnam. Here answer is option B. China. China is the world's largest producer of rice, followed by India and Indonesia. Second question. What is the scientific name of the crop that forms a staple food in most Asian countries? A. Triticum aestivum, B. Oryza sativa, C. Siam maize, D. Hordeum vulgare. Your answer is option B. Oryza sativa. Third question. Which vitamin is enriched in golden rice? Vitamin C, vitamin B12, vitamin A, vitamin D. Here option is C, vitamin A. Fourth question. Which cereal crop is associated with the green revolution in India? Wheat, rice, sorghum, barley. Here answer is option A, wheat. Fifth question. The International Rice Research Institute is located in which country? India, Philippines, Thailand, Bangladesh. Here answer is option B, Philippines. IRRI is an independent non-profit research and educational institute founded in 1960 by the Ford and Rockefeller Foundations with support from the Philippines government. The institute, headquartered in Los Banos, Philippines, has offices in 17 rice-growing countries in Asia and Africa, and more than 1,000 staff. Sixth question. Who was the first Asian Director General of the International Rice Research Institute IRRI in the Philippines? A. Dr. M.S. Swaminathan, B. Sri Sambitroda, C. Dr. Shamaprasad Mukherjee, D. None of these. Your answer is option A, Dr. M.S. Swaminathan. Seventh question. What is the protein percentage in maize on an average? 5%, 10%, 15%, 20%. Your answer is option B, 10%. Protein content varies in common varieties from about 8 to 11% of the kernel weight. Most of it is found in the endosperm. The protein in maize kernels has been studied extensively. Eighth question. What is a significant byproduct of rice milling used in livestock feed? A. Bran, B. Husk, C. Pith, D. Chaff. Your answer is option A, bran. Ninth question. International Year of Rice 2016, 2002, 2004, 2006. Your option is C, 2004. On 16 December 2002, the United Nations General Assembly declared 2004 the International Year of Rice to focus the world's attention on the role that rice can play in providing food security and poverty alleviation. 10th question. What is the primary reason for soaking pulses before cooking? Improve taste, remove anti-nutrients like phytic acid, reduce cooking time, all of the above. Your answer is D, all of the above. Reducing levels of tannins and polyphenols through soaking helps increase our body's ability to absorb minerals including iron, zinc and calcium, as well as proteins found in foods such as chickpeas, mung beans, lentils and peas. 11th question. Which Indian state is the largest producer of pigeon pea, tur dal? Maharashtra, Karnataka, Andhra Pradesh, Madhya Pradesh. Your answer is option A, highest production of pigeon pea from Maharashtra, which is around 30% of national production. 12th question. Which pulse has the highest protein content? Soybean, chickpea, lentils, green gram. Here answer is option A, soybean. 13th question. Which year was declared the International Year of Pulses by United Nations? 2016, 18, 20, 22. Here answer is option A, 2016. Question number 14. What is the scientific name of chickpea? Lens culinaris, Caesar ericinum, Pisum sativum, Phaseolus vulgaris. Here answer is option B, Caesar ericinum. 15th question. Which pulse contains the anti-nutritional factor lathyrism? Pigeon pea, green pea, grass pea, chickpea. Here answer is C, grass pea. 16th question. Which pulse is used for the preparation of tofu? Soybean, lentil, black gram, chickpea. Here answer is A, soybean. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers. You can directly purchase from Amazon link which is available in the description. 17th question. Which chemical is used for preserving pulses for longer storage? Methyl bromide, phosphine, sodium benzoate, calcium carbonate. Your answer is option B, phosphine. Brood or pulses beetles or bean weevils are serious pests of stored pulses, and the only curative chemical option approved for use in India is fumigation with phosphine. 18th question. Which millet is called ragi in India? Sorghum, pearl millet, finger millet, foxtail millet. Here answer is option C, finger millet. 19th question. Which year was declared the International Year of Millets by United Nations? 2023, 2022, 2021, 2020. Here answer is option A, 2023. Food and Agriculture Organization and United Nations has recognized 2023 as International Year of Millets for awareness about health and nutritional benefits of millets. The Indian government proposed to celebrate 2023 as International Year of Millets. 20. Which millet is rich in dietary fiber and used as substitute for rice? Pearl millet, finger millet, kodo millet, little millet. Here answer is option D, little millet or foxtail millet. Whole grains are so versatile that it is such an easy rice replacement. 21. Which state in India is the largest producer of pearl millet? Bajra, Rajasthan, Maharashtra, Gujarat, Karnataka. Your answer is option A. Rajasthan accounts for 44% of pearl millet produced in India. UP, Haryana, Maharashtra and Gujarat are the next major producers contributing 15%. 22. Which millet is traditionally used to make jowar bhakri? Sorghum, pearl millet, foxtail millet, finger millet. Your answer is option A. Sorghum. 23. Which millet is known as Navane in Karnataka? Sorghum, foxtail millet, little millet, barnyard millet. Your answer is option B, foxtail millet. 24. Which nutrient in millets helps manage diabetes? High protein, low glycemic index, high fiber, both B and C. Your answer is both B and C. 25. Which millet is best suited for cultivation in arid regions? Pearl millet, finger millet, foxtail millet, little millet. Your answer is option A, pearl millet. 26. What is the common name of Panicum miliaceum? Foxtail millet, proso millet, little millet, kodo millet. Your answer is option B, proso millet. 27. Which Indian state is the largest producer of groundnut? Gujarat, Andhra Pradesh, Tamil Nadu, Maharashtra. Your answer is option A, Gujarat. 28. Which oil seed has the highest oil content by percentage? Sesame, sunflower, soybean, groundnut. Your answer is option A, sesame. Which country is the largest producer of soybeans globally? USA, Brazil, Argentina, India. Your answer is option B, Brazil. 30. Which oil seed crop is scientifically known as Helianthus annuus? Sunflower, groundnut, mustard, soybean. Your answer is option A, sunflower. 31. What is the primary fatty acid found in mustard oil? Oleic acid, erucic acid, linoleic acid, palmitic acid. Your answer is option B, erucic acid. Mustard oil has about 60% of monounsaturated fatty acids, 42% erucic acid and 12% oleic acid. It has about 21% polyunsaturated fat, 6%, the omega-3 alpha linolenic acid and 15% the omega-6 linolenic acid, and it has about 12% of saturated fats. 2. Question number 32. Which oil seed is widely used for biodiesel production? Sunflower, soybean, rapeseed, castor. Your answer is option C, rapeseed. 33. Which crop is known as the king of oil seeds? Soybean, groundnut, sesame, sunflower. Your answer is option B. In India, groundnut is considered the king of oil seeds. Groundnut is also called a wonder nut or poor man's almond. 34. Which oil seed crop produces linseed oil? Flaxseed, castor, groundnut, sesame. Your answer is option A, flaxseed. 35. What is the primary use of castor oil besides culinary purposes? Industrial lubricants, biodiesel production, soap manufacturing, all of the above. Your answer is option D, all of the above. 36. Which year was declared as the International Year of Oil Seeds by FAO? 2023, 2016, 2020. No such year has been declared. Your answer is option D, no such year has been declared. 37. Which oil seed is a major source of tocopherols, vitamin E? Sunflower, soybean, groundnut, sesame. Your answer is option A, sunflower. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. You can directly purchase through Amazon link. 38. Which country is the largest producer of barley? Russia, Canada, China, Ukraine. Your answer is option A, Russia. 39. Which variety of wheat is most suitable for bread making due to its high gluten content? Durum wheat, hard red wheat, soft white wheat, club wheat. Your answer is option B, hard red wheat. 40. Which Indian pulse is popularly used in the preparation of dal makhani? Black gram or urad dal, red gram or tur dal, green gram or moong dal, Bengal gram or chana dal. Which is the answer? Your answer is option A, black gram or urad dal. 41. Which nutrient is present in highest concentration in pulses? Protein, carbohydrates, fats, fiber. Your answer is option A, protein. 43. King of pulses is chickpea, soybean, pigeon bee, none of the above. Your answer is option A, chickpea. 44. Which disease is caused by the deficiency of niacin found in maize-based diets? Beriberi, pellagra, scurvy, rickettsia. Your answer is option B, pellagra. 45. Which pulse crop is most drought resistant? Chickpea, pigeon pea, lentil, cowpea. Your answer is option B, pigeon pea. 46. Which microorganism is used to enhance nitrogen fixation in pulses? Rhizobium, azospirillum, mycorrhiza, trichoderma. Your answer is option A, rhizobium. 47. Which country was the largest exporter of wheat in 2023? Russia, Canada, United States, Australia. Your answer is option A, Russia. 48. Which millet is most commonly used in bird feed? Foxtail millet, proso millet, little millet, pearl millet. Your answer is option B, proso millet. 49. Which mineral is present in significant amounts in finger millet? Iron, calcium, magnesium, zinc. Which is the answer? Your answer is option B, calcium. 50. Question number 50. Which millet is known for its anti-diabetic properties? A. Foxtail millet. B. Little millet. C. Barnyard millet. D. Pearl millet. Your answer is option C. Barnyard millets. Barnyard millets have a low glycemic index. This means that they are digested and absorbed more slowly than high GI foods, glycemic index foods. This slow digestion results in a gradual release of glucose into the bloodstream. This prevents sharp spikes in blood sugar levels. 51. Which state in India launched the millet mission to boost millet production? Odisha, Karnataka, Andhra Pradesh, Madhya Pradesh. Your answer is option A, Odisha. 52. Which millet has the highest protein content? Finger millet, foxtail millet, proso millet, pearl millet. Your answer is option B, foxtail millet. 53. Which is the primary carbohydrate found in millets? Amylose, amylopectin, glucose, fructose. Your answer is option B, amylopectin. Starch is the major storage form of the carbohydrate in sorghum and millets. It consists of amylopectin, a branch chain polymer of glucose, and amylose, a straight chain polymer. 54. Which international organization promotes millet consumption as part of sustainable food practices? FAO, WHO, UNICEF, UNEP. Your answer is option A, FAO. International Year of Millets offers an incredible opportunity to drive progress towards achieving the 2030 Sustainable Development Goals by transforming local agri-food systems for better production and overall a better life, ensuring no one is left behind. 55. Which millet is also known as bajra in India? Pearl millet, finger millet, foxtail millet, little millet. Your answer is option A, pearl millet. 56. Which country declared 2023 as the year of millets? India, China, Ethiopia, United States. Your answer is option A, India. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. You can directly purchase from Amazon link which is available in the description. 57. Which variety of maize is specially cultivated for its high oil content? Dent maize, sweet corn, high oil maize, flint corn. Your answer is option C, high oil maize. 58. Which pulse is known as tur dal in India and is a primary source of protein in vegetarian diets? Pigeon pea, chickpea, lentil, green gram. Your answer is option A, pigeon pea. 59. What is the optimum protein content range in high quality durum wheat? 8 to 10%, 10 to 12%, 12 to 15%, 15 to 18%. Your answer is option C, 12 to 15%. 60. Question number 60. Which state leads India in the production of Kabuli chickpeas? Madhya Pradesh, Rajasthan, Maharashtra, Uttar Pradesh. Your answer is option C, Maharashtra. 61. Which millet is used for brewing alcoholic beverages in certain tribal regions of India? Finger millet, foxtail millet, barnyard millet, pearl millet. Your answer is option A, finger millet. Madhua, also one of the most popular and common millet-based fermented beverage prepared by almost all tribes of Arunachal Pradesh in all districts. It is a traditional alcoholic drink prepared from finger millets. 62. Which cereal crop is used in the production of malted beverages like beer? Barley, wheat, rice, maize. Your answer is option A, malted barley. 63. Which country ranks highest in global millet exports? India, Niger, Nigeria, Sudan. Your answer is option A, India. 64. Which nutrient in millets makes them beneficial for heart health? Omega-3 fatty acids, soluble fiber, iron, magnesium. Your answer is option B. Millets contains magnesium, which helps to regulate heart rhythm. Consuming millet may also elevate levels of the protein adiponectin, which can protect cardiovascular tissues. Millet also contains vitamin B3 or niacin. 65. Which oil seed was the first to have a genetically modified variety commercialized? Soybean, cottonseed, rapeseed, or canola? Groundnut. Your answer is option C, rapeseed or canola. 66. What percentage of the world's groundnut production is contributed by India? 15%, 19%, 25%, 30%. Your answer is option B, 19%. 67. Which part of the coconut is used to extract edible coconut oil? Kernel, husk, shell, water. Your answer is option A, kernel. 68. Which is the predominant carbohydrate in pulses? Starch, cellulose, fructose, pectin. Your option A is correct, starch. 69. Which year did the FAO officially launch the Global Soil Partnership to improve crop productivity including oilseeds? 2010, 2012, 2015, 2017. Your answer is option B, 2012. 70. Which millet is most resistant to salinity and can grow in arid regions? Pearl millet, finger millet, little millet, kodo millet. Your answer is option B, finger millet. 71. which Indian state contributes the highest to sunflower oil seed production? Karnataka, Andhra Pradesh, Maharashtra, Tamil Nadu. Your answer is option A, Karnataka. Which oil seed containing flower is known as poor man's saffron? Safflower, marigold, sunflower, rose. Your answer is option A, safflower. 73. Which protein component in wheat is responsible for gluten formation? Albumin, globulin, gliadin, casein. Your answer is option C, gliadin. Wheat and other related grains including barley and rye contain a mixture of two proteins, glutenin and gliadin. When flour made from grinding these grains is mixed with water, the two proteins combine and form gluten. Without water, gluten is not formed. The more the dough is mixed, the more gluten is developed. 74. What is the primary benefits of phytochemicals found in millets? Anti-cancer properties, enhanced taste, gluten formation, none of the above. Your answer is option A, anti-cancer properties. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. You can directly purchase through Amazon link. 75. Which oilseed byproduct is commonly used in organic farming as a biopesticide? Neem cake, mustard cake, coconut husk, groundnut shell. Your answer is option A, neem cake. 76. Which country recently developed the first millet-based infant food? India, Nigeria, Ethiopia, Brazil. Your answer is option A, India. Question number 77. Which oil seed is identified as future biofuel alternative due to its high oil yield? Castor, palm, jatropha, coconut. Your answer is option C, jatropha. Next question, 78. Which Indian government scheme was launched to promote pulses as part of PM ASHA? A. Principal Support Scheme A. Price Support Scheme PSS. B. Market Assurance Scheme MAS. C. Pradhan Mantri Krishi Vikas Yojana. D. National Food Security Mission. Your answer is option A. Price Support Scheme PSS. This Price Stabilization Fund or PSF. The PSF will continue to protect consumer from sharp price spikes in agricultural and horticultural commodities by maintaining a strategic buffer stock of pulses and onions. 79. Which region of India is most suited for groundnut cultivation due to its black soil? Gujarat, Tamil Nadu, Kerala, Andhra Pradesh. Your answer is option A, Gujarat. Question number 80. Kidney beans also provide insoluble fibers known as dash, which may cause diarrhea and flatulence in some people. A. Amylose. B. Fructose. C. Alpha galactosides. D. Chitin. Your answer is option C. Alpha galactosides. 81. Question number 81. Which millet crop contributes to carbon sequestration and climate resilience? A. Pearl millet. B. Sorghum. C. Finger millet. D. Kodo millet. Your answer is option B. Sorghum. Many millets, including sorghum, use the C4 photosynthesis pathway. This pathway is more efficient in utilizing carbon dioxide, particularly in conditions of high temperature and water stress, contributing to the adaptability of millets in challenging climates. Question number 82. Which oil seed contains the highest level of omega-6 fatty acids? Walnut, mustard, groundnut, sesame. Your answer is option A, walnut. Question number 83. Which type of millet has the highest calcium content among all cereals? Pearl millet, finger millet, barnyard millet, foxtail millet. Your answer is option D, finger millet. 84. Which FAO initiative focuses on reducing food loss during post-harvest stages of cereals? A. Save Food Initiative. B. Zero Hunger Initiative. C. Sustainable Food Systems Initiative. D. Global Food Waste Alliances. Your answer is option A. Save Food Initiative. 85. Which country is the leading exporter of soybean oil? Brazil, USA, Argentina, India. Your answer is option C, Argentina. 86. Question number 86. Which region is known as the Pulse Bowl of India? Bundelkhand, Northern Karnataka, Indo-Gangetic Plains, Malwa regions. Your answer is option B, Northern Karnataka. 87. Which state in India is famous for cultivating the aromatic rice variety Govindo Bhog? A. West Bengal, B. Odisha, C. Bihar, D. Assam. Your answer is option A, West Bengal. 88. Which pulse crop is most suitable for intercropping with sugarcane? PNP, green gram, chickpea, lentil. Your answer is option A, PNP. 89. Which oil seed contains the highest concentration of lignans beneficial for human health? Sesame, sunflower, mustard, soybean. Your answer is option A, sesame. Among edible plant components, the most concentrated lignan sources are sesame and flax seeds. 90. Which millet is considered the cereal of the future for its climate resilient properties? Sorghum, finger millet, pearl millet, little millet. Your answer is option B, finger millet. 91. Which vitamin is uniquely abundant in finger millet compared to other cereals? Vitamin C, vitamin D, vitamin B1, vitamin K. Your answer is option C, vitamin B1. Question number 92. Which oil seed is the primary source of resinolic acid used in the cosmetics industry? A. Castor, B. Coconut, C. Sunflower, D. Soybean. Your answer is option A. Castor. 93. Which cereal is most tolerant to saline soil conditions? Barley, rice, wheat, sorghum. Your answer is option A, barley. Barley is the most salt tolerant cereal, but it has poor waterlogging tolerance. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers or you can directly buy from Amazon link which is available in the description. 94. Which country is the largest producer of mustard oil globally? Canada, India, China, Ukraine. Your answer is option B, India. 95. Which pulse crop is traditionally referred to as dalchini in Indian markets? Horse gram, chickpea, lentil, black gram. Your answer is option A, horse gram. 96. Which oil seed contains the highest concentration of monounsaturated fatty acids? Olive, sunflower, groundnut, sesame. Your answer is option A, olive. Common vegetable oil high in MUFA include canola, rapeseed, and olive oil. 97. Which cereal is the richest source of lysine, an essential amino acid? Wheat, maize, oats, rice. Your answer is option C, oats. Oats contains 3.8 to 4% of lysine. 98. Which cereal has the highest glycemic index? Barley, white rice, millet, sorghum. Your answer is option B, white rice. Question number 99. Which vitamin is most abundant in unpolished rice? Vitamin C, vitamin D, thiamine, vitamin A. Your answer is option C, vitamin B1 or thiamine. Question number 100. Which mineral is the highest in oats? Potassium, iron, phosphorus, magnesium. Your answer is option D, magnesium. 101. Which pulse is the richest source of iron? Chickpeas, lentils, mung beans, kidney beans. Your answer is option B, lentils. 102. Which pulse is highest in zinc content? Soybean, green peas, lentils, chickpeas. Your answer is option D, chickpeas. 103. Which amino acid is least available in most pulses? Valine, lysine, methionine, leucine. Your answer is option C, methionine. Unlike lysine, which is the limiting amino acid for cereals, the limiting amino acids for pulses were found to be methionine, cysteine, and tryptophan. 104. Which millet has the lowest glycemic index? Foxtail millet, kodo millet, little millet, barnyard millet. Your answer is option D, barnyard millet. 105. Which fatty acid is predominantly found in millets? Linoleic acid, oleic acid, palmitic acid, stearic acid. Your answer is option A, linoleic acid. 106. Which oil seed is the richest source of omega-3 fatty acids? Sunflower seeds, flax seeds, sesame seeds, mustard seed. Your answer is option B, flax seed. 107. Which oil seed has the highest protein content? Peanut, soybean, sesame, sunflower. Your answer is option B, soybean. 108. Which oil seed is known for its anti-inflammatory properties due to its lignan content? Sunflower seed, sesame seed, mustard seed, flax seed. Your answer is option D, flax seed. Question number 109. Which vitamin is almost entirely absent in cereals and must be supplemented through other dietary sources? Vitamin C, vitamin B12, vitamin D, vitamin K. Your answer is option B, vitamin B12. 110. Which anti-nutritional factor in legumes inhibit protein digestibility by interfering with trypsin activity? Phytates, oxalates, protease inhibitors, tannins. Your answer is C, protease inhibitors. 111. Which anti-nutritional factor found in leafy vegetables binds calcium, making it less bioavailable? A. Saponins, B. Oxalates, C. Lectins, D. Phytates. Your answer is option B. Oxalates. 112. What is the main anti-nutritional factor present in soybeans that interferes with thyroid function? Goitrogens, tannins, cyanogenic glycosides, lectins. Your answer is option A. Goitrogens. 113. Which anti-nutritional compound is responsible for bitter taste in pulses like chickpeas? Phytates, saponins, tannins, oxalates. Your answer is option B, saponins. 114. What is the major anti-nutritional factor in linseed that can release toxic hydrogen cyanide? A. Oxalates, B. Cyanogenic glycosides, C. Tannins, D. Lectins. Your answer is option B, cyanogenic glycosides. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Direct link to purchase the book is available in the description. 115. Which anti-nutritional factor in cereals forms insoluble complexes with iron, reducing its absorption? Phytates, saponins, goitrogens, protease inhibitors. Your answer is option A, phytates. 116. Which anti-nutritional factor found in legumes can cause red blood cell clumping, hemagglutination? Lectins, tannins, saponins, oxalates. Your answer is option A, lectins. 117. Which anti-nutritional factor reduces the bioavailability of zinc in plant-based diets? Oxalates, phytates, lectins, protease inhibitors. Your answer is option B, phytates. 118. Which anti-nutritional compound found in raw egg whites inhibit biotin absorption? Lectins, avidin, tannins, goitrogens. Your answer is option B, avidin. 119. What anti-nutritional compound in spinach can lead to kidney stone formation when consumed in excess? Phytates, oxalates, tannins, lectins. Your answer is option B, oxalates. 120. Which anti-nutritional factor is destroyed by moist heat, making legumes safer to consume? Lectins, tannins, oxalates, cyanogenic glycosides. Here option is A, lectins are correct. 121. What is the primary mechanism by which tannins act as an anti-nutritional factor? Enzyme inhibition, protein precipitation, iron binding, cyanide release. Your answer is option B, protein precipitation. 122. Which anti-nutritional factor in green tea can reduce the bioavailability of non-heme iron? Oxalate, tannins, phytates, cyanogenic glycosides. Here option B is correct, tannins. 123. Which method is most effective at reducing phytates in cereals and legumes? Boiling, fermentation, sun drying, roasting. Your answer is option B, fermentation. 124. Which anti-nutritional compound in kidney beans can cause food poisoning if not properly cooked? Tannins, lectins, protease inhibitors, saponins. Your answer is option B, lectins, phytohemagglutinin. 125. What is the main mechanism by which saponins exhibit anti-nutritional properties? Inhibition of digestive enzymes, reduction in nutrient absorption, damage to intestinal mucosa, formation of insoluble complexes. Your answer is option C, damage to intestinal mucosa. It is possible that in very concentrated amounts and very large doses, saponins could cause some intestinal damage. 126. Which anti-nutritional factor in grains is responsible for flatulence in humans? Saponins, raffinose family oligosaccharides, phytates, tannins. Here answer is option B, raffinose family oligosaccharides. RFOs primarily cause flatulence in humans and animals. Hence, RFOs are considered the single most deterring factor for the wide acceptance of legumes in human and animal diets. Question number 127. Which anti-nutritional factor in raw soybeans can reduce the activity of the enzyme urease? Tannins, protease inhibitors, lectins, oxalates. Your answer is option B, protease inhibitors. 128. What is the primary anti-nutritional factor in mustard seeds that interferes with thyroid hormone synthesis? Oxalates, saponins, glucosinolates, phytates. Your answer is option C, glucosinolates. The anti-nutritional factor present in rapeseed and mustard oil cakes are glucosinolates that are a large group of plant secondary metabolites. The glucosinolates are mainly found in cruciferous plants and are natural compounds of various pungent plants including cabbage, horseradish and mustard. Question number 129. Which anti-nutritional compound in sorghum forms insoluble complexes with dietary proteins? Phytates, tannins, lectins, oxalates. Here answer is option B, tannins. The tannin content in sorghum seeds ranges from 0.10 to 3.6%, which is located in the pericarp, testa, aileron layer and endosperm. Soaking is an important process in the processing of sorghum seed to improve nutrition by reducing the tannin content. Question number 130. What is the main anti-nutritional factor found in bamboo shoots that requires through the boiling to remove toxicity? Cyanogenic glycosides, lectins, protease inhibitors, saponins. Your answer is option A, cyanogenic glycosides. The most predominant anti-nutrient in bamboo shoot is cyanogenic glycosides. It causes increase in blood glucose and lactic acid levels and a decrease in the ATP-ADP ratio, indicating the shift from aerobic to anaerobic metabolism. The anti-nutrients such as phytate can cause vitamins and mineral deficiencies. Question number 132. Which cooking method is most effective at reducing tannins in sorghum and millets? Boiling, soaking and fermentation, roasting, steaming. Here answer is option B, soaking and fermentation. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers. Amazon link to purchase the same is available in the description. 133. Which anti-nutritional factor found in buckwheat can cause photosensitivity in livestock? Tannin, phagopyrin, phytates, oxalates. Your answer is option B, phagopyrin. 134. Which anti-nutritional factor in cassava is significantly reduced by soaking and drying methods? A. Oxalates, B. Cyanogenic glycosides, C. Tannins, D. Lectins. Your answer is option B. Cyanogenic glycosides. 135. Which anti-nutritional compound present in tea reduces the bioavailability of dietary iron? Catechins, tannins, oxalates, flavonoids. Your answer is option B. Tannins. 136. What is the mechanism by which saponins reduce nutrient absorption? Protein denaturation, interaction with cell membranes, enzyme inhibition, chelation of minerals. Your answer is option B, interaction with cell membranes. Saponins have long been known to have lytic action on erythrocyte membranes. 137. What is the main mechanism by which phytates reduce mineral bioavailability? Enzyme inhibition, B. Formation of insoluble complexes, C. Protein precipitation, D. Fatty acid chelation. Your answer is option B. Formation of insoluble complexes. 138. Potent skin irritant toxin in raw cashew. Saponins, urushiol, tannins, oxalates. Your answer is option B. Urushiol. 139. Which toxic compound in apricot seeds can release cyanide upon hydrolysis? Phytates, amygdalin, tannins, lectins. Your answer is option B, amygdalin. Amygdalin is a compound found in the pits or seeds of apricots, apples, peaches, plums, fruit cherries, and other fruits. It's also in bitter almonds. A partly man-made purified form of amygdalin known as lateral was patented in the 1950s and became a popular alternative cancer treatment during 1960s and 70s. 140. Which anti-nutritional factor in tomatoes can cause gastrointestinal irritation in sensitive individuals? Lectins, oxalates, protease inhibitors, saponins. Your answer is option A, lectins. 141. Which compound in rye interferes with gluten formation and affects dough elasticity? Arabinoxylans, phytates, tannins, protease inhibitors. Here answer is option A, arabinoxylans. 142. Which anti-nutritional factor in raw fava beans can trigger hemolytic anemia in individuals with G6PD deficiency? Cyanogenic glycosides, oxalate, vicin and convicin, protease inhibitors. Here answer is option C, vicin and convicin. 143. Which method is most effective in reducing the trypsin inhibitor activity in legumes? Sun drying, high pressure cooking, soaking, fermentation. Here answer is option B, high pressure cooking. 144. Which anti-nutritional factor in quinoa causes a bitter taste and requires thorough washing to remove saponins, tannins, lectins, oxalates? Here answer is option A, saponins. 145. Which anti-nutritional factor is most commonly associated with kidney damage in livestock consuming fava beans? Cyanogenic glycosides, B. Oxalates, C. Vicin, D. Lectins. Your answer is option C, Vicin. 146. Which advanced detoxification technology is now being explored to neutralize anti-nutritional factors in underutilized wild grains? A. Enzyme immobilization techniques, nano encapsulation of chelating agents, precision heat mapping, fermentative biodegradation. Your answer is option B, nano encapsulation chelating agents. 147. Richest food source of selenium, Brazil nuts, almond, blueberry, cherry. Your answer is option A, Brazil nuts. 148. Which recently isolated anti-nutritional compound in hemp seeds interferes with amino acid transport in the gut? Cannabinoid glycosides, phytic acid derivatives, polyacetylens, lectin analogs. Your answer is option D, lectin analogs. 149. Which millet has the highest phosphorus content, making it essential for energy metabolism? Pearl millet, kodo millet, foxtail millet, finger millet. Your answer is option A, pearl millet. Food Safety Officer book is available with 1000 MCQs from all sections. Contact number. Contact to these two numbers. Amazon link to purchase the book is available in the description. 150. Which millet has highest antioxidant content? Barnyard millet, Rosso millet, little millet, finger Italian millet. Your answer is option D, finger Italian millet. If you like this channel, please share and subscribe. Don't forget to click the bell button to get important notifications. Don't forget to subscribe the channel. Which of the following is true about the use of chocolate agar in microbiology? Statement 1. Chocolate agar is used to cultivate fastidious organisms such as Haemophilus and Neisseria species. Hemoglobin is added to chocolate agar providing essential growth factors. It is selective for gram-positive organisms only. The heating process releases 10th and 5th factors needed for bacterial growth. Option. Statements 1, 2 and 4. B. Statement 1 and 4. C. All statements, statement 2 and 3. Which is the correct answer? Here answer is A. Statement 1, 2 and 4 are correct. Explanation. Chocolate agar is an enriched medium used to culture fastidious organisms and contains hemoglobin, which releases hemine and NAD factors. It is not selective for gram-positive bacteria. സൂക്ഷിച്ച് ആൻസർ ചെയ്യണം. Second question. In the context of microbiology media preparation, which of the following statements is correct? Statements. Autoclaving at 121 degrees Celsius for 15 minutes is the standard sterilization procedure for most media. Media containing heat-sensitive ingredients must be sterilized by filtration. Sugar containing media require a higher autoclaving temperature to prevent caramelization. The pH of the media should be adjusted after sterilization. Which is the correct answer? Here answer is C. Statement 1 and 2. Explanation. Autoclaving at 121 degrees Celsius for 15 minutes is typical for most media, while heat-sensitive components are sterilized by filtration. Sugar in media does not require higher temperatures and pH is usually adjusted before sterilization. So here answer is C. Statement 1 and 2. Next question. Third question. Which of the following is true about the preparation and use of Muller-Hinton agar in susceptibility testing? Statements: it is commonly used for antimicrobial susceptibility testing. Starch in the medium absorbs toxic substances. It is enriched to support the growth of fastidious bacteria. The depth of the agar must be standardized for accurate results. ഏതാതിൻ്റെ ആൻസർ വരിക? Here answer is A. Statement 1, 2 and 4. Explanation: Mullen-Hinton agar or MH agar is a standard medium for susceptibility testing due to its consistency and starch content which absorbs toxins. It is not enriched and agar depth must be uniform for accurate results. Fourth question: which statements are correct regarding the use of Thayer-Martin agar? Statements: it is selective for Neisseria species, particularly gonorrhea and meningitis. Contains antibiotics like vancomycin, colistin and nystatin to suppress contaminating organisms. This medium can also be used to isolate Haemophilus species. Thayer-Martin agar is a modification of blood agar. Here, which is the answer? Here answer is A. Statement 1, 2 and 4 are correct. Explanation: Thayer-Martin agar is selective for Neisseria, contains antibiotics to suppress other bacteria. It is a modification of chocolate agar, not blood agar, and is not suitable for Haemophilus isolation. Fifth question: which of the following statements is true about SDA? SDA Saboros dextrose agar. Statements: it is used for the cultivation of fungi and yeast. നമുക്കറിയാം എസ് ഡി എ ഫംഗസിനെ പി ഡി അല്ലെങ്കിൽ എസ് ഡി എ ആണ് ഫംഗസിനെ ഐസൊലേറ്റ് ചെയ്യാൻ വേണ്ടി കൾച്ചർ ചെയ്യാൻ വേണ്ടി യൂസ് ചെയ്യുക. എസ് ഡി എ ഹാസ് എൻ acidic pH that inhibits bacterial growth. Third statement: chloramphenicol may be added to further suppress bacterial growth. It is used for culturing anaerobic bacteria. ഏതാ അതിൻ്റെ ആൻസർ ചെയ്യാൻ? ഞാനിതിൻ്റെ ആൻസർ പറയുന്നില്ല, നിങ്ങൾ എന്ത് ചെയ്യുക, നോക്കി കൃത്യമായിട്ട് നോക്കി മനസ്സിലാക്കി ആൻസർ ചെയ്യുക. ഇതിൻ്റെ ആൻസർ ഏതാ വരിക? ആൻസർ ചെയ്തോളൂ. Here answer is A. Statement 1, 2 and 3. SDA is commonly used for fungal cultivation and has a low pH that inhibits bacterial growth. It may contain antibiotics like chloramphenicol, but it is not intended for anaerobic bacteria. Sixth question: which of the following statements about nutrient agar is correct? Nutrient agarന്റെ composition അറിയാലോ. nutrient agarൽ ഏതൊക്കെയാ അതിലുള്ള statements correct എന്ന് നമുക്ക് നോക്കാം. It is a non-selective general purpose medium. It is used to differentiate bacterial species based on colony morphology. Nutrient agar contains indicators for lactose fermentation. It is suitable for the cultivation of most non-fastidious organisms. ഇവിടെ ആൻസർ ചെയ്തോളൂ, കൃത്യമായി വായിച്ച് ആൻസർ ചെയ്തോളൂ. Here answer is B. Statement 1 and 4. First statement എന്താ? It is a non-selective general purpose medium. Fourth one: it is suitable for the cultivation of most non-fastidious organisms. Explanation: nutrient agar is a non-selective and general purpose supporting the growth of many non-fastidious organisms, but it lacks indicators for differentiation such as lactose fermentation. So here seventh question: which of the following statements is correct regarding transport media? They are designed to maintain microbial viability during specimen transport. Amis and Stewart's media are examples of transport media. Transport media contain nutrients to enhance microbial growth during transport. These media are typically prepared in liquid form. Which is the answer? Answer ചെയ്തോളൂ. Here answer is A. Statement 1, 2 and 4. Explanation: transport media maintain viability without supporting growth. Amis and Stewart's are common examples. These media do not contain growth promoting nutrients and may be prepared in liquid or semi-solid form. Eighth question: which of the following statements best describes enrichment media? Statements: enrichment media selectively favor the growth of certain organisms. They are often used to enhance the growth of pathogens in a sample. Alkaline peptone water is an enrichment medium for Vibrio species. Enrichment media is used primarily for aerobic bacteria only. Here which is the answer? Here answer is A. Statement 1, 2 and 3 are correct. Here explanation: enrichment media favor pathogen growth and include media like alkaline peptone water for Vibrio. They are not limited to aerobic bacteria. Next, ninth question: which of the following is correct for buffered charcoal yeast extract BC by E agar? Statement 1: It is used for isolating Legionella species. PCYE contains charcoal to detoxify metabolic products. This medium includes cysteine and iron to support Legionella growth. It is commonly used for isolating Campylobacter species. Here, which is the answer? Here, answer is A. Statement 1, 2, and 3. Explanation. BCYE is specialized for Legionella species containing charcoal to detoxify byproducts and essential nutrients like cysteine and iron. It is not used for Campylobacter. 10th question. Which of the following is true about CLED? CLED means cysteine lactose electrolyte deficient agar. Statements. It is used to culture urinary pathogens. Lactose fermentation on clad agar leads to yellow colored colonies. Electrolyte deficiency in the medium prevents swarming of Proteus species. It is highly selective for gram-negative bacteria. Here, which among the following is the answer? Here, answer is A. Statement 1, 2, and 3. Clad agar is used for culturing urinary pathogens. Lactose fermenters turn yellow on this medium and electrolyte deficiency prevents Proteus swarming. However, it is not selective for gram-negative bacteria exclusively. If you like this channel, please share and subscribe. Don't forget to click the bell button to get important notifications. What are aflatoxins? Aflatoxin varnating Aspergillus flavus. It is produced by toxin produced by Aspergillus flavus and Aspergillus parasiticus. Frequently contaminates crops like maize, peanut, cottonseed, tree nuts, and other spices under warm and humid conditions. അതായത് മൈസ് അതുപോലെ തന്നെ പീനട്ട് അതുപോലെ തന്നെ ഹ്യൂമിഡിറ്റി കൂടുതലുണ്ടെങ്കിൽ 15% ഓർ എബോവ് ഹ്യൂമിഡിറ്റി ഉണ്ടെങ്കിൽ അതിൽ ഫംഗസ് ഗ്രോ ചെയ്യും, അഫ്ലാടോക്സിൻ പ്രൊഡ്യൂസ് ചെയ്യും, ആസ്പെർജില്ലസ് ഫ്ലേവസും ആസ്പെർജില്ലസ് പാരസൈറ്റിക്കസും അതിൽ ഗ്രോ ചെയ്യും. ടൈപ്സ് ഓഫ് അഫ്ലാടോക്സിൻ ഏതൊക്കെ ടൈപ്പ് ഉള്ളത്? B1, B2, G1, G2. പിന്നെ M1 ഉണ്ട്. M1 എന്ന് പറഞ്ഞിട്ടുണ്ടെങ്കിൽ മെറ്റബോളിക് പ്രൊഡക്റ്റ് ഓഫ് അഫ്ലാടോക്സിൻ B1 ആണ്. അഫ്ലാടോക്സിൻ B1 എന്ന് പറഞ്ഞാൽ ബ്ലൂ ഫ്ലൂറസൻസ് കാണിക്കുന്നതുകൊണ്ടാണ് B1 എന്ന് പറയുന്നത്. B1 ഉം B2. ഗ്രീൻ ഫ്ലൂറസൻസ് കാണിക്കുന്നതാണ് G1 ഉം G2. അപ്പോൾ മോസ്റ്റ് പൊട്ടന്റ് ആൻഡ് കാർസിനോജനിക് എന്ന് പറഞ്ഞാൽ അഫ്ലാടോക്സിൻ B1 ആണ്. അപ്പോൾ അതാണ് പ്രത്യേകം ശ്രദ്ധിക്കേണ്ട. അഫ്ലാടോക്സിൻ B1 ആണ് ഏറ്റവും പൊട്ടന്റ് ആയിട്ടുള്ളത്. പിന്നെ B2 പറഞ്ഞാൽ ലെസ് ടോക്സിക് ആണ്. പിന്നെ G1 G2 ഫൗണ്ട് ഇൻ സം ക്രോപ്സ് ലെസ് ടോക്സിക് ദാൻ B1. അപ്പോൾ B1 ആണ് ഏറ്റവും ഇമ്പോർട്ടൻ്റ് ആയിട്ടുള്ള ഹെപ്പാറ്റോടോക്സിൻ, ഹെപ്പാറ്റോ കാർസിനോജൻ. പിന്നെ ഇതിൽ മനസ്സിലാക്കേണ്ട ഒരു ഇമ്പോർട്ടൻറ്റ് റിസർച്ച് പേപ്പറിൽ പറയണത് എന്താ വെച്ചാൽ അഫ്ലാടോക്സിൻ M1, അഫ്ലാടോക്സിൻ B1 നമ്മളെ കൗസ് എന്ത് ചെയ്യുക, പശുക്കൾ കൺസ്യൂം ചെയ്യും അഫ്ലാടോക്സിൻ B1. അപ്പോൾ എത്ര ആ പശു അഫ്ലാടോക്സിൻ B1 കൺസ്യൂം ചെയ്യുന്നുണ്ടോ അതിൻ്റെ 1 to 6% M1 ആയിട്ട് പ്രൊഡ്യൂസ് ചെയ്യും മിൽക്കിലൂടെ. ആ മിൽക്കിലൂടെ എന്ത് ചെയ്യും, ആ കുടിക്കുന്ന കുട്ടിയോ അല്ലെങ്കിൽ ആ കുടിക്കുന്ന ഹ്യൂമൻ ബീയിങ്സിൻ്റെ ശരീരത്തിലേക്ക് എത്തും. അതായത് ഒരു 100 ppm ആണ് അതിലുണ്ട് വെച്ചാൽ 1 to 6%, അതായത് 1 ppm to 6 ppm ആരുടെ ശരീരത്തിലേക്ക് എത്തും, അത് കൺസ്യൂം ചെയ്യുന്ന ആളുടെ ശരീരത്തിലേക്ക് എത്തിപ്പെടും. അതായത് അഫ്ലാടോക്സിൻ B1 എത്ര എമൗണ്ട് ഉണ്ടോ അതിൻ്റെ 1 to 6% പശുവിൻ്റെ പാലിലൂടെ ട്രാൻസ്ഫർ ചെയ്യും, മനുഷ്യൻ്റെ ശരീരത്തിലെത്തും. ടോക്സിസിറ്റി, അക്യൂട്ട് എഫക്ട്സ്. ഹൈ ഡോസസ് കോസ് അക്യൂട്ട് toxicosis with symptoms like liver failure, jaundice, abdominal pain and hemorrhage. Chronic effects, long-term exposure to low levels can cause hepatocellular carcinoma. Next, major classes of mycotoxin associated with food and health effects and species affected. Aflatoxin, Aspergillus flavus and Aspergillus parasiticus. Contaminated products, corn, peanuts, cottonseed, tree nuts, dairy products. Animals affected, swine, dog, fish, cattle, poultry, humans. Liver lesions, clinical effects, liver cancer, growth impairment. Next, sources of contamination. Contaminated crops during the growth in the field under hot and humid conditions, improper drying and storage. Secondary storage sources, contaminated animal feed leading to toxin residues in milk, meat and eggs. Risk factors, environmental, high humidity and temperature, അതുപോലെ തന്നെ economic poor storage infrastructure in developing countries, അതുപോലെ തന്നെ dietary depending on aflatoxin prone staples, maize, peanuts. അതായത് ഹ്യൂമിഡിറ്റി ഏറ്റവും കൂടുതൽ ആർദ്രത കൂടുതലുണ്ടെങ്കിലാണ് അവിടെ എന്ത് ചെയ്യുക, കൂടുതലായിട്ട് ഫംഗസ് ഗ്രോ ചെയ്യുക. ഇതുപോലെ തന്നെ ഇത് ചൂടാക്കി കഴിഞ്ഞാൽ പെട്ടെന്ന് നശിച്ചു പോകില്ല. ചൂടാക്കി കഴിഞ്ഞാൽ 148 ഡിഗ്രി സെൽഷ്യസിലൊക്കെ ചൂടാക്കി കഴിഞ്ഞാൽ അരമണിക്കൂർ ഹാഫ് ആൻ അവറൊക്കെ വെച്ച് കഴിഞ്ഞാൽ ഇത് എന്ത് ചെയ്യുന്നു, ഡീഗ്രേഡ് ചെയ്ത് പോകുള്ളൂ. അപ്പോൾ എളുപ്പമല്ല അത് ഇതാക്കാൻ വേണ്ടി, ഡീഗ്രേഡ് ചെയ്യാൻ എളുപ്പമല്ല. അതുകൊണ്ട് തന്നെ ഈ ശരീരത്തിലേക്ക് പെട്ടെന്ന് എത്തിപ്പെടാൻ ചാൻസ് കൂടുതലാണ്. ഡിറ്റക്ഷൻ മെത്തേഡ്സ്. Analytical techniques include high performance liquid chromatography, enzyme linked immunoassay, അതായത് എലീസിയും HPLC ഒക്കെയാണ് നമ്മൾ യൂസ് ചെയ്യുക. ഫ്ലൂറസൻസ് ബേസ്ഡ് അസേ യൂസ് ചെയ്യും, TLC ഒക്കെ യൂസ് ചെയ്യും. എന്നാലും ഏറ്റവും LECയും HPLC മെയിനായിട്ട് യൂസ് ചെയ്യും. ഗ്ലോബൽ ഹെൽത്ത് ഇമ്പാക്ട് estimated to cause 25,000 to 1,55,000 liver cancers causes annually worldwide. ഇത് മൂലം ഇത്രത്തോളം ആൾക്കാർ മരണപ്പെടുന്നെന്ന് ആലോചിച്ച് നോക്കൂ. Particularly prevalent in sub-Saharan Africa and South Asia, where food safety measures are less robust. അതായത് കുറേ രാജ്യങ്ങളിൽ അഫ്ലാടോക്സിൻ്റെ ഗവൺമെൻറ്റിൻ്റെ ലോ ഇത് ചെയ്യുന്നുണ്ടെങ്കിലും എൻഫോഴ്സ് ചെയ്യുന്നുണ്ടെങ്കിലും കൃത്യമായിട്ട് അത് പാലിക്കപ്പെടില്ല. അങ്ങനെ ഒരുപാട് ആൾക്കാർക്ക് ഡിസീസ് കാർസിനോജനാണ്, ഹെപ്പറ്റോസില്ലാർ കാർസിനോമ ഉണ്ടാവും. അപ്പോൾ അത് തേർഡ് വേൾഡ് കൺട്രീസിലാണ് കൂടുതൽ ഉണ്ടാവുക. 8. Prevention and control. Agricultural practices. Crop rotation and use of resistant crop varieties. Proper harvesting, drying and storage techniques. Biological control. Application of non-toxic strains of Aspergillus to out complete toxin producing strains. അതൊക്കെയാണ് ബയോളജിക്കൽ കൺട്രോളിൽ. കെമിക്കൽ കൺട്രോൾ. യൂസ് ഫംഗിസൈഡ്സ് ആൻഡ് പോസ്റ്റ് ഹാർവസ്റ്റ് ട്രീറ്റ്മെന്റ്. അതൊക്കെ എത്രത്തോളം പ്രായോഗികമല്ല ഫംഗിസൈഡ്സ് ഒക്കെ യൂസ് ചെയ്യുന്നത്. ഫുഡ് പ്രോസസ്സിംഗ്. സോർട്ടിംഗ് ആൻഡ് ഡിസ്കാർഡിംഗ് വിസിബിലിറ്റി മോൾഡി ഗ്രെയിൻസ്. ഇതെന്താ വെച്ചാൽ ഹ്യൂമിഡിറ്റി, ടെമ്പറേച്ചർ, ക്ലൈമറ്റ് അത് കൺട്രോൾ ചെയ്തിട്ട് ചെയ്യാൻ പറ്റുകയാണെങ്കിൽ മാത്രമേ ചെയ്യുള്ളൂ. എങ്ങനെ ഹ്യൂമിഡിറ്റി ഉണ്ടെങ്കിൽ എന്തായാലും ഫംഗൽ ഗ്രോത്ത് ഉണ്ടാവും. ഒരു പരിധി വരെ ഇത് പ്രായോഗികമല്ല. അത് കിട്ടിക്കഴിഞ്ഞിട്ട് അടുത്തുള്ള ടെക്നോളജി നോക്കിയതിന് ശേഷം സ്റ്റോറേജിന് ശേഷം അത് സംഭരിച്ചതിന് ശേഷം എന്ത് ചെയ്യണം, വേറെ ടെക്നോളജീസ് യൂസ് ചെയ്തിട്ട് വേണം അതിൻ്റെ ടോക്സിൻ നമുക്ക് ഡീഗ്രേഡ് ചെയ്യാൻ വേണ്ടിയിട്ട്. ഫസ്റ്റ് ക്വസ്റ്റ്യൻ നമുക്ക് കുറച്ച് MCQയിലേക്ക് പോകാം. ഫസ്റ്റ് ക്വസ്റ്റ്യൻ. അതിനു മുമ്പ് ഇഫ് യു ലൈക്ക് ദിസ് ചാനൽ പ്ലീസ് ഷെയർ ആൻഡ് സബ്സ്ക്രൈബ്. ഡോണ്ട് ഫോർഗെറ്റ് ടു ക്ലിക്ക് ദ ബെൽ ബട്ടൺ. ചാനൽ സബ്സ്ക്രൈബ് ചെയ്യാൻ ആരും മറക്കരുത്. First question. In 2023, which country faced a ban on the export of peanuts due to excessive aflatoxin levels in shipments? India, Brazil, United States, Vietnam. Your answer is India. Explanation. In 2023, several peanut shipments from India were rejected in European Union markets due to non-compliance with aflatoxin limits, highlighting global regulatory concerns. Second question. Which advanced molecular technique has recently developed to detect aflatoxin B1 in contaminated food samples with higher sensitivity? CRISPR-based biosensor into MALDI-TOF and real-time PCR and NMR spectroscopy. ഇവിടെ ആൻസർ ചെയ്യാണ് CRISPR-based biosensors. CRISPR-based biosensors have been gaining traction for aflatoxin detection due to their rapid, precise and cost-effective applications. CRISPR എന്ന clustered regularly interspaced short palindromic repeats ആണ് CRISPR. Third question. What is the maximum allowable limit for aflatoxin M1 in infant formulas as per the Codex Alimentarius 2023 updated guidelines? Your answer is 0.05 microgram per kg. Fourth question. Which advanced analytical technique is considered the gold standard for detecting and quantifying aflatoxins in food samples with minimal matrix interference? GS ആണോ, ഗ്യാസ് ക്രോമാറ്റോഗ്രാഫി ആണോ, അല്ല ഹൈ പെർഫോമൻസ് ലിക്വിഡ് ക്രോമാറ്റോഗ്രാഫി ആണോ? അതെ, HPLC ആണ് ഇവിടെ നമ്മൾ യൂസ് ചെയ്യുന്നത്. HPLC ഫ്ലൂറസൻസ് ഡിറ്റക്ഷൻ ആണ് നമ്മൾ യൂസ് ചെയ്യുക, ഗോൾഡ് സ്റ്റാൻഡേർഡ് ആയിട്ട് കൺസിഡർ ചെയ്യും. ഫിഫ്ത്ത് ക്വസ്റ്റ്യൻ. വാട്ട് ഈസ് ദ മെയിൻ മെക്കാനിസം ബൈ വിച്ച് aflatoxin cause hepatocellular carcinoma in humans? Induction of oxidative stress, formation of DNA adducts, methylation of tumor suppressor genes. ഇതിൽ ആൻസർ ഏതാ? അതിൻ്റെ ആൻസർ ഒരു B ആണ്, ഫോർമേഷൻ ഓഫ് DNA adducts. Aflatoxin B1 forms DNA adducts particularly at guanine bases, leading to mutations in the TP53 gene and hepatocellular carcinoma. അപ്പോൾ അഡക്സ് ഫോർമേഷൻ ആണ് ഇതിൽ, DNA അഡക്സ് ഫോർമേഷൻ ആണ് ഇതിൻ്റെ പ്രത്യേകത. സിക്സ്ത് ക്വസ്റ്റ്യൻ. ദ റീസെന്റ് 2024 FSSAI guidelines mandate aflatoxin testing for which newly included food category? ഏതിനാണ് ഏറ്റവും കൂടുതൽ പ്രോട്ടീൻ സോഴ്സ് ആയിട്ട് യൂസ് ചെയ്യുന്നത്? ഇപ്പോൾ nowadays has been changed from meat to plant-based protein products. So here answer is plant-based protein products. So explanation. Due to the rising popularity of plant-based proteins, FSSAI now includes them in aflatoxin testing to prevent contamination risk during processing and storage. Seventh question. Which innovative packaging method was introduced in 2023 to limit aflatoxin formation during food storage? Vacuum sealed nitrogen flushing ആണോ, edible biopolymer coating ആണോ, active oxygen absorbing films? ആക്റ്റീവ് ഓക്സിജൻ അബ്സോർബിംഗ് ഫിലിംസ് ആണ് ഇതായിട്ട് യൂസ് ചെയ്തിട്ടുള്ളതാണ് അതിൻ്റെ ആൻസർ. ദ പാക്ചേജ് മെറ്റീരിയൽ ഇൻഹിബിറ്റ് ഫംഗൽ ഗ്രോത്ത്. അതിനെന്താണ് ഗുണം? ഫംഗൽ ഗ്രോത്തിനെ ഇൻഹിബിറ്റ് ചെയ്യും, ദെയർബൈ റെഡ്യൂസിങ് അഫ്ലാടോക്സിൻ കണ്ടാമിനേഷൻ ഇൻ സ്റ്റോർഡ് ഗ്രെയിൻസ് ആൻഡ് നട്ട്സ്. 8th question. Aflatoxins are known to be classified as which type of carcinogen by the International Agency for Research on Cancer? ഇന്റർനാഷണൽ IARC 3-4 ക്രൈറ്റീരിയ വെച്ചിട്ട് എന്ത് ചെയ്തിട്ടുണ്ട്? അതിനെ ക്ലാസിഫൈ ചെയ്തിട്ടുണ്ട് ക്യാൻസറിൻ്റെ. ഡിപ്പെൻഡ്സ് അപ്പോൺ ദ സിവിയറിറ്റി. ഹിയർ ഇറ്റ് കംസ് അണ്ടർ ഗ്രൂപ്പ് 1. ഗ്രൂപ്പ് 1-ലാണ് ഇന്റർനാഷണൽ ഏജൻസി ഫോർ റിസർച്ച് ക്യാൻസർ classified AFB1 and AFM1 as human carcinogens belongs to group 1 in the case of AFB1, and group 2B aflatoxin M1 classified in group 2B. So here answer is A, group 1 carcinogenic to humans. So that is the answer. 9th question. What role does the enzyme lactase play in the detoxification of aflatoxins in food according to 2024 research finding? It breaks down aflatoxin DNA, it catalyzes degradation of aflatoxin molecules, it inactivates fungal growth, it enhances food safety testing sensitivity. Here answer is option B, it catalyzes the degradation of aflatoxin molecules. Lactase enzymes degrade aflatoxin molecules. Lactase enzymes degrade aflatoxins in non-toxic byproducts, making them promising tools for aflatoxin detoxification in food. Certain mushrooms ഹെയിൽ വൈക്കോലിലാണ് നമ്മൾ ഗ്രോ ചെയ്യുക. ആ വൈക്കോലിൽ ലക്കേസ് എൻസൈംസ് ഉണ്ടാവും ചില മഷ്റൂംസ്. അപ്പോൾ മഷ്റൂം കൾട്ടിവേഷന് ശേഷം ആ ഹേ നമുക്ക് വൈക്കോൽ യൂസ് ചെയ്ത് കഴിഞ്ഞാൽ അതിലുള്ള ലക്കേസ് എൻസൈം യൂസ് ചെയ്തിട്ട് എന്ത് ചെയ്യാൻ പറ്റും? നമുക്ക് പൗഡർ ചെയ്തിട്ട് ഇതിനെ നമുക്ക് എന്ത് ചെയ്യാം? അഫ്ലാടോക്സിനെ ഡീഗ്രേഡ് ചെയ്യാൻ പറ്റും. 10th question. What is the current maximum limit for aflatoxin in spices as per FSSAI regulations? 15 microgram per kg, 25, 30, 35. Here answer is, according to new regulation, 30 microgram per kg. Here answer is 30 microgram per kg. Which microorganism is most commonly associated with the production of gas and frothing in milk? A. E. coli, B. Streptococcus lactis, C. Clostridium perfringens, D. Bacillus cereus. Here answer is A. E. coli. Second question. What is the optimal growth temperature for psychotrophic bacteria in milk? A. 25 to 30 degrees Celsius, B. 4 to 7 degrees Celsius, C. 50 to 55 degrees Celsius, D. 10 to 15 degrees Celsius. Your answer is B. 4 to 7 degrees Celsius. Third question. Which microorganism is associated with the production of ropey milk? A. Alkaligens viscolactis, B. Streptococcus thermophilus, C. Bacillus subtilis, D. Lactobacillus casei. Here answer is A. Alkaligens viscolactis. Fourth question. Which enzyme produced by psychotrophic bacteria is responsible for off flavors in milk? A. Protease, B. lipase, C. amylase, D. peroxidase. Here answer is B. lipase. Fifth question. Which microorganism is associated with the production of histamine in fermented milk products? A. Lactobacillus brevis, B. Streptococcus thermophilus, C. E. coli, D. Pseudomonas aeruginosa. Here answer is A. Lactobacillus brevis. Next question, sixth question. Which microorganism is primarily responsible for the bitty cream defect in milk? A. Bacillus cereus, B. Pseudomonas fluorescens, C. Micrococcus luteus, D. Streptococcus lactis. Here answer is A. Bacillus cereus. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Seventh question. The presence of Pseudomonas species in raw milk is of concern due to their ability to produce endotoxins at low temperature. B. Rapidly ferment lactose. C. Produce heat-resistant lipases and proteases. D. Cause souring during refrigeration. Your answer is C. Produce heat-resistant lipases and proteases. 8th question. Which microorganism is most commonly responsible for mastitis in dairy animals leading to bacterial contamination of milk? A. Staphylococcus aureus. B. Streptococcus thermophilus. C. Lactobacillus acidophilus. D. Clostridium sporogens. Your answer is A. Staphylococcus aureus. 9th question. Which microorganism is capable of growing in milk stored at sub-zero temperatures, posing a challenge for frozen milk products? A. Psychrobacter species. B. Salmonella typhi. C. Listeria monocytogenes. D. Streptococcus agalactiae. Your answer is Psychrobacter species A. 10th question. Which anaerobic spore-forming bacterium causes milk spoilage characterized by stormy fermentation? A. Clostridium botulinum. B. Clostridium tyrobutylicum. C. Clostridium perfringens. D. Clostridium sporogens. Your answer is C. Clostridium perfringens. Next question. Which test is used to detect presence of formalin in milk? A. Starch iodine test. B. Chromotropic acid test. C. Gerber test. D. Prosylic acid test. Your answer is B. Chromotropic acid test. What is the Gerber method? It is a primary and historic chemical test to determine the fat content of substances most commonly in milk. The Gerber method was developed and patented by Dr. Nicholas Gerber of Switzerland in 1891. In this method, milk fat is separated from proteins by adding sulfuric acid of that particular specific gravity. What is borax and boric acid test? It is a test in milk. If the turmeric paper turns red, it indicates the presence of borax or boric acid. Confirmation can be made by adding a drop of ammonia solution on the turmeric paper, and if the red color changes to green, it shows the presence of boric acid. Next, detection of neutralizers in milk. Prosylic acid test or soda test. In 5 ml of milk, add 5 ml alcohol followed by 4 to 5 drops of prosylic acid. If the color of milk changes to pinkish red, then the milk is adulterated with sodium carbonate or sodium bicarbonate and hence unfit for human consumption. Next question. Which fatty acid is predominant in cow milk fat? A. Oleic acid. B. Palmitic acid. C. Linoleic acid. D. Stearic acid. Here answer is B. Palmitic acid. Next question. What is the primary objective of the phosphatase test in milk? To detect added water, to confirm proper pasteurization, to measure protein content, to assess bacterial contamination. Your answer is B. To confirm proper pasteurization. Next question. What is the primary function of the lactoperoxidase system in milk? A. Enhance the flavor. B. Act as a natural antibacterial agent. C. Increase milk viscosity. D. Boost fat content. Here answer is B. Act as a natural antibacterial agent. The lactoperoxidase is an oxidoreductase enzyme secreted in the milk having an important function in the protection of the mammary glands, lactation and intestinal tract in newborn children against pathogenic microorganisms. It is a paper cited Naidu 2000. Next question. The presence of ropey or stingy milk is often caused by Lactococcus lactis, B. Bacillus cereus, C. Alkaligens viscolactis, D. Streptococcus thermophilus. Which is the answer? Here answer is C. Alkaligens viscolactis. Unravel the mystery of the melamine incident. What is melamine incident? First question. Which chemical is the culprit of Chinese milk scandal 2008? A. Urea, B. Malathion, C. Melamine, D. Formalin. Which is the answer? Here answer is C. Melamine. Melamine is the answer. In 2008, a melamine contaminated milk powder incident occurred in China, which caused many urinary calculi in infants and young children, means urinary stones. The Sanlu Group, one of the largest dairy producers in China, was identified as the chief culprit. But as the scandal unfolded, more Chinese dairy farms became implicated. And some of these children developed kidney injury and even died. And almost 3 lakhs Chinese infants and young children with 6 reported deaths. What is melamine? Melamine had been deliberately added at milk collecting stations. For what purpose they added melamine? Melamine they added to dilute, to increase the nitrogen concentration or to increase the protein concentration of the milk. Melamine 2,4,6 triamino 1,3,5 triazine is a heterocyclic compound containing 6 atoms of nitrogen. As a result of its composition, it contains 66% of nitrogen and in its molecule. It is a raw material in the production of plastics, pesticides, cleaning agents, fertilizers and many other products. So, in milk, to increase the nitrogen concentration, they are adding melamine. Unravel the mystery of the Parmesan cheese fraud 2016. Most of the Parmesan cheeses are adulterated with 2 to 3 types of cheeses with wood pulp. So FDA in USA says Parmesan cheese might be cheddar or wood pulp. Means some of the companies adulterated Parmesan cheese with cheddar, wood pulp or mozzarella cheese. Next, unravel the mystery of the capnophiles. What are capnophiles? Capnophiles means capnophiles are microorganisms that thrive in the presence of high concentrations of carbon dioxide. What are the examples of capnophiles? Campylobacter, Neisseria gonorrhoeae. Neisseria gonorrhoeae is facultative anaerobic capnophiles. Haemophilus influenzae, Streptococcus pneumoniae. Not all the Streptococcus pneumoniae are capnophiles. About 8% of all pneumococcal isolates are capnophiles. And Helicobacter pylori is also capnophile. So these are the bacteria examples for capnophiles. Food Safety Officer book is available with 1000 MCQs from all sections. Contact number is mentioned here. Amazon link is available in the description. Which scientist is primarily credited with the development of the modern atomic absorption spectrophotometer? A. Albert Einstein, B. Alan Walsh, C. Joseph Priestley, D. Ernest Rutherford. Which is the answer? Your answer is option B. Alan Walsh. Second question. The principle of AAS was indirectly inspired by which fundamental law? A. Beers-Lambert's law, B. Planck's quantum theory, C. Newton's law of cooling, D. Dalton's atomic theory. Which is the answer? Your answer is option A. Beers' law. Third question, atomic absorption spectroscopy is used to detect metals, non-metals, semi-metals, all of these. Here, which is the answer? Your answer is A, metals. Fourth question, atomic absorption spectroscopy gained prominence in the food industry for detecting which toxic metal? Cadmium, mercury, lead, all of these. Your answer is option D of these. Fifth question, which other spectroscopic method is most commonly confused with AAS due to similar applications? A. Ultraviolet visible spectroscopy. B. Inductively coupled plasma spectroscopy. C. Mass spectrometry. D. Fluorescence spectroscopy. Which is the answer? Your answer is option B. Inductively coupled plasma spectroscopy. Sixth question, what is the role of the monochromator in AAS? To atomize the sample, to separate the light source into its component wavelengths, to amplify the signal from the detector, to vaporize the sample. Which is the answer? Here answer is option B, to separate the light source into its component wavelengths. Explanation. The monochromator isolates the specific wavelength of light absorbed by the analyte, ensuring that only the relevant spectral line is measured. Seventh question. Which type of light source is typically employed in AAS for element-specific analysis? Tungsten lamp, B. Xenon lamp, C. Hollow cathode lamp, D. LED. Which is the answer? Your answer is option C. Hollow cathode lamp is used. Eighth question. In AAS, the term matrix effect primarily refers to A. The absorption of light by the sample solvent, B. The interference caused by the sample's composition, C. The scattering of light by particulate matter, D. The reduction in detector sensitivity over time. Which is the answer? Your answer is option B. The interference caused by the sample's composition. Food Safety Officer book is available with 1000 MCQs from all sections. Contact numbers are mentioned here. Amazon link is available in the description. You can directly order from this Amazon link. Ninth question. Which of the following is the principle of atomic absorption spectroscopy? Radiation is absorbed by non-excited atoms in vapor state and are excited to higher states. P. Medium absorbs radiation and transmitted radiation is measured. C. Color is measured. D. Color is simply observed. Which is the answer? Your answer is option A. Atoms in gaseous state absorbs the radiation and are excited to higher state. Since the higher state is unstable, the atom returns the ground state with the emissions of radiation, which is measured in AAS. 10th question. In AAS, what material is used to construct the cathode in the hollow cathode lamp? A. Tungsten. Option B. Quartz. Option C. The element being analyzed. Option D. Aluminum. Your answer is option C. The element being analyzed. 11th question. In food safety, AAS is preferred for detecting heavy metals due to high throughput analysis, high sensitivity and element specificity, cost effectiveness, resistance to matrix interference. Which is the answer? Your answer is option B, high sensitivity and element specificity. 12th question. Why is a deuterium lamp used as a background correction in AAS? A. It provides a continuous spectrum. Option B. It is more sensitive than a hollow cathode lamp. C. It eliminates stray light interference. D. It corrects for scattering and molecular absorption. Which is the answer? Here answer is option D. It corrects for scattering and molecular absorption. 13th question. What is the main advantage of graphite furnace AAS, GFAAS over flame AAS? A. Faster analysis time. B. Higher sensitivity for trace metals. C. Simplicity of operation. D. Ability to analyze gases. Which is the answer? Your answer is option B. Higher sensitivity for trace metals. 14th question. Which food matrix is most likely to cause matrix interference in AAS? A. Water. B. Oils and fats. C. Grains. D. Beverages. Which is the answer? Your answer is option B. Oils and fats. 15th question. Which chemical modifier is used to reduce interference during GFAAS analysis? A. Magnesium nitrate. B. Sodium chloride. C. Potassium permanganate. D. Ammonium sulfate. Which is the answer? Your answer is option A. Magnesium nitrate. Mixtures of palladium nitrate and magnesium nitrates are regularly used. 16th question. AAS is limited to detecting metals and metalloids because A. Non-metals do not absorb visible light. B. Non-metals lack free atoms in the ground state. C. Non-metals cannot be atomized effectively. D. Non-metals have too high excitation energies. Which is the answer? Your answer is option B. Non-metals lack free atoms in the ground state. 17th question. What is the typical detection range of AAS for most metals? Microgram per liter, gram per liter, parts per million, parts per billion. Which is the answer? Your answer is option D. Parts per billion. 18th question. In AAS, which gas is commonly used in the flame atomizer to enhance combustion? Helium, argon, acetylene, hydrogen. Which is the answer? Your answer is option C. Acetylene. There are two common gas mixtures that are burnt to fuel the flame. They are air acetylene and nitrous oxide acetylene, producing a flame around 2300 degrees Celsius. Air acetylene is suitable for most of the elements. 19th question. What is the primary function of the atomizer in the emission system of AAS? A. To split the beam into two. B. To convert steady light into pulsating light. C. To break large volumes of liquid into fine droplets. D. To reduce the sample to its atomic state. Which is the answer? Your answer is option C. To break large volumes of liquid into fine droplets. Food Safety Officer book is available with 1000 MCQs from all sections. You can directly order in using Amazon link, or you can contact these numbers. 20. What is the primary advantage of Zeeman background correction in AAS? It corrects for chemical interferences in the sample. It minimizes background noise caused by molecular absorption. It increases the sensitivity of heavy metal detection. It simplifies the calibration process. Here option B is correct. It minimizes background noise caused by molecular absorption. 21. The operating temperature of graphite furnace atomizer typically ranges from 200 degrees Celsius to 600, 500 to 1000, 1200 to 3000, 3000 to 5000. This is the answer. Here answer is option C, 1200 to 3000. 22. Why are inert gases such as argon or nitrogen used in the operation of graphite furnace AAS? To stabilize the flame in the atomizer, to prevent oxidation of the graphite tube, to enhance atomization of non-metallic compounds, to improve the resolution of the monochromator. Your answer is B. To prevent oxidation of graphite tube. 23. The limit of detection in AAS refers to the maximum concentration of the instrument can measure accurately, the smallest concentration of analyte that can be reliably detected. See the threshold beyond which signal noise dominates the output, the point at which calibration fails. That limit of detection means the smallest concentration of analyte that can be reliably detected, option B. 24. In atomic absorption spectroscopy, the most strongly absorbed light is called as resonance line, streamline, baseline, stroke line. The answer here answer is option A, resonance line. Next question, 25. This is the last question of this session. In AAS, the graph drawn between concentration versus emission, absorbance versus concentration, absorbance versus wavelength. D, none of this. Your answer is option B, absorbance versus concentration. Codex Alimentarius Commission Facts and MCQs. The Codex Alimentarius Commission was established by which two organizations? A. FAO and WHO. B. World Trade Organization and World Health Organization. C. Food and Agriculture Organization and World Trade Organization. D. United Nations and World Health Organization. What is the answer? Your answer is option A. FAO and WHO. The 11th session of Food and Agriculture Organization Conference in 1961 and the 16th World Health Assembly in 1963 both passed resolutions to establish the Codex Alimentarius Commission. The two bodies also adopted the statutes and rules of procedures for the Commission. Second question. What is the primary purpose of the Codex Alimentarius Commission? A. Promote global trade. Option B. Protect the health of consumers, ensure fair practices in the food trade. Option C. Harmonize food labeling laws. Option D. Regulate genetically modified organisms. Your answer is option B. Protect the health of consumers, ensure fair practices in the food trade. The main purpose of this program is to protect the health of consumers, ensure fair practices in the food trade, and promote coordination of all food standards work undertaken by international governmental and non-governmental organizations. Third question. Which year marked the establishment of Codex Alimentarius Commission? 1958, 1963, 1971, 1985. Which is the answer? Your answer is option B, 1963. The Codex Alimentarius Commission is an international food standards body established jointly by FAO and the World Health Organization in May 1963 with the objective of protecting consumers' health and ensuring fair practices in food trade. Fourth question. The term Codex Alimentarius is derived from which language? Latin, Greek, French, Spanish. Which is the answer? Here answer is option A, Latin. The term Codex Alimentarius is taken from Latin and means food code. It is the code of food standards for all nations. Food standards for all nations. Fifth question. What does Codex Alimentarius translate to in English? Food guide, food law, food code, food standards. Which is the answer? Your answer is food code. Sixth question. The Codex standards are advisory, but they are referenced in dispute under which international agreement? General Agreement on Tariffs and Trade, GATT. B. Agreement on the application of sanitary and phytosanitary measures, SPS agreement. C. Trade-related aspects of intellectual property rights, TRIPS. D. Agreement on agriculture. Which is the answer? Your answer is option B, SPS agreement. Codex standards are used worldwide to harmonize national food safety regulations and are recognized in the WTO agreement on the application of sanitary and phytosanitary measures as the international reference point for food safety. Food Safety Officer book is available with 1000 MCQs from all sections. You can purchase it from link which is highlighted in the description. Seventh question. Which Codex committee focuses on pesticide residues in food? Codex Committee on Food Hygiene, Codex Committee on Residues of Veterinary Drugs, Codex Committee on Pesticide Residues, Codex Committee on Contaminants in Food. Which is the answer? Your answer is option C, Codex Committee on Pesticide Residues, CCPR. Seventh question. The Codex General Principles document provides guidelines for setting maximum residue limits, conducting microbiological analysis, risk analysis in food safety, labeling organic foods. Your answer is option C, risk analysis in food safety. Ninth question. Which body administers the Codex Trust Fund? WTO, WHO, FAO, Joint FAO, WHO Secretariat. Which is the answer? Your answer is option D, Joint Food and Agriculture Organization, World Health Organization Secretariat. 10th question. Codex standards for food additives are established by which committee? CCFH, CCFA, CCFL, CCNFSDU. Which is the answer? Your answer is option B, CCFA, Codex Committee on Food Additives. 11th question. Codex Alimentarius Commission headquarters is located at New York, New Delhi, Rome, Madrid. Your answer is option C, Rome. 12th question. How many member countries are there in the Codex Alimentarius Commission? CAC 196, option B 195, option C 188, option D 100. Which is the answer? Your answer is option C 188. The Codex Alimentarius Commission CAC is the international food standard setting body comprised of 189 members. Total of members 189 members, including 188 member countries plus one member organization of European Union. It is part of the Joint Food and Agriculture Organization and World Health Organization Food Standards Program. 13th question. Which country has recently become a member of the Codex Alimentarius Commission? India, USA, Japan, Bangladesh. Here answer is option A, India. India has been unanimously elected as a member representing Asian region in the Executive Committee of Codex Alimentarius Commission during its 46th meeting being held at Food and Agriculture Organization headquarters at Rome. 14th question. What is the Codex General Standard for food additives known as Codex Standard 201, Codex Standard 192, option C, Codex Standard 3, option D, Codex Standard 193. Which is the answer? Here answer is option D, Codex Standard 192. 15th question. Who is the chairperson of Codex Alimentarius Commission? A. Shane Warne, option B, Stephen Wayne, option C, Donald Trump, option D, Shiv Khera. Which is the answer? Your answer is B, Stephen Wayne. Tea, cocoa and coffee MCQs. Criollo is a variety of cocoa. Criollo is a variety of cocoa. Second question. The International Coffee Organization headquarters is located at? Where is International Coffee Organization headquarters is located? London. Answer is London. Third, cappuccino ingredients are espresso, milk and foam are the ingredients of cappuccino. Fourth question, which country is the top producer of coffee? Answer is Brazil. Brazil is the top producer of coffee. Fifth question, which place is considered as the coffee capital of India? The coffee capital of India is situated in Karnataka. Kur is the coffee capital of India. Sixth question, which city is known as tea city of India? Tea cities, a city of India is Dibrugarh, Assam. Dibrugarh in Assam is known as tea city of India. Seventh question, which city is known as the tea capital of the world? Which city is known as the tea capital of the world? Jorhat. Jorhat is known as the tea capital of the world. Eighth question. The place is known as the chocolate capital of the world is? Which place is known as the chocolate capital of the world? Brussels, Belgium. Brussels is known as the chocolate capital of the world. Ninth question. Which country is producing the highest quality cocoa? Which country is producing the highest quality of cocoa? Answer is Ecuador. Ecuador is producing the highest topmost quality of cocoa. 10th question. Which country is the largest producer of cocoa in the world? Which country is the largest producer of cocoa in the world? Answer is Ivory Coast. Ivory Coast is the largest producer of cocoa. 11th question. Which country is the global leader in tea production? Which country is the global leader in the tea production? Answer is China. China is playing crucial or leading role in tea production. 12th question. Which country consumes the most tea per capita? Which country? Turkey. Turkey consumes a lot of tea. 13. Which country has the highest per capita coffee consumption? Which country has the highest per capita coffee consumption? Answer is Finland. 14. Headquarters of Tea Board of India is located in? Tea Board headquarters is located in Kolkata. 15th question. Who invented espresso coffee? Luigi Bezzera. Who invented espresso coffee? Luigi Bezzera. 16th question. A heat-tolerant, drought-resistant species of coffee bean discovered by French Agricultural Research Center for International Development. What is the name of that particular coffee plant? Coffea stenophila. Most of the world's high-end coffee lovers enjoy drinking Arabica, Coffea arabica, which is under the threat from rising global temperature. It cannot withstand high temperature. Global warming is the threat for Coffea arabica. But stenophila can thrive in temperature 6 degrees Celsius above that of the beloved artisan bean. So it can withstand more temperature and environment friendly. So Coffea stenophila is the answer. 17th question. Which is the catechin produced showing Anti-cancer properties in green tea. Epigallocatechin is the catechin compound showing anti-cancer properties in green tea, known as EGCG. 18th question. International Cocoa Organization head office is situated at sorry. International Cocoa Organization head office is situated at Abidjan, Ivory Coast. Abidjan, Ivory Coast. 19th question. What is Kopi Luwak? We all heard about this Kopi Luwak recently in news. What is the peculiarity of Kopi Luwak? Kopi Luwak, also called a civet coffee. It is made from coffee cherries, partially digested by the Asian palm civet. Name of that Asian palm civet is Paradoxurus hermaphroditus. As the cherries pass through the civet's digestive system, the enzyme will act upon that particular beans and they undergo natural fermentation. And after being excreted along with other waste, they are collected for processing, and we use to make coffee from that beans. That coffee is known as Kopi Luwak. 20th question, last question. Which tea is considered as the finest tea in the world? Which tea is considered as the finest tea in the world? Answer is First Flush Darjeeling tea is considered as the finest tea in the world. First Flush Darjeeling tea. Foodborne diseases. First question. Neuroletharism. Statement. Neuroletharism is associated with the consumption of Lathyrus sativus or grass pea. The primary toxin responsible for neuroletharism is beta-ODAP. Symptoms include progressive and irreversible spastic paralysis of the lower limbs. The condition is prevalent in regions with drought-prone climates where other food sources are scarce. Conclusion. Which of the following best describes the primary cause and clinical outcome of neuroletharism? Options A. Lathyrus sativus is the only plant responsible and it causes temporary paralysis. B. Lathyrus sativus contains beta-ODAP which causes permanent spastic paralysis. C. Neuroletharism occurs only in tropical climates. D. The toxin responsible is ODAP, causing both upper and lower limb paralysis. Which is the answer? Here answer is B. Lathyrus sativus contains beta-ODAP which causes permanent spastic paralysis. Explanation. Lathyrus sativus contains beta-ODAP and the condition leads to permanent lower limb paralysis, especially in resource poor areas. Next, second question. Aflatoxins. Aflatoxins statements. Aflatoxins are produced by Aspergillus flavus and Aspergillus parasiticus. Aflatoxin B1 is considered the most potent hepatocarcinogen. Aflatoxins are commonly found in peanuts, corn, and tree nuts. Regulatory limits for aflatoxins are the same worldwide due to their carcinogenicity. Conclusion. What best summarizes the characteristics and concerns of aflatoxins? Options. A. Aflatoxins are produced by multiple molds and can be found in all food crops. B. Aflatoxin B1 is highly carcinogenic and is primarily a concern in grain and nut storage. C. Regulatory limits for aflatoxins vary by country despite their non-carcinogenicity. D. Aflatoxins are safe at low levels but accumulate in fatty tissues. Which is the correct answer over here? The answer is C. Regulatory limits for aflatoxins vary by country despite their non-carcinogenicity. Explanation. While aflatoxins are highly carcinogenic, countries establish different limits based on detection technology and acceptable risk levels. Third question. Ergodism. Statements. Ergodism results from ingesting alkaloid produced by Claviceps purpureae. Symptoms of ergodism include hallucinations, convulsions, and gangrene. It predominantly affects cereal crops like rye. Historically, ergotism has been linked to St. Anthony's fire in medieval Europe. Conclusion: which of the following best describes ergotism and its historical context? Options: A. Ergotism is caused by fungal contamination in tropical climates only. B. Claviceps purpurea affects all cereal crops equally, causing hallucinations. C. Ergotism symptoms include gangrene, linked historically to St. Anthony's fire. D. Ergotism has been eradicated in modern farming with no health concern today. Which is the answer? Your answer is C. Ergotism symptoms include gangrene, linked historically to St. Anthony's fire. Here explanation: ergot alkaloids from Claviceps purpurea cause severe symptoms and historical outbreak were referred to as St. Anthony's fire. Fourth question: epidemic dropsy. Statements: epidemic dropsy is caused by the consumption of mustard oil adulterated with argemone oil. Symptoms include edema, glaucoma, and breathlessness. Sanguinarine in argemone oil is toxic and affects the cardiovascular system. The condition is commonly seen in countries with mustard oil usage in cooking. Conclusion: what best characterizes the cause and symptoms of epidemic dropsy? Options: A. Sanguinarine causes neurological symptoms mainly affecting mustard consumers. B. Epidemic dropsy is a cardiovascular condition from argemone oil adulteration. C. Glaucoma is the most severe symptom of epidemic dropsy. D. The use of pure mustard oil prevents epidemic dropsy entirely. Answer over here. Your answer is B. Epidemic dropsy is a cardiovascular condition from argemone oil adulteration. Explanation: sanguinarine in argemone oil causes cardiovascular symptoms, primarily affecting populations using mustard oil. Fifth question: typhoid fever. Statements: typhoid fever is caused by Salmonella typhi. The disease is transmitted via the fecal oral route. Symptoms include prolonged fever, abdominal pain, and rash. Typhoid vaccines are effective in preventing infection. Conclusion: which option best summarizes the transmission and control of typhoid fever? Option A. Typhoid fever is spread by airborne droplets and prevented by vaccines. B. Salmonella typhi is waterborne and vaccination is the only preventive measure. C. Typhoid spreads via the fecal oral route with vaccination providing protection. D. Antibiotics are ineffective in treating typhoid due to its resistance. Here, which is the answer? Your answer is C. Typhoid spreads via the fecal oral route with vaccination providing protection. Explanation: Salmonella typhi spreads through contaminated water or food, and vaccines are effective for prevention. Sixth question: salmonellosis. Statement: salmonellosis is caused by multiple Salmonella serotypes, primarily Salmonella enteritidis and Salmonella typhimurium. It is commonly associated with contaminated poultry, eggs, and dairy products. Symptoms include gastroenteritis, abdominal cramps, and diarrhea. Salmonellosis can be severe in immunocompromised individuals and young children. Conclusion: which of the following best explains the transmission and risk factors associated with salmonellosis? Options: A. Only Salmonella enteritidis causes salmonellosis and it's limited to dairy products. B. Multiple Salmonella serotypes can cause infection with severity higher in immunocompromised groups. C. Salmonellosis is a respiratory disease with severe symptoms in all age groups. D. Only eggs transmit Salmonella as it cannot survive on other foods. Which is Here answer is B. Multiple Salmonella serotypes can cause infection with severity higher in immunocompromised groups. Explanation: salmonellosis is caused by various salmonella serotypes and its severity varies with host immunity and age. Seventh question: staphylococcal intoxication. Statements: staphylococcal food poisoning is due to the ingestion of enterotoxins produced by Staphylococcus aureus. The toxins are heat stable and can survive cooking processes. Symptoms appear quickly, typically within 1 to 6 hours after consumption. Staphylococcal intoxication is often associated with improperly stored or handled foods. Conclusion: what is the best characterization of staphylococcal food poisoning? A. It is caused by direct bacterial invasion of the gut. Symptoms develop slowly days after ingestion. The toxins are heat sensitive, so proper cooking can prevent intoxication. D. Staphylococcus aureus enterotoxins cause rapid onset food poisoning. Which is the answer? Here answer is D. Staphylococcus aureus enterotoxins cause rapid onset food poisoning. Explanation: staphylococcal food poisoning is caused by heat stable toxins, leading to rapid onset symptoms shortly after ingestion. Eighth question: botulism. Statements: botulism is caused by Clostridium botulinum, which produces a potential neurotoxin. The toxin blocks acetylcholine release, leading to muscle paralysis. Infant botulism can occur from spore in honey, which germinate in the infant gut. Symptoms of botulism include difficulty in swallowing, muscle weakness, and respiratory paralysis. Conclusion: which of the following best describes the cause and effects of botulism? Option A. Clostridium botulinum infection is due to inhaling spores affecting only adults. B. The botulinum toxin causes muscle rigidity by increasing acetylcholine release. Botulism, botulism results from a neurotoxin that inhibits acetylcholine, leading to paralysis. D. Botulism can only occur in infants consuming honey. Which is the answer? Here answer is C. Botulism results from a neurotoxin that inhibits acetylcholine, leading to paralysis. Explanation: botulinum toxin blocks acetylcholine, causing muscle weakness and paralysis, and it can affect all ages depending on exposure. Ninth question: Bacillus cereus food poisoning. Statements: Bacillus cereus produces two types of toxins, an emetic toxin and an enterotoxin. The emetic toxin causes vomiting, while the enterotoxin causes diarrhea. Bacillus cereus is commonly found in starchy foods like rice and pasta. Heat treatment can kill Bacillus cereus, but its spores and toxins may survive. Conclusion: which of the following is true regarding Bacillus cereus food poisoning? Options: only one toxin type is produced, causing primary diarrhea. B. The emetic toxin affects dairy products more than rice. C. Bacillus cereus produces both emetic and enterotoxins associated with starchy foods. D. Proper cooking completely prevents all Bacillus cereus foodborne illness. Which is the answer? Here answer is C. Bacillus cereus produces both emetic and enterotoxins associated with starchy foods. Bacillus cereus produces two toxins that cause different symptoms. Spores can survive cooking, especially in starchy foods. 10th question: E. coli diarrhea. Statements: E. coli strains causing diarrhea is often enterotoxigenic ETEC or EHEC. EHEC strains like E. coli O157 can cause hemorrhagic colitis and hemolytic uremic syndrome. ETEC strains cause travelers diarrhea by producing enterotoxins. Proper cooking and hygiene are effective in preventing E. coli infections. Conclusion: which best describes E. coli strains and the infections they cause? Options: A. Only EHEC strains are pathogenic in humans. B. ETEC strains are more dangerous than EHEC, often causing HUS, means hemolytic uremic syndrome. Both EHEC ETEC strains can cause diarrhea, with EHEC linked to HUS. E. coli infections are common in dairy products only. Which is the answer? Here answer is C. Both EHEC and ETEC strains can cause diarrhea with EHEC linked to HUS. Explanation. ETEC cause mild diarrhea while EHEC can cause severe disease including HUS. Cholera. Statements. Cholera is caused by Vibrio cholerae, particularly serogroups O1 and O139. The cholera toxin induces watery diarrhea known as rice water stools. Transmission is typically through contaminated water and food. Vaccination and sanitization measures are effective in cholera prevention. Conclusion. Which of the following best describes cholera? Option A. Cholera is an airborne disease caused by Vibrio cholerae O1 and O139. B. Cholera toxin causes severe dehydration by inducing watery diarrhea. C. Only contaminated food spreads cholera, not water. D. Cholera can be prevented solely by vaccination without sanitation measures. Here, which is the answer? Here, answer is option B. Cholera toxin causes severe dehydration by inducing watery diarrhea. Explanation. Cholera is transmitted through contaminated water, with the cholera toxin causing profuse watery diarrhea and dehydration. Second question, Shigellosis. Statements. Shigellosis is caused by Shigella species, primarily Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella sonni. The Shiga toxin produced by some Shigella toxins strains can lead to hemolytic uremic syndrome or HUS. Shigellosis is primarily transmitted through the fecal oral route. Symptoms include bloody diarrhea, fever, and abdominal pain. Conclusion. Which of the following best describes Shigella infections? A. Shigella is airborne and rarely causes severe symptoms. B. Only Shigella dysentery produces Shigella toxin linked to HUS. C. Shigella spreads via the fecal oral route with symptoms including bloody diarrhea. D. Shigellosis is mild and self-limiting in most cases, requiring no treatment. Answer is option C. Shigella spreads via the fecal oral route with symptoms including bloody diarrhea. Explanation. Shigella spreads via contaminated food or water, and its symptoms can include severe bloody diarrhea and abdominal pain. If you like this channel, please share and subscribe. Don't forget to click the bell button. Third question, brucellosis. Statements. Brucellosis is caused by Brucella species, primarily Brucella abortus in cattle, Brucella melitensis in goats, Brucella suis in pigs. It is transmitted to humans through direct contact with infected animals, consumption of unpasteurized dairy products, and inhalation of aerosols. Brucellosis primarily affects the liver and spleen, leading to a chronic condition with symptoms such as fever, joint pain, and malaise. The disease can lead to complications such as endocarditis and osteomyelitis. Conclusion. Which of the following is the most accurate description of brucellosis transmission and symptoms? Options. A. Brucellosis is transmitted only through direct contact with animals with no risk associated with food consumption. B. Brucella species are commonly transmitted through unpasteurized dairy, causing flu-like symptoms and chronic complications. C. Brucellosis primarily causes gastrointestinal symptoms and is easily transmitted or treated with over-the-counter medications. Brucellosis can be transmitted via the respiratory route but only affects cattle and does not affect humans. Which is answer here? Answer is option B. Brucellosis species are commonly transmitted through unpasteurized dairy and causing flu-like symptoms and chronic complications. Explanation. Brucellosis is transmitted through contact with infected animals and consumption of unpasteurized dairy. It causes systemic infections with long-term complications. Fourth question. Typhoid fever. Statements. Typhoid fever is caused by Salmonella enterica seroba typhi. Symptoms include prolonged fever, abdominal pain, and sometimes a characteristic rose-colored rash. The bacteria are transmitted through contaminated water and food, often in areas with poor sanitation. Typhoid fever can cause complications such as intestinal perforation and septicemia. Conclusion. Which of the following correctly describes transmission and complications of typhoid fever? A. Typhoid fever is primarily spread through respiratory droplets with GI symptoms being rare. B. Salmonella typhi is transmitted through contaminated food or water and the disease can lead to severe complications if untreated. C. Typhoid fever is caused by a viral infection and is not associated with foodborne transmission. D. Only travelers to developing countries are at risk of contracting typhoid fever. Here which is the answer? Here answer is option B. Salmonella typhi is transmitted through contaminated food or water and the disease can lead to severe complications if untreated. Explanation. Typhoid fever is a foodborne infection caused by Salmonella typhi with the potential for severe complications if not treated, especially in resource-limited areas. Fifth question. Aflatoxins. Statements. Aflatoxins are a group of mycotoxins produced by Aspergillus species, particularly Aspergillus flavus and Aspergillus parasiticus. These toxins are potent carcinogens and are most commonly found in improperly stored crops like peanuts, maize, and tree nuts. Aflatoxin exposure can cause acute poisoning, liver damage, and long-term carcinogenic effects. Aflatoxin contamination is regulated by food safety agencies, which set maximum allowable levels in food products. Conclusion. Which of the following is true regarding aflatoxins? A. Aflatoxins are produced by bacteria and are not carcinogenic. B. Aspergillus species produce aflatoxins which are highly regulated and can cause liver cancer and acute toxicity. C. Aflatoxins are primarily found in dairy products which cause respiratory problems. D. There is no regulatory limit for aflatoxin levels in food and it is not associated with cancer. Here which is the answer? Here answer is option B. Aspergillus species produce aflatoxins which are highly regulated and can cause liver cancer and acute toxicity. Explanation. Aflatoxins are produced by Aspergillus species and are highly regulated due to their toxic and carcinogenic effects. Sixth question. Ergodism. Statements. Ergodism is caused by alkaloid toxins produced by Claviceps purpurea fungus, which infects cereal grains like rye. Symptoms of ergodism include vasoconstriction, gangrene, and neurological disturbances. Historically, ergodism was linked to the Salem witch trials, where it was believed to be the cause of mass hysteria. The toxin produced by Claviceps is used in modern medicine for inducing labor but can cause serious health issues when consumed in high amounts. Conclusion. Which of the following is the most accurate description of ergodism? Option A. Ergodism is caused by bacterial contamination of wheat with minimal symptoms. B. Ergodism symptoms include neurological and vascular symptoms caused by toxins from Claviceps purpurea. C. Ergodism only affects humans who eat contaminated wheat and does not impact livestock. D. The toxin produced by Claviceps is safe and widely used in medicinal practices without side effects. Answer is option B. Ergodism symptoms include neurological and vascular symptoms caused by toxin from Claviceps purpurea. Explanation. Ergodism is caused by Claviceps purpurea toxins, which lead to severe vascular and neurological symptoms due to vasoconstriction. 8. Aflatoxins. Statements. Aflatoxins are produced by Aspergillus flavus and Aspergillus parasiticus. They are found in foods like peanuts, maize, and cottonseed and are highly carcinogenic. Aflatoxins are regulated by food safety agencies and must be minimized in agricultural products. Aflatoxin B1 is the most potent and well-studied aflatoxin in terms of carcinogenicity. Conclusion. Which statement about aflatoxins is true? A. Aflatoxins are not a concern in agriculture products due to effective prevention methods. B. Aspergillus produces aflatoxins which can cause liver cancer, especially aflatoxin B1. C. Aflatoxins are only found in fruits and vegetables, not grains. D. Aflatoxin contamination is not regulated or monitored in food products. Here which is answer? Here answer is option B. Aspergillus produces aflatoxins which can cause liver cancer, especially aflatoxin B1. Explanation. Aflatoxins, particularly aflatoxin B1, are potent carcinogens produced by Aspergillus species and pose serious risks when present in food. Ninth question. Typhoid fever. Statements. Typhoid fever is caused by Salmonella enterica serotype typhi. Symptoms include prolonged fever, abdominal pain, and characteristic rose-colored rash. Typhoid fever can be fatal if not treated with appropriate antibiotics. Typhoid fever is primarily transmitted through contaminated water and food in areas with poor sanitation. Conclusion. Which of the following best describes the transmission and symptoms of typhoid fever? A. Typhoid fever is transmitted through the air and affects primarily the lungs. B. Salmonella typhi is transmitted through contaminated food and water, causing systemic symptoms including fever and rash. C. Typhoid fever symptoms include gastrointestinal distress, but it does not affect other systems. D. Typhoid fever can be cured without antibiotics if treated early. Your answer is option B. Salmonella typhi is transmitted through contaminated food and water, causing systemic symptoms including fever and rash. Explanation: typhoid fever is foodborne infection caused by Salmonella typhi with systemic symptoms including fever, abdominal pain, and rash. Proper treatment is required to prevent severe complications. Next, last question, 10th one. Botulism. Statements: botulism is caused by a neurotoxin producing strains of Clostridium botulinum. The toxin affects the nervous system, leading to paralysis and respiratory failure. Botulism is often caused by improperly canned foods, especially low-acid vegetables. The neurotoxin produced by Clostridium botulinum is one of the most potent toxins known to humans. Conclusion: which of the following statements is most accurate regarding botulism? Option A. Botulism is caused by a viral infection and is primarily transmitted via respiratory droplets. B. Clostridium botulinum produces potent neurotoxin that causes paralysis, typically through contaminated canned foods. C. Botulism is a mild disease and often resolves without medical intervention. D. Botulism only affects infants and is not a concern for adults. Here answer is option B. Clostridium botulinum produces a potent neurotoxin that causes paralysis, typically through contaminated canned foods. Explanation: botulism is caused by the neurotoxin produced by Clostridium botulinum primarily through contaminated food, often from improperly canned or preserved food products. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. First question: the FAO's Global Soil Partnership Initiative launched in 2012 aims to address food loss and waste reduction. B. Promote sustainable soil management globally. C. Enhance biodiversity conservation. D. Strengthen aquaculture system. Which is the answer? Your answer is option B. Promote sustainable soil management globally. Second question: the Hand in Hand initiative launched in 2019 primarily focuses on strengthening international trade policies, eradicating poverty and hunger in most vulnerable regions. C. Promoting genetically modified crop research. D. Ensuring food quality standards globally. Which is the answer? Your answer is option B. Eradicating poverty and hunger in the most vulnerable regions. Third question: in which year was the Save Food initiative aimed at reducing global food loss and waste launched? 2011, 2013, 2015, 2017. Which is the answer? Your answer is 2011. Fourth question: what is the primary focus of the FAO's Blue Growth Initiative? A. Conservation of water resources. B. Sustainable management of aquatic food systems. Option C. Encouraging renewable energy in agriculture. Option D. Promoting urban farming practices. Which is the answer? Your answer is option B. Sustainable management of aquatic food systems. Fifth question: globally important agriculture heritage system GIAHS established 2002 primarily aims to enhance urban agriculture. B. Protect traditional farming systems with cultural heritage. C. Introduce digital tools for smart farming. D. Regulate international food trade. Which is the answer? Your answer is option B. Protect traditional farming systems with cultural heritage. Sixth question: in the FAO's Youth and United Nations Global Alliance Young initiative, what is the purpose of challenge badges? Certifying completion of farming modules, recognizing achievements in environmental stewardship. C. Granting funding for agribusiness startups. D. Awarding agricultural innovations. Which is the answer? Here answer is B. Recognizing achievements in environmental stewardship. The purpose of the United Nations Challenge Badges is to raise awareness, educate, and most of all motivate young people to change their behavior and be active agents of change in their local communities. Seventh question: the FAO Fisheries Program aligned with the Code of Conduct for Responsible Fisheries 1995 emphasizes A. Genetic engineering in aquaculture. Option B, reducing illegal, unreported and unregulated fishing. C, supporting industrial scale fishing. D, promoting fish exports from developing nations. Which is the answer? Your answer is reducing illegal, unreported and unregulated fishing. Option B. Eighth question. Which the thematic priority is addressed by the FAO's Urban Food Agenda initiative initiated in 2019? Strengthening rural markets, enhancing food security in urban centers, C. Subsidizing agricultural exports, D. Promoting genetic crop diversification. Which is the answer? Your answer is option B. Enhancing food security in urban centers. Ninth question. The FAO's 2021 For Better's strategic vision focuses on better nutrition, better production, better environment, and dash. Fill with correct option. A. Better collaboration, better life, better economics, better development. Which is the answer? Your answer is option B. Better life. 10th question. Which Food and Agriculture Organization initiative is associated with the theme Our Actions Are Our Future? Better production, better nutrition, a better environment, and a better life. World Food Day 2021. International Year of Farming, Family Farming. Food Safety Guidelines 2019. Global Food Summit 2022. Which is the answer? Your answer is option A. World Food Day 2021. 11th question. What is the primary objective of Agricultural Market Information System AMIS initiated by FAO in 2011? Monitoring agricultural subsidies, to enhance food market transparency and policy response for food security, promoting private sector participation in agriculture, enhancing organic farming networks. Which is the answer? Your answer is option B, to enhance food market transparency and policy response for food security. 12th question. FAO declared 2023 as the International Year of Millets to address gluten intolerance in diets, to promote sustainable cultivation of drought-resistant crops, enhance soil health globally, D. Develop fortified cereal varieties. Which is the answer? Your answer is option B. Promote sustainable cultivation of drought-resistant crops. IYM 2023 will be an opportunity to raise awareness of and direct policy attention to the nutritional and health benefits of millets and their suitability for cultivation under adverse and challenging or changing climatic conditions. 13. The FAO SFERA Fund was established in 2004 to manage emergency responses for food crisis. B. Support rural women and entrepreneurs. C. Enhance agricultural tourism networks. D. Fund bioengineering research farming. G is the answer. Your answer is option A. Manage emergency responses for food crisis. 14th question. In which year was the International Treaty on Plant Genetic Resources for Food and Agriculture adopted by Food and Agricultural Organization? 2001, 2003, 2005, 2007. Which is the answer? Your answer is option A. 2001. The International Treaty on Plant Genetic Resources for Food and Agriculture is legally binding instrument adopted by the FAO conference in 2001 and it entered into force on 29th June 2004. 15th question. FAO's Global Action on Fall Armyworm Control launched in 2019 aims to create genetically modified pest-resistant crops, provide sustainable solutions to combat fall armyworm outbreaks, establish pesticide-free farming zones, D. Monitor soil degradation rates. Which is the answer? Your answer is option D. Provide sustainable solutions to combat fall armyworm outbreaks. In December 2019, FAO launched a 3-year global action for fall armyworm control and recognized the importance of prevention and preparedness to better respond to FAW rapid spread. 16th question. Which FAO initiative focuses on transforming agri-food system to reduce hunger by 2013? Zero Hunger Sustainable Development Goal 2030, Food Security Vision 2050, Agri-Sustainable Plan 2030, Global Nutrition Framework. Which is the answer? Your answer is option A. Zero Hunger SDG 2030. 17. The FAO's Tephritide Workers Database supports research on fruit fly pest management, wheat rust surveillance, genetically improved cattle breeds, algal bloom control in aquaculture. Which is the answer? Your answer is option A. Fruit fly pest management 18th question, the FAO emergency prevention system includes components for livestock, crop and aquatic animal diseases, climate adaptation in farming practices, enhanced digital mapping for land usage, D. Agro-tourism partnerships. Which is the answer? Here answer is option A, livestock, crops and aquatic animal diseases. EMPRS has two components created after 1994 by a new policy of the Director General of FAO to better direct the FAO. The plant pest component focuses on the desert locust, whereas the animal diseases component focuses primarily in rinderpest, but also on other epidemic diseases too. 19th question. What was the central theme of World Food Day 2020 coordinated by FAO? Climate is changing, food and agriculture must too. Grow, nourish, sustain, together our actions are our future. Our actions are our future. Healthy diets for a zero hunger world. Which is the answer? Here answer is option B. Grow, nourish, sustain, together our actions are our future. 20th question. What is the primary focus of the FAO Locust Watch program initiated in 1978? Tracking desert locust outbreak and providing early warnings, conducting genetic research on locust control, promoting pesticide-free pest control techniques, studying the life cycle of grasshoppers. Which is the answer? Your answer is option A, tracking desert locust outbreaks and providing early warnings. 21. Which FAO initiative launched in 2016 addresses antimicrobial resistance in agriculture? A. The FAO Action Plan on Antimicrobial Resistance. Option B. Global Health Strategy for Animals. Option C. Veterinary Sustainability Program. Option D. Pathogen Reduction Initiative. Which is the answer? Your answer is option A. The FAO Action Plan on Antimicrobial Resistance. The FAO Action Plan on Antimicrobial Resistance 2016 to 2020. Increasing global antimicrobial resistance is a major threat to human and animal health. It endangers modern human and veterinary medicine and undermines the safety of our food and environment. 22. Who is the first Director General of FAO? Sir John Boyd or Antonio Guterres, Tedros Adhanom Ghebreyesus, Okonjo Iweala? Which is the answer? Your answer is option A, Sir John Boyd or. 23. What innovation is central to the FAO's e-agriculture strategy guide released in 2016? Satellite-based weather monitoring for crop planning. B. Promoting digital tools to empower agricultural stakeholders. C. Blockchain technology for global food trade. D. Artificial intelligence-driven genetic crop modification. Which is the answer? Your answer is option B. Promoting digital tools to empower agricultural stakeholders. 24. FAO's One Country, One Quality product initiated in 2021 aims to focus national efforts on diversifying agricultural production, identify and promote specific high-value agricultural products, eliminate imports of staple crops, advocate for a uniform global agriculture trade policy. Which is the answer? Your answer is option B. Identify and promote specific high-value agricultural products. The OCOP was developed as a country-led and country-driven initiative that focuses on special agricultural products which have unique qualities and special characteristics that are associated with geographical locations, farming practices, and cultural heritages or untapped potential. 25. FAO launched on 16th October 1945. October 16, 1945 in Quebec City, Canada. 26. The Green Cities Initiative introduced by FAO in 2020 aimed to improving the livelihoods and well-being of urban populations, improving the livelihood and well-being of peri-urban populations, improve urban and peri-urban populations at least 100 cities around the world in the next 3 years, looking to have 1,000 cities joined by 2030. Which is the answer? Your answer is option D, all of the above. 27. The FAO Global Action on Pollination initiated in 2021 addresses conservation and sustainable use of pollinators for agriculture, introduction of robotic pollinators in agriculture, expansion of monoculture farming practices, genetic modification of pollinator species. Which is the answer? Your answer is option A, conservation and sustainable use of pollinators for agriculture. 28. In 2021, FAO launched the Rec Soil initiative focusing on soil carbon sequestration to combat climate change, introducing hydroponic farming to arid regions, C. Large-scale fertilizer subsidy programs, D. Commercializing degraded soil for urban expansion, which is the answer. Your answer is option A. Soil carbon sequestration to combat climate change. Rugged soil creates an enabling environment at a national level and empowers individual farmers to adopt sustainable practices that increase soil health, resilience to climate change, food security, and livelihoods. 29. Who is the Director General of FAO? Q. Donkey, Antonio Guterres, Tedros Adhanom Ghebreyesus, Okonjo Iweala. Which is the answer? Your answer is option A. Q. Donkey. Food Safety Officer book is available with 1000 MCQs from all sections. You can order it through Amazon link. It is available in the description. Last question number of this session, 30. How many members are there in FAO? 195, 196, 194, 193. Which is the answer? Your answer is option A, 195. The FAO comprises 195 members, including 194 countries and the European Union. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Important MCQs of microbiology. First question, which marine biotoxin causes amnesic shellfish poisoning, ASP? Saxitoxin, brevitoxin, domoic acid, ciguatoxin. Your answer is option C, domoic acid. Domoic acid is a marine biotoxin called amnesic shellfish poisoning, which is produced by the diatom Pseudonychia species, a type of naturally occurring microscopic algae. Shellfish eat this algae and can retain the toxin. People can become ill from eating shellfish contaminated with amnesic shellfish poison. Second question. Which foodborne illness is caused by prions rather than bacteria, viruses, or parasites? Brucellosis, Creutzfeldt-Jakob disease, CJD, listeriosis, hepatitis E. Here answer is option B, Creutzfeldt-Jakob disease, CJD. CJD is a rare rapid worsening brain disorder that causes unique changes in brain tissue and affects muscle coordination, thinking, and memory. There are about 350 cases per year in US. The two main symptoms of CJD are severe mental deterioration and dementia. Third question. What is a novel strategy for the control of Clostridium difficile infections in the food industry, particularly in the context of antibiotic resistance? Development of bacteriophage therapy, probiotic supplementation to reduce pathogen load, use of phage display technology for rapid diagnosis, incorporation of antibiotic-free practices in food production. Your answer is option A, development of bacteriophage therapy. Fourth question, which foodborne pathogen has recently been implicated in causing infections through contaminated frozen vegetables, and its presence is linked to an unusual resistance to freezing temperatures? Salmonella enterica, Listeria monocytogenes, Clostridium botulinum, E. coli O15787. Your answer is option B, Listeria monocytogenes. Fifth question, which of the following novel therapeutic approaches is being investigated to combat Salmonella enterica infections in food safety? Development of bacteriocins as selective antimicrobial agents, CRISPR-Cas9 gene editing to block virulence factors, immunization strategies using synthetic bacterial antigens, D, all of the above. Your answer is all of the above. Sixth question, which antimicrobial resistant mechanism is commonly associated with Campylobacter jejuni in poultry, often linked to fluoroquinolone resistance? A, methylation of ribosomal RNA, B, efflux pump overexpression, C, alteration of DNA gyrase, D, reduced outer membrane permeability. Your answer is option C, alteration of DNA gyrase. Alteration of DNA gyrase The primary mechanism of resistance to fluoroquinolones are mutations that result in alteration of the target proteins, DNA gyrase encoded by GYR-B gene and topoisomerase encoded by part C, part E gene, and decrease intracellular drug accumulation due to drug efflux or changes in outer membrane proteins. Seventh question, the occurrence of Cryptosporidium species in foodborne outbreaks has been associated with which of the following food contamination routes? Your answer option is A. Option A. Raw vegetables contaminated with agricultural runoff. B. Cross contamination through undercooked poultry. C. Contaminated milk and dairy products. D. Seafood contaminated during harvesting. Your answer is option A. Raw vegetables contaminated with agricultural runoff. Contamination of vegetables, fresh fruits, juices, unpasteurized raw milk, uncooked meat, and fish by Cryptosporidium oocysts occurred through infected food handlers, sewage-based contamination, agricultural effluents, infected animal manure being used as biofertilizers, etc. Eighth question. What is the mechanism of action of Bacillus cereus emetic toxin? It disrupts cell membranes. It binds to serotonin receptors in the gut. It inhibits protein synthesis in cells. D. It increases chloride ion secretion in the intestines. Your answer is option B. It binds to serotonin receptors in the gut. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers or directly order through Amazon link which is available in the description. Ninth question. Which of the following pathogens is commonly associated with raw or undercooked seafood and can cause septicemia in immunocompromised individuals? Vibrio parahemolyticus, Vibrio vulnificus, E. coli O15787, Salmonella enterica. Your answer is option B, Vibrio vulnificus. Individuals at increased risk of developing severe Vibrio vulnificus infection include those with liver disease such as cirrhosis, cancer, HIV infection, diabetes, or a certain blood disorder, and those who take immunosuppressants or medications to decrease stomach acid levels. 10th question. Which bacillus species is capable of producing both enterotoxins and neurotoxins, making it a significant concern in foodborne outbreaks? A. Bacillus anthracis. B. Bacillus thuringiensis. C. Bacillus cereus. D. Bacillus subtilis. Your answer is option C. Bacillus cereus. The emetic toxin directly acts on the afferent vagus nerve and vomiting center in the central nervous system. 11th question. Which streptococcus species recently associated with dairy products produces a heat stable enterotoxin and can cause foodborne illness resembling staphylococcal food poisoning? Streptococcus pyogenes, Streptococcus thermophilus, Streptococcus dysgalactiae, Streptococcus agalactiae. Your answer is option D, Streptococcus agalactiae. Streptococcus agalactiae is an obligate pathogen of the bovine mammary gland and is spread in a contagious manner during milking. 12th question. Which of the following foodborne viruses is notable for its persistence on surfaces and in foods for extended periods even under refrigeration, making it a major concern in food safety? Hepatitis A virus, norovirus, rotavirus, human enteric adenovirus. Your answer is option B, norovirus. Norovirus is a very contagious virus that causes vomiting and diarrhea. It is sometimes called the stomach flu or the stomach bug. However, norovirus illness is not related to the flu. The flu is caused by the influenza virus. Norovirus causes acute gastroenteritis and inflammation of the stomach or intestines. This reference from CDC. 13. Which biopolymer produced by Bacillus cereus plays a key role in its ability to form biofilms, thus contributing to its persistence in food processing environments? PHB, PGA, PIA, UPS. Your answer is B, polyglutamic acid. Polyglutamic acid, a polyamino acid produced by Bacillus species, is a naturally occurring anionic, water-soluble, biodegradable, non-toxic, viscous, edible biopolymer containing D-N-L-glutamic acid residues with different industrial applications. 14th question. Which bacterial pathogen is commonly linked to brined or pickled food due to its halotolerance? A. Vibrio parahemolyticus, B. Staphylococcus aureus, C. Listeria monocytogenes, D. Bacillus cereus. Here answer is option B. Staphylococcus aureus. Gram-positive Staphylococcus aureus is one of the most halotolerant eubacteria. A previous study reported that Staphylococcus aureus can grow in sodium chloride concentration up to 3.5 molar. Reference Vijayankul et al. 1995. 15th question. Which foodborne pathogen can mimic appendicitis and is commonly linked to contaminated milk or pork? Yersinia enterocolitica, Campylobacter jejuni, Salmonella enterica, Listeria monocytogenes. Your answer is option A. Yersinia enterocolitica. Reference is mentioned below. 16th question. Which type of foodborne illness results from consuming preformed toxins rather than bacterial colonization in the gut? Intoxication, infection, toxin-mediated infected zoonosis. Your answer is option A. Intoxication. 17th question. Which bacterial toxin is heat stable and poses a risk even after cooking contaminated food? Staphylococcus enterotoxin, botulinum toxin, cholera toxin, diphtheria toxin. Your answer is Staphylococcus enterotoxin. 18th question. Which analytical technique is most effective for detecting norovirus in contaminated food samples? Enzyme-linked immunosorbent assay, RT-PCR, mass spectrometry, flow cytometry. Your answer is option B, qPCR. 19th question. What is the primary mechanism by which E. coli O157H7 causes hemolytic uremic syndrome? Endotoxin production, Shiga toxin mediated endothelial damage, invasion of intestinal epithelial cells, biofilm formation in the gastrointestinal tract. Here option B, Shiga toxin mediated endothelial damage. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers or you can directly purchase through Amazon link which is available in the description. 20. Question number 20. The consumption of raw or undercooked freshwater fish can lead to infections with which parasite? Clonorhynchus sinensis, Toxoplasma gondii, Trichinella spiralis, Anisakis simplex, Clonorhynchus sinensis. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. The largest food park in India is located at Godavari Mega Aqua Park, Srini Mega Food Park, Chittoor, Andhra Pradesh, West Godavari, Andhra Pradesh, Northeast Mega Food Park, Nalbari, Assam. Your answer is option B, Srini Mega Food Park, Chittoor, Andhra Pradesh. Second question. Which Indian state introduced Anna Bhagya scheme for free rice distribution? Andhra Pradesh, Karnataka, Bihar, Odisha. Your answer is option B. Third question. The blue revolution in India is associated with aquaculture, dairy farming, water conservation, poultry farming. Your answer is option A. Fourth question. Which river basin is known as the rice bowl of India? Godavari, Krishna, Krishna Godavari Delta region, Ganga. Here answer is option C, Krishna Godavari Delta region. Fifth question. The Kisan Rail service launched by the Indian Railway focuses on transporting machinery, facilitate the transportation of perishable goods including fish, meat and milk across the country, sand, fertilizers. Here answer is option B, facilitate the transportation of perishable goods including fish, meat and milk across the country. Sixth question. Which Indian state topped the food safety index in 2023-24? Tamil Nadu, Gujarat, Karnataka, Kerala. Your answer is option D. Seventh question. Largest tea producing state in India? Kerala, Assam, Andhra Pradesh, Sikkim. Your answer is B. Assam. Eighth question. Operations Greens aims to stabilize the prices of which commodities? Rice, wheat and pulses, tomato, onion and potato, sugarcane and cotton, milk and eggs. Here answer is option B, tomato, onion and potato. The primary objectives are to stabilize the supply and prices of tomato, onion and potato crops by promoting efficient agro-logistics processing facilities and professional management. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Link to purchase the book is available in the description. Ninth question. In 2019, which Indian city was named the UNESCO Creative City of Gastronomy? Jaipur, Mumbai, Hyderabad, Varanasi. Your answer is option C. 10th question. The PM Portion Scheme focuses on providing meals to which group? Farmers, school children, pregnant women, senior citizens. Your answer is option B, schools, children. The PM Portion Scheme is being implemented by the Ministry of Education across all government and government-aided schools in India. It provides nutritional meal to each and every child of Balvatika, pre-primary or before class first, and the children studying in class 1 to 8. 11th question. Operation Flood was launched by which Indian organization? FSSAI, NABARD, NDDB, ICAR. Your answer is option C, NDDB. 12th question. The Sampada scheme is associated with food processing and agro industries, crop insurance, fertilizer subsidy, dairy development. Here option A, Sampada scheme is being implemented by Ministry of Food Processing Industries. It aims to provide a renewed thrust to the food processing sectors and supply chain management. 13th question. Which Indian state is the leading producer of coffee? Tamil Nadu, Karnataka, Kerala, Andhra Pradesh. Your answer is Karnataka. 14th question. India recently developed fortified wheat flour to combat vitamin A deficiency, iron deficiency, anemia, protein deficiency, iodine deficiency. Your answer is option iron deficiency, anemia. 15th question. Which Indian state launched the Nutri Garden initiative? Madhya Pradesh, Rajasthan, Odisha, Telangana. Your answer is B, Rajasthan. The Nutri Garden initiative is a program implemented in Rajasthan, India to address the persistent issue of malnutrition and to improve the nutritional status of population, particularly among vulnerable groups. 16th question. The Mission Organic Value Chain Development is focused on which region? Northern India, Western India, Northeast India, Southern India. Your answer is option C, Northeast India. Considering the special observation on the existing fertile soil and favorable climatic condition in northeastern region, the Ministry of Agriculture and Farmers Welfare Government of India has launched a central sector scheme named as Mission Organic Value Chain Development for Northeastern region. 17th question. The Golden Revolution in India is related to horticulture, sericulture, honey production, both A and C. It is related to horticulture and honey production. So here answer is option D, both A and C. Golden Revolution is related to the production of honey and horticulture. It is a part of the important agricultural revolutions of India. 18th question. Largest food park in India is located at Godavari Mega Aqua Park, Srini Mega Food Park, Chittoor, Andhra Pradesh, West Godavari, Andhra Pradesh, Northeast Mega Food Park, Nalbari, Assam. Your answer is Srini Mega Food Park, Chittoor, Andhra Pradesh. 19th question. Which day is marked as National Farmers Day, Kisan Divas in India? January 14th, December 23rd, March 12th, August 15th. Here option B, December 23rd. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Link to purchase the book is available in the description. Last question. The World Egg Day is celebrated on the second Friday of which month? March, April, October, November. Your answer is option C, October. The theme for the 2024 World Egg Day is United by Eggs. That theme was selected as eggs can be found in cuisines across the world's cultures and countries, a testament to their universal appeal and nutritional benefits. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Important MCQs of Food Technology. First question, where is the headquarters of the Food Safety and Standards Authority of India is located? A. Mumbai, B. New Delhi, C. Bangalore, D. Chennai. Your answer is option B, Delhi. Second question, the Indian Institute of Food Processing Technology is located in which state? Karnataka, Tamil Nadu, Maharashtra, Gujarat. Your answer is option B, Tamil Nadu. National Institute of Food Technology, Entrepreneurship and Management, Tanjavur, formerly Indian Institute of Food Processing Technology. Now it is known as National Institute of Food Technology, Entrepreneurship and Management. Third question, the Central Food Technological Research Institute is located in which Indian city? A. Bangalore, B. Hyderabad, C. Mysore, D. Coimbatore. Your answer is option C, Mysore. Fourth question, which of the following is a flagship institute under the Ministry of Food Processing Industries? NIFTEM, ICAR, FSSAI, CFTRI. Your answer is A. NIFTEM. It is situated in Kundli, Haryana and Tanjavur, Tamil Nadu. Fifth question, the National Institute of Nutrition is based in which city? New Delhi, Hyderabad, Kolkata, Pune. Your answer is option B, Hyderabad. 6. Which institute collaborates extensively with industries for food technology innovations in India? Central Food Technological Research Institute, ICR Institute, IIFPT, MOFPI. Ministry of Food Processing Industries. Your answer is option A, CFTRI. Seventh question, which organization provides funding and support to food-related startups in India? FSSAI, MOFPI, Niti Aayog, NIFTEM. Your answer is option B, Ministry of Food Processing Industries. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Eighth question. The food testing laboratory at CFTRI is accredited by which international standard? ISO 9001, ISO 17025, HACCP, GMP. Your answer is option B, ISO 17025. Ninth question, which Indian institute is primarily involved in research on micronutrient deficiencies in the diet? NIFTEM, IIFPT, NIN, CFTRI. Your answer is option C, National Institute of Nutrition. 10th question, which institute is tasked with developing advanced packaging solutions for food products in India? NIFTEM, Indian Institute of Packaging, CFTRI, IIFPT. Your answer is option B, Indian Institute of Packaging. The institute is headquartered at Mumbai and its regional offices are located at Chennai, 1971, Kolkata, Delhi, Hyderabad and Ahmedabad. 11th question, which organization in India plays a key role in promoting food fortification initiatives? CFTRI, FSSAI, MOFPI, IIP. Your answer is option B, FSSAI. 12th question, which food related institute in India is credited with the development of nutritious Mysore snacks? CFTRI, NIFTEM, IIPT, NIN. Your answer is A, CFTRI. 13th question, the Food and Agricultural Organization collaborates with which Indian Institute for promoting sustainable agriculture and food safety? CFTRI, IIFPT, NIFTEM, ICAR. Your answer is option D, ICAR. 14th question, which Indian institute is actively engaged in research on reducing food wastage during storage and transportation? CFTRI, ICAR, CIPET, NIFTEM, NIN. Here answer is option B, ICAR, CI, CIPET. ICAR, CIPET, full form Central Institute of Posh Harvesting Engineering and Technology. 15th question, which organization is responsible for accrediting food laboratories in India? FSSAI, NABL, CFTRI, MOFPI. Here answer is B, National Accreditation Board for Testing and Calibration Laboratories. 16th question, which Indian institute pioneered the development of ready-to-eat military rations? A, CFTRI, DRDO, Defense Food Research Laboratory, IIFPT, NIFTEM. Your answer is option B, DRDO. Defense Food Research Laboratory. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link to purchase the same is available in the description. If you like this channel, please share and subscribe. 17th question. Which Indian institute is primarily responsible for promoting and advancing dairy research and education? National Dairy Research Institute, CFTRI, IAFET, NIFTM. Here answer is option A. National Dairy Research Institute. NDRI in India is premier institute for dairy research established in 1955, located in Karnal, Haryana, having been accorded with the status of deemed university since 1989. 18th question. Which institute is a global leader in developing value-added products from millets? Indian Institute of Millets Research, NIFTEM, IAFPT, CFTRI. Here answer is option A. Indian Institute of Millets Research. 19th question. Which Indian institute developed the Enhanced Nutribund program? A. The Department of Science and Technology's Food and Nutrition Research Institute, DOST FNRI. B. NIN. C. NIFTEM. D. IAFPT. Here answer is option A. The Department of Science and Technology's Food and Nutrition Research Institute. 20. Question number 20. The International Crop Research Institute for Semi-Arid Tropics is situated at Bangalore, Cochin, Hyderabad, Chennai. Here answer is option C. Hyderabad. 21. The Institute of Food Security under the Food Corporation of India is located in which city? Gurugram, Delhi, Hyderabad, Mumbai. Here answer is option A. Gurugram. 22. Question number 22. Which institute developed the double fortified salt with iron and iodine? NIN, CFTRI, FSSAI, IAFPT. Here answer is option A. NIN. 23. The question number 23. The ICAR Directorate of Groundnut Research is located in which state? Gujarat, Tamil Nadu, Andhra Pradesh, Maharashtra. Here answer is option A. Gujarat. 24. Which institute developed the super bar for astronaut food in India? DRDO, DFRL, CFTRI, IFPL, ICAR. Your answer is option A. DRDO, DFRL. 25. Which institute in India developed food product named Nutra Chicky with added spirulina to combat malnutrition? DFRL, DRDO, CFTRI, NIN. Your answer is option C. CFTRI, Mysore. 26. Which Indian institute developed an antifreeze container for transporting fruits, veggies, and at high altitude terrains? A. DFRL, B. DRDO, C. CFTRI, D. National Institute of Nutrition. Here answer is option A. DFRL. DFRL, Mysore. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. You can directly purchase it from Amazon link which is available in the description. If you like this channel, please share and subscribe. 27. Which Indian institute developed biodegradable water bottles? DFRL, DRDO, CFTRI, NIN. Your answer is option A. DFRL, Mysuru. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Food related full forms. First question. What does FSSAI stands for? A. Food Safety and Sanitation Authority of India. Food Standards and Safety Association of India, Food Safety and Standards Authority of India, Food Safety and Security Agency of India. Which is the answer? Here answer is option C. Food Safety and Standards Authority of India. Second question. What is the meaning of FOSTAC in FSSAI regulations? A. Food Safety Training and Certification. B. Food Standards Testing and Certification. C. Food Safety Testing and Analysis Council. D. Food Sustainability Testing and Certification. Which is the answer? Here answer is option A. Food Safety Training and Certification. Third question. FLRS is an essential tool under FSSAI. What does FLRS stands for? A. Food Licensing and Registration System. B. Food Labeling and Registration Standards. C. Food Laboratory Regulatory Standards. D. Food Law and Regulation Service. Here answer is option A. Food Licensing and Registration System. Fourth question. In the context of FSSAI, what does NABL signify? National Accreditation Board for Laboratories. B. National Analytical and Biochemical Laboratory. C. National Accreditation Board for Testing and Calibration Laboratories. D. National Agency for Biochemical Labs. Which is the answer? Here answer is option C. National Accreditation Board for Testing and Calibration Laboratories. Fifth question. What does FBO stand for in FSSAI guidelines? What does FBO stand for in FSSAI guidelines? A. Food business operator. B. Food brand oversight. C. Food biosecurity organization. D. Food based operations. Which is the answer? Here answer is option A. Food business operator. Sixth question. RDA on food labels regulated by FSSAI refers to A. Recommended dietary allowance. B. Required daily amount. C. Recognized dietary application. D. Registered daily average. Here answer is A. Recommended dietary allowance. Seventh question. What does TDS stands for in food quality checks under FSSAI? What does TDS stand for in food quality checks under FSSAI? A. Total dissolved solids, total dietary standards, testing dietary substances. D. Traceable dissolution standards. Here answer is total dissolved solids. A. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. You can directly purchase from the Amazon through Amazon link. Eighth question. What is the meaning of INS in the context of food additives? A. International Numbering System. B. Ingredient Notification System. C. Ingredient Nutrition Standards. D. International Nutrient Standardization. Which is the answer? Here answer is option A. International Numbering System. Ninth question. What does GDA signify on food packages? A. Guideline daily amount. B. General dietary allowance. C. Grams of daily average. D. Global diet assessment. Here answer is option A. Guideline daily amount. Guideline daily amounts are a labeling guide to help you make informed choices about the food you eat. It gives an overview of the percentage of energy and key nutrients that are in a product per serving, specifically those nutrients that may increase risk of diet-related illness. 10th question. Which term does GMF stand for under FSSAI labeling guidelines? Genetically modified foods, good manufacturing foods, general monitoring framework, graded manufactured foods. Your answer is A. Genetically modified foods. What does HACCP stand for in food safety management? A. Hazard analysis and critical control points. B. Hygiene assurance and control plan. C. Health and cleanliness certification program. D. Hazard assessment and corrective plan. Your answer is option A. Hazard analysis and critical control points. 12th question. FSSAI frequently refers to GMP in food processing units. What does GMP signify? A. Good manufacturing practices. B. General monitoring practices, procedures. C. Graded manufacturing processes. D. Government managed policies. Your answer is option A. Good manufacturing practices. 13th question. What does ISO stand for in FSSAI certified food laboratories? A. International Organization for Standardization. B. Indian Standard Organization. C. International Standards for Operations. D. Industry Standards Objective. Your answer is option A. International Organization for Standardization. 14th question. Which term does SOP represent in food safety inspections? Standard operating procedure, safety oversight program, sanitation operating practices, systematic oversight protocol. Your SOP stands for standard operating procedure. What is the meaning of FSMS in FSSAI guidelines? Food Safety Management System, Food Standards and Monitoring System, Food Safety Monitoring Standards, Food Security and Management Standards. Your answer is option A. Food Safety Management System. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. 16th question. What does FCT stand for in nutrition research? Food composition tables, food chemical testing, food chain tracking, food consumption trends. Your answer is option A. Food composition tables. 17th question. In nutritional labeling, what does NRV signify? Nutrient reference value, nutrition and regulatory values, nutrition recommended value, nutritional risk value. Here answer is option A. Nutrient reference value. 18th question. What does RUTF stand for in malnutrition treatment programs? Reduced use therapeutic food, regulated utility for therapeutic foods, rapid uptake of therapeutic food, recommended use of treatment food. Your answer is option A, ready-to-use therapeutic food. Ready-to-use therapeutic food, RUTF, is the abbreviation for ready-to-use therapeutic food, a life-saving essential supply item that treats severe wasting in children under 5-year-old. Wasting is defined as a low weight for height and happens when someone has not had enough food or food of adequate quality. 19th question. What does ICMR stand for in food research collaboration with FSSAI? Indian Council for Medical Research, International Center for Microbial Research, Indian Council for Microbiome Research, Integrated Council for Medical Regulations. Your answer is option A, Indian Council for Medical Research. 20. Question number 20. Which of the following is the correct abbreviation for CIF in food import regulations? Cost insurance and freight, centralized import framework, certification of important food, compliance in food imports. Here answer is option A, cost insurance and freight. 21. What does GHP signify in FSSAI hygienic practices? Good hygiene practices, general health protocols, government hygiene programs, graded hygiene procedures. Your answer is A, good hygiene practices. 22. What does NRL stand for FSSAI's lab network? National Reference Laboratory, Nutritional Research Lab, National Regulation of Laboratories, Nutrient Reference Levels. Your answer is option A, National Reference Laboratory. 23. What does FICS stand for in food certification schemes? A. Food Import Certification Scheme, B. Food Industry Certification Scheme, C. Food Inspection and Certification Scheme, D. Food Information Compliance Standards. Your answer is option B, Food Industry Certification Scheme. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. You can directly purchase from Amazon link. 24. What does InfoSan mean in the context of international food safety? A. International Food Safety Authorities Network, B. Integrated Food Safety Analysis Network, C. Indian Food Safety Analysis Network, D. International Nutrition and Food Safety Network. Your answer is option A, International Food Safety Authorities Network. 25. Which term does GAP represent in sustainable agriculture? Good agricultural practices, general agronomic protocol, government agriculture policy, graded agriculture procedure. Your answer is option A, good agricultural practices. 26. What does FLINT stand for in food traceability system? Food labeling and inspection technology, food linked inventory and traceability, food lab inspection and networking techniques, food logistics and international tracking. Your answer is option B, food linked inventory and traceability. 27. What does FFRC stand for in FSSAI's Fortification Initiative? Food Fortification Resource Center, Fortified Foods Regulatory Commission, Food and Fortification Review Council, Fortified Foods Research Center. Your answer is option A, Food Fortification Resource Center. The Food Fortification Resource Center is established under India's government department that regulates food, known as the FSSAI, in collaboration with Data Trust. 28. What does InfoNet refers to in FSSAI's laboratory system? Indian Food Laboratories Network, Indian Food Laboratories for Nation, Indian Food Additives for Laboratories, International Food Laboratories for Nation. Your answer is A, Indian Food Laboratories Network. 29. What does MAP stand for in food packaging technologies? Modified atmosphere packaging, maximum allowable packaging, molecular adjustment process, D. Multi-application packaging. Your answer is option A, modified atmosphere packaging. 30. What is the meaning of HPP in food preservation methods? High pressure processing, hazard prevention procedure, hydrostatic pasteurization process, high precision packaging. Your answer is option A, high pressure processing. 31. What does TTI stand for in food safety monitoring? Time temperature indicator, traceable testing index, temperature tracking and inspection, thermal trace indicator. Your answer is A, time temperature indicator. Question number 32. What does TPC mean in food microbiological testing? A, total plate count, B, traceable pathogen check, C, thermal process certification, D, target pathogen count. Here answer is option A, total plate count. 33. What does RASFF represent in the Global Food Safety Network? A. Rapid Alert System for Food and Feed, B. Regulatory alert for safety of foods. C. Risk assessment system for food. D. Real-time analysis of safety in foods. Your answer is option A. Rapid alert system for food and feed. As part of the food safety tools, the rapid alert system for food and feed was established to ensure the exchange of information between member countries to support swift reaction by food safety authorities in case of risk to public health resulting from the food chain. Implemented by EU. 34th question. What does PDCAAS stand for in food protein evaluation? A. Protein digestibility corrected amino acid score. Processed dietary compliance and amino acid safety. Protein density compliance assessment system. D. Provisional digestibility and composition analysis score. Your answer is option A. Protein digestibility corrected amino acid score. Food Safety Officer book is available with 1000 MCQs from all sections. Contact to these numbers. Amazon link is available in the description. Last question 35. FSSAI has formed IFSA to help solve India's food waste and hunger crisis by integrating various partner organizations, food recovery agencies and NGOs. What is the full form of IFSA? A. Indian Food Sharing Alliance. B. Indian Food Sharing Association. C. Indian Street Food Alliance. D. Indian Food Sharing Alliance. Here answer is option A. Indian Food Sharing Alliance. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Veterinary Sciences Facts and MCQs. Which natural preservative is derived from lactic acid bacteria and used in meat products? Sodium benzoate, nisin, sodium nitrite, potassium sorbate. Which is the answer? Here answer is option B. Nisin. Second one, which chemical is often used illegally to enhance the freshness appearance of fish? Sodium metabisulfite, borax, formalin, potassium permanganate. This is the answer. Your answer is option C. Formalin. Third question, which preservation method is specifically designed to minimize lipid oxidation in processed meats? Smoking, curing, antioxidant application, fermentation. Your answer is option C. Antioxidant application. Fourth question, which antimicrobial agent is often used to inhibit microbial growth in chilled fish? Potassium permanganate, sodium acetate, sodium metabisulfite, citric acid. Which is the answer? Here answer is option C. Sodium metabisulfite. Fifth question, which method is used to preserve the quality of fish during transportation over long distances? Use of dry ice, vacuum packaging, icing, active packaging with oxygen scavengers. Your answer is option D. Active packaging with oxygen scavengers. Oxygen scavengers are a type of active packaging technique which absorb dissolved oxygen or oxygen in the headspace, leading to extended shelf life and retainment of the original food quality. Sixth question, what is the key role of polyphenols in the preservation of seafood? A. Enhances flavor. B. Prevents enzymatic browning. C. Retains moisture in muscle tissue. D. Acts as an antimicrobial agent. Your most appropriate answer is option C. Retains moisture in muscle tissue. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. You can purchase through the Amazon link too. Seventh question, which antimicrobial peptide is naturally present in fish slime and is being explored for preservation applications? Piscins, defensins, meganins, cathalycidins. Which is the answer? Here answer is option A. Piscidins. Piscidins are a family of cationic AMPs that are produced by fish and have broad spectrum antimicrobial activity against bacteria, fungi and viruses. Eighth question, what is a common chemical marker for identifying horse meat substitution? Collagen, myoglobin, creatinine, carnosine. Here most appropriate answer is D. Carnosine. Ninth question, what is the main function of nitrites in meat preservation? A. Enhance flavor. B. Inhibit microbial growth. C. Improve texture. D. Prevent lipid oxidation. Here, which is the most appropriate answer? Here answer is option B. Inhibit microbial growth. prevent rancidity, and by also controlling the growth of bacteria, preserve the red pinkish color of the meat. 10th question. The huddle technology for meat preservation involves the combination of which factors? A. Temperature, pH and water activity, temperature and irradiation, packaging, salt concentration, none of the above. Here answer is option A, temperature, pH and water activity. 11th question. Which chemical is often used to prevent the blackening of shrimp during storage? Sodium benzoate, sodium bisulfite, potassium sorbate, ascorbic acid. Here answer is option B, sodium bisulfite. 12th question. The process of salting fish helps in preservation by reducing which parameter? Enzymatic activity, water activity, oxygen availability, microbial diversity. Salting helps in water activity, lower to low water activity. Answer is B, water activity. 12th question. What is the primary reason for using carbon monoxide in the preservation of aquatic foods? Flavor enhancement, color stabilization, inhibition of microbial growth, reduction of oxidation. Which is the answer? Here answer is B, color stabilization. 14th question. Which packaging technology is widely used for frozen seafood to prevent freezer burn? Vacuum packaging, modified atmosphere packaging, edible coatings, ice glazing. Your answer is option D, ice glazing. Food Safety Officer book is available with 1000 MCQs. You can order through Amazon link. It is available in the description or contact through these numbers. 15th question. Which enzyme is responsible for the spoilage of fish under aerobic storage conditions? Catalase, protease, lipase, trimethylamine oxide reductase. Which is the answer? Here answer is option D, trimethylamine oxide reductase. Fish spoilage bacteria reduce trimethylamine oxide to trimethylamine in the presence of triamine oxidase. Fish bacteria are mostly psychrophilic, growing at between 0 and about 30 degrees Celsius, with some growing at temperature as low as minus 7.5 degrees Celsius. Fish bacteria are sensitive to low pH too. 16th question. High levels of histamine in preserved fish or meat can lead to what health issue? Foodborne botulism, scromboid poisoning, gastroenteritis, anaphylactic shock. Your answer is option B, scromboid poisoning. Scromboid fish poisoning, also known as histamine fish poisoning, is an allergic type reaction that occurs within a few hours of eating fish contaminated with high levels of histamine. 17th question. The quality of frozen fish is primarily evaluated based on which parameter? Microbial load, protein content, drip loss, texture. Your answer is option C, drip loss. 18th question. What is a common term for the fraudulent practice of adding water or brine to meat to increase its weight? Overhydration, water gain adulteration, meat enhancement, watering down. Here answer is option C, meat enhancement. Enhanced meat can be defined as fresh, whole muscle meat that has been injected with a solution of water and other ingredients that may include salt, phosphates, antioxidants, and flavorings. 19th question. Which chemical is in meat preservation is regarded as the potential carcinogen? A. Sodium nitrate, B. Potassium sorbate, C. Benzoic acid, D. Sodium chloride. Here which is the answer? Here answer is option A, sodium nitrate. IARC also reported that ingested nitrate from processed meat can lead to colorectal cancer in human. Nitrate in sodium nitrate reacts with certain enzymes and proteins in the meat and produces nitrosamines. In the gastrointestinal tract, nitrosamine is non-cancer-causing agent and is associated with gastric, colorectal, and other cancers. Question number 20. Which antimicrobial coating is gaining popularity in meat packaging for extending shelf life? Chitosan-based films, cellulose acetate films, pectin-infused polymers, polyethylene glycol sheets. Here answer is option A, chitosan-based films. One of the most noteworthy applications of chitosan is the development of antimicrobial films. These films are utilized as food packaging material which can extend the shelf life of food products by inhibiting the growth of spoilage causing microorganisms. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notification. First question, Nilambur teak, a GI tagged wood product, is found in which Indian state which is also home to the GI tagged Wayanad coffee? Tamil Nadu, Kerala, Karnataka, Goa. Which is the answer? Here answer is option B, Kerala. Second question, the GI tagged gamosa, a traditional Assamese handloom product, shares cultural importance with which GI tagged beverage of Assam? Assam Orthodox tea, Nilgiri tea, Kangra tea, Sikkim tea. Your answer is option A, Assam Orthodox tea. Third question, which GI tagged chili variety grown in Karnataka is used for its bright red color rather than its spiciness? Pyadgi chili, Guntur chili, Food Jolokia, Kashmiri chili. Which is the answer? Your answer is option A, Byadgi chili. In the Haveri district of Karnataka lies the town of Byadgi, where the Byadgi varieties of chilies are grown. These chilies were granted the geographical indication tag in February 2011 with the tag number 129, and there are two main varieties from this area. Fourth question, Tezpur lychee, a GI tagged fruit from Assam, is grown primarily in which type of soil? Alluvial soil, loamy soil, sandy soil, lateritic soil. Which is the answer? Your answer is option A, alluvial soil. Fifth question, the GI tagged Manipuri black rice, also called chakao, is rich in which nutrient, giving it a distinct health benefit? Calcium, protein, iron, all of this? Your answer is option D, all of this. Manipuri black rice, also known as chakao, got its GI tag recently in 2020. This is yet another variant of rice, but with double the nutritional value, and its unique black color boasts of its rich antioxidant qualities. This rice is rich in essential vitamins and minerals, including iron and calcium. Sixth question, what are the GI tagged foods from Nagaland? Naga mircha, Naga tree tomato, Naga cucumber. Is the answer D, all of these? Seventh question, Bikaneri Phujia, a GI tag product, is known for its crunchiness and flavor. What flour forms its base? Chickpea flour, rice flour, corn flour, wheat flour. Which is the answer? Here answer is A, chickpea flour, made of high spices with moth besan. Chickpea flour. Moth besan gives hardness to texture, and it is blended with spice like black peppercorns, cinnamon, cloves, coriander, cilantro seeds, etc. to give sharp and intense taste. Eighth question, which GI tagged variety of coffee from Karnataka is grown under the canopy of forest trees, enhancing its flavor? Coorg Arabica coffee, Wayanad Robusta coffee, Baba Budagiri Arabica coffee, Araku Valley coffee. Which is the answer? Your answer is option C, Baba Budangiri Arabica coffee. Arabica beans grown at Baba Budangiri mountain range has been conferred with the prestigious GI tag in the year 2019 as 355th GI tag product of India. 10th question, Kovilpatti Kadale Mithai, a GI tag sweet from Tamil Nadu, uses jaggery and what primary ingredient? Rice flour, groundnuts, coconut, sesame seeds. Which is the answer? Here answer is option B, groundnuts. 11th question, the GI tagged Bastar Thokra, crafted in Chhattisgarh, is commonly associated with which GI tagged rice variety from the same state? Jeera Full rice, Sona Masuri rice, Gobi Thobong rice, Ambemohar rice. Which is the answer? Your answer is A, Jeera Full rice. 12th question, which GI tagged fruit from West Bengal, often served during festivals, is known for its smooth texture and high pulp content? A, Malda mango, B, Himasagar mango, C, Fazli mango, D, Lakshman Bhog mango. Which is the answer? Your answer is B, Himasagar mango. 13th question, the GI tagged Phalia wheat, grown in dry climates, is used to make which staple food of Gujarat? Thokla, Tepla, Bhakri, Fafda. Which is the answer? Your answer is option C, Bhakri. 14th question. The GI tagged Balangir costly sari shares its origin state with which GI tagged sweet made from fresh chena? Bengal's Sandesh, Odisha's Rasagola, Bihar's Khaja, Andhra Pradesh Uttarekulu. Which is the answer? Your answer is option B, Odisha's Rasagola. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers. Amazon link is available in the description. 15th question. The Katarni rice, known for its fine grains and fragrance, is a GI tag product from which state? Bihar, Uttar Pradesh, Odisha, West Bengal. Which is the answer? Your answer is option A, Bihar. Safety Officer book is available with 1000 MCQs from all sections. Contact to these numbers. Amazon link is available in the description. First question. Which of the following methods is used to detect synthetic coloring in food products? A. Chromatographic analysis. B. Iodometric titration. C. Nessler's test. D. Flame photometry. Which is the answer? Your answer is option A. Chromatographic analysis. Second question. Which adulterant is commonly used to enhance the sweetnesses of rice or sugar? Sodium bicarbonate, chalk powder, lead nitrate, boric acid. Which is a common adulterant in sugar or rice? Your answer is option B, chalk powder and urea are used. Most commonly used chalk powder. Third one, the retention time in HPLC is depend on mobile phase composition, column temperature, nature of analyte, all of the above. Which is the answer? Your answer is D, all of the above. Fourth question, what is the primary health hazard associated with the adulteration of mustard seeds with argemone seeds? Neurotoxicity, epidemic dropsy, hepatotoxicity, cardiomyopathy. Which is the answer? Your answer is option B, epidemic dropsy. We have discussed earlier. Fifth question, what is the most common stationary phase material in reverse phase HPLC? Silica gel, C18 bonded silica, polyacrylamide, cellulose derivatives. This is the answer. Your answer is option B, C18, carbon 18 bonded silica. Sixth question, gradient elution in HPLC is mainly used to enhance separation of complex mixtures, increase resolution for all analytes, ensure stable retention times, prevent column degradation. Which is the answer? Your answer is option A, enhance separation of complex mixtures. Seventh question, in HPLC the term tailing factor is used to describe peak asymmetry, solvent polarity, detector sensitivity, column efficiency. Which is the answer? Your answer is option A, peak asymmetry. Eighth question, in pesticide residue analysis, which of the following is used to extract residues from fruits and vegetables? Acetone, sodium hydroxide, ethyl acetate, hexane. Which is the answer? Your answer is option C, ethyl acetate. Food Safety Officer book is available with 1000 MCQs from all sections. Contact these numbers. Amazon link is available in the description. Ninth question. Why is a guard column used in HPLC systems? To extend the lifespan of the main column, to improve separation, to increase retention time, to decrease peak broadening. Which is the answer? Your answer is A, to extend the lifespan of the main column. 10th question. Which adulterant in milk is commonly detected using lactometer? Water, starch, urea, detergent. Here answer is option A, water. 11th question. The QUE-CHERS method is widely used in pesticide residue analysis because it is cost effective and involves fewer steps, specific to organochlorine pesticides, suitable only for non-polar pesticides, a visual detection method. Which is the answer? Your answer is option A, cost effective and involves fewer steps. The quick, easy, cheap, effective, rugged and safe method, that is known as QUECHERS method, has transformed sample preparation for many analytical methods. Developed initially to support pesticide residues testing in food, this technique now has applications across various fields. 12th question. What is the purpose of the diode array detector or DAD in HPLC? A. To identify compounds based on fluorescence. B. To simultaneously monitor multiple wavelengths. C. To separate compounds with similar polarity. D. To enhance resolution. Which is the answer? Your answer is option B. To simultaneously monitor multiple wavelengths. 13. Which food product is most susceptible to adulteration with methanol yellow? Milk, turmeric powder, wheat flour, sugar syrup. Which is the answer? Your answer is option B, turmeric powder. 14th question. The presence of formalin in fish is tested using iodine solution, chromic acid, Schiff's reagent, potassium permanganate solution. This is the answer. Your answer is option C, Schiff's reagent. 15th question. Which instrument is most suitable for analyzing pesticide residues in food analysis? HPLC, AAS, GCMS, UVV spectrophotometry. Which is the answer? Your answer is most sensitive one. Option is C, GCMS. 16th question. What is the primary detection method for melamine adulteration in milk? ELISA, near infrared spectroscopy, gas chromatography, Redal method. Which is the answer? Your answer is option B, near infrared spectroscopy. 17th question. The presence of starch in milk can be tested using Benedict solution, iodine solution, ninhydrin reagent, Filene solution. Here option B is correct, iodine solution. 18th question. Which method is used for detecting synthetic sugar substitutes in soft drinks? HPLC with refractive index detector, TLC with UV detector, GCMS with flame ionization detector, DAS with flame atomization. Which is the answer? Here answer is option A, HPLC with refractive index detector. 19th question. Which method is used to detect urea in milk? Benedict's test, bromothymol blue test, DMAB, dimethyl amino benzaldehyde test, resorcinol test. This is the answer. Your answer is option C, DMAB, dimethyl amino benzaldehyde. The addition of urea to milk can be detected by using para-dimethyl amino benzaldehyde. This method is based on the principle that urea forms a yellow complex with DMAB in a low acidic solution at room temperature. Food Safety Officer book is available with 1000 MCQs from all sections. You can contact these numbers. Amazon link is available in the description. 20th question, last question of this session. Adulteration of honey with glucose syrup can be detected by sulfur test, Figs test, Benedict's test, phenolphthalein test. Which is the answer? Your answer is Figs test. Add a few drops of Figs reagent to honey. If it turns red, it may be adulterated with starch. Cobalt chloride test. Add a few drops of cobalt chloride to honey. If it turns blue, it may be adulterated with glucose or other sugars. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Veterinary science. First question. Which project funded by the World Bank aims to create an ecosystem for a better animal health management system in India using the One Health approach? A. ADAHSSOH NAPI ICR NARM NHMS. Your answer is A. Animal Health System Support for One Health. Second question. Which Indian state recorded the highest number of new animal species discoveries in 2022? A. Tamil Nadu, B. Goa, C. Kerala, D. Uttarakhand. Your answer is Kerala, C. Kerala. Third question. Which institute in India is leading research on the indigenous vaccine for lumpy skin disease in cattle? A. IVRI Izadnagar, B. NDRI Karnal, C. TNAU was Chennai, D. ICR CIBA Chennai. Your answer is A. IVRI Izadnagar. Next question, fourth question. Which zoonotic disease associated with camels has been under WHO surveillance due to its potential pandemic risk? A. Nipah virus, B. MERS-CoV, C. Avian influenza. D. Brucellosis. Here answer is B. MERS-CoV. Fifth question. Which Indian veterinary institution recently developed rapid diagnostic kit for foot and mouth diseases? A. TNAU was. B. ICR Nivedi. C. IVRI. D. ICR NRC on camel. Answer is B. ICR NIVEDI. Next question. Sixth question. Which state in India launched a comprehensive insurance scheme for cattle in 2024? A. Gujarat. B. Uttar Pradesh. C. Karnataka. D. Punjab. Here answer is C. Karnataka. Seventh question. Which bacteria pathogen was recently identified as a major cause of neonatal mortality in piglets in India? A. E. coli. B. Clostridium perfringens. C. Salmonella. D. Streptococcus suis. Your answer is D. Streptococcus suis. Eighth question. The National Livestock Mission 2024 includes provisions for which new livestock category? A. Rabbits. B. Goats. C. Yak. D. Guinea pigs. Here answer is C. Yak. Ninth question. Which Indian veterinary research institute is working on genome editing to enhance disease resistance in cattle? A. ICAR IVRI. B. NDRI. C. ICRI Nivedi. D. ICR NRC on Mithun. Your answer is A. ICR IVRI. Next question, last question. The One Health approach focuses on the interconnection between humans, animals, and which of the domain? A. Economics. B. Environment. C. Technology. D. Genetics. Here answer is B. Environment. Which of the following bacteria is commonly associated with undercooked chicken and can cause a foodborne illness known as Campylobacteriosis? A. E. coli. B. Campylobacter jejuni. C. Staphylococcus aureus. D. Clostridium perfringens. Here answer is B. Campylobacter jejuni. Second question. Which foodborne illness is caused by a neurotoxin and is often linked to improperly canned foods? A. Botulism. B. Salmonellosis. C. Listeriosis. D. Typhoid fever. Here answer is A. Botulism. Third question. Which pathogen is most associated with hemolytic uremic syndrome or HUS, a severe complication of foodborne illness? A. Listeria monocytogenes. B. Vibrio cholerae. C. E. coli O157. D. Bacillus cereus. Here answer is C. E. coli O157. Fourth question. Which of the following foods is most commonly associated with Listeria monocytogenes contamination? A. Canned vegetables. B. Refrigerated ready-to-eat foods. Excel meat, soft cheeses. C. Cooked rice. D. Fresh fruits. Answer is B. Refrigerated ready-to-eat foods. Fifth question. The presence of which pathogen is commonly used as an indicator of fecal contamination in water supplies? A. Staphylococcus aureus. B. E. coli. C. Listeria monocytogenes. D. Vibrio cholerae. Here answer is B. E. coli. Sixth question. Which of the following pathogens can cause foodborne illness even after the food is reheated? A. Bacillus cereus, B. Clostridium botulinum, C. Campylobacter jejuni, D. Vibrio parahemolyticus. Here answer is A. Bacillus cereus. Seventh question. The term food intoxication refers to illness caused by A. Pathogens that multiply in the human body after ingestion, B. Ingesting toxins produced by pathogens in food, C. Viral infections contracted through food, D. Allergic reactions to certain food proteins. Here answer is B. Ingesting toxins produced by pathogens in food. Eighth question. Which type of hepatitis virus is most commonly transmitted through contaminated food and water? A. Hepatitis B, B. Hepatitis C, C. Hepatitis A, D. Hepatitis E. Here answer is C. Hepatitis A. Ninth question. Which of the following pathogens is associated with foodborne illness due to consumption of raw or undercooked seafood? A. Norovirus, B. Vibrio parahemolyticus, C. Clostridium perfringens, D. Salmonella typhimurium. Answer is B. Vibrio parahemolyticus. 10th question. Staphylococcus aureus causes food poisoning due to the production of A. Endotoxins, B. Enterotoxins, C. Exotoxins, D. Mycotoxins. Your answer is B. Enterotoxins. If you like this channel, please share and subscribe. Don't forget to click the bell button to get notifications. Which of the following bacteria is commonly associated with undercooked chicken and can cause a foodborne illness known as Campylobacteriosis? A. E. coli, B. Campylobacter jejuni, C. Staphylococcus aureus, D. Clostridium perfringens. Your answer is B. Campylobacter jejuni. Second question. Which foodborne illness is caused by a neurotoxin and is often linked to improperly canned foods? A. Botulism, B. Salmonellosis, C. Listeriosis, D. Typhoid fever. Here answer is A. Botulism. Third question. Which pathogen is most associated with hemolytic uremic syndrome or HUS, a severe complication of foodborne illness? A. Listeria monocytogenes, B. Vibrio cholerae, C. E. coli O157, D. Bacillus cereus. Here answer is C. E. coli O157. Fourth question. Which of the following foods is most commonly associated with Listeria monocytogenes contamination? A. Canned vegetables, B. Refrigerated ready-to-eat foods, excel meat, soft cheeses, C. Cooked rice, D. Fresh fruits. Answer is B. Refrigerated ready-to-eat foods. Fifth question. The presence of which pathogen is commonly used as an indicator of fecal contamination in water supplies? A. Staphylococcus aureus, B. E. coli, C. Listeria monocytogenes, D. Vibrio cholerae. Here answer is B. E. coli. Sixth question. Which of the following pathogens can cause foodborne illness even after the food is reheated? A. Bacillus cereus, B. Clostridium botulinum, C. Campylobacter jejuni, D. Vibrio parahemolyticus. Here answer is A. Bacillus cereus. Seventh question. The term food intoxication refers to illness caused by A. Pathogens that multiply in the human body after ingestion, B. Ingesting toxins produced by pathogens in food, C. Viral infections contracted through food, D. Allergic reactions to certain food proteins. Here answer is B. Ingesting toxins produced by pathogens in food. Eighth question. Which type of hepatitis virus is most commonly transmitted through contaminated food and water? A. Hepatitis B, B. Hepatitis C, C. Hepatitis A, D. Hepatitis E. Here answer is C. Hepatitis A. Ninth question. Which of the following pathogens is associated with foodborne illness due to consumption of raw or undercooked seafood? A. Norovirus, B. Vibrio parahemolyticus, C. Clostridium perfringens, D. Salmonella typhimurium. Answer is B. Vibrio parahemolyticus. 10th question. Staphylococcus aureus
causes food poisoning due to the production of A. Endotoxins, B. Enterotoxins, C. Exotoxins, D. Mycotoxins. Your answer is B. Enterotoxins.
Neuroletharism is primarily caused by the excessive consumption of lentils, grass pea, chickpeas, soybeans. The answer is grass pea. B. Grass pea Letharis sativus. Neuroletharism is a neurologic disorder caused by the prolonged consumption of grass pea, which contains a neurotoxic amino acid. It affects motor neurons. Second question. The main toxic compounds in grass pea that causes neuroletharism is C and the answer. It is a neurotoxic amino acid found in grass pea which causes neuroletharism. Third, which of the following symptom is not typically associated with neuroletharism? Spastic paraparesis, muscle weakness, vision impairment, paralysis of the lower limbs. CL ever answer with C. Fourth question. Neuroletharism primarily affects which part of the body? Respiratory system or digestive system or lower limbs or upper limbs or lower limbs. Lower limbs. Neuroletharism predominantly causes paralysis spasticity in the lower limbs. Fifth question. Which of the following population is most at risk of neuroletharism? Population consuming high amounts of processed foods, people in arid famine-prone regions relying on grass pea as a food source, urban population with varied diets, individuals following vegan diet. അതായത് ആൻസർ ഇവിടെ വരിക ബി ആണ്. പീപ്പിൾ ഇൻ ആരിഡ് ഫാമിൻ പ്രോൺ റീജിയൻസ് റിലൈങ് ഓൺ ഗ്രാസ് പീ as a food source. Sixth question. Which historical figure is credited with the earliest known documentation of letharism? Galen, Hippocrates, Avicenna, Paracelsus. നമ്മൾ ഡിസ്കസ് ചെയ്തു ആരാണ് ഹിപ്പോക്രാറ്റസ്, ഫാദർ ഓഫ് മെഡിസിൻ. അദ്ദേഹമാണ് ആദ്യം ഇതിൻ്റെ റിപ്പോർട്ട് ചെയ്തിട്ടുള്ളത്. ഈ ഡീറ്റെയിൽസ് നമ്മൾ ഡിസ്കസ് ചെയ്താണ് മുമ്പത്തെ സ്ലൈഡിൽ. സെവൻത് ക്വസ്റ്റ്യൻ. ന്യൂറോലെത്താരിസം പ്രൈമർലി എഫക്ട്സ് വിച്ച് പാർട്ട് ഓഫ് ദി നെർവസ് സിസ്റ്റം? അതായത് അപ്പർ മോട്ടോർ ന്യൂറോൺസ് ആണ് ആൻസർ. യെസ്, അപ്പർ മോട്ടോർ ന്യൂറോൺസ് ആണ് അതിൻ്റെ ആൻസർ. എയ്ത്ത് ക്വസ്റ്റ്യൻ. വിച്ച് ഓഫ് ദ ഫോളോയിങ് പ്രിവെന്റീവ് മെഷേഴ്സ് ക്യാൻ ഹെൽപ്പ് റെഡ്യൂസ് ദ റിസ്ക് ഓഫ് ന്യൂറോലെത്താരിസം? ഈറ്റിംഗ് ഗ്രാസ് പീ വിത്ത് വൈറ്റമിൻ സി, genetic modification of grass pea to reduce beta ODAP levels, increasing grass pea consumption during famine, avoiding all legumes. പിന്നെ ആൻസർ വരിക ബി ആണ് ആൻസർ വരിക. ജനറ്റിക് മോഡിഫിക്കേഷൻ ഓഫ് ഗ്രാസ് പീ ടു റെഡ്യൂസ് ദ ODAP levels. Reducing beta ODAP content in grass pea through genetic modification can lower its neurotoxic risk, making it safer for consumption. Ninth question. In which countries is neuroletharism currently considered endemic? കാനഡ ആൻഡ് യുണൈറ്റഡ് സ്റ്റേറ്റ്സ്, എത്യോപ്യ, ഇന്ത്യ, ബംഗ്ലാദേശ്. നമ്മൾ നേരത്തെ നോക്കിയല്ലേ, ബി ആണ് അതിൻ്റെ ആൻസർ വരിക. Ethiopia, India and Bangladesh. 10th question. Which of the following symptoms is typically seen in the advanced stages of neuroletharism? Complete lower limb paralysis and spasticity. That's the answer. And the answer is complete lower limb paralysis and spasticity. If you like this channel, please share and subscribe. Don't forget to click the bell button. Which of the following statements about selective culture media is correct? Statements. Selective media contain substances that inhibit the growth of certain microbes. They are primarily used to isolate fastidious organisms. Antibiotics are often added to these media for selective inhibition. Selective media are used in diagnostic microbiology to differentiate closely related species. Options A, B, C, D. Which is correct? The answer is A. Statements 1 and 3. Explanation. Selective media contain inhibitors like antibiotics to suppress the growth of unwanted organisms. They are used to isolate specific organisms, but not typically for fastidious species or species differentiation, which are usually the purpose of enriched and differential media. Second question. Which of the following media is particularly useful for isolating and identifying Mycobacterium tuberculosis? Statements. Lowenstein-Jensen medium, LJ medium is commonly used. The medium contains malachite green as an inhibitory agent. Egg-based media are preferred to support the lipid-rich cell wall. Charcoal is added to enhance growth. Which is correct? Answer is B. Statement 1, 2 and 3. Explanation. LJ medium is egg-based and contains malachite green to inhibit contaminants. Charcoal is not typically used in media for Mycobacterium tuberculosis. Third question. Which of the following statements accurately describes blood agar? Statements. 1. It is a differential medium that allows observation of hemolytic activity. Second one, it is both selective and differential for fastidious organisms. Third, blood agar is enriched with sheep blood to support bacterial growth. It is useful for identifying hemolytic patterns of Streptococcus species. Which is the answer? Here answer is A. Statement 1, 3 and 4. Explanation. Blood agar is enriched but not selective. It allows observation of hemolysis alpha, beta and gamma and supports the growth of fastidious organisms like Streptococcus species. Fourth question. Which of the following culture techniques utilizes a low nutrient medium to promote biofilm formation? Statements. This technique is called a Calgary biofilm device. Nutrient restriction enhances biofilm formation as microbes adhere to surfaces. This technique is not suitable for high throughput testing. Biofilm growth on these media is observed with crystal violet staining. Option statement 1, 2, 4 or all statements. Which is correct? Here is A. Statements 1, 2 and 4. The Calgary biofilm device supports biofilm studies using a low nutrient medium. Crystal violet staining quantifies biofilm formation. However, it is designed for high throughput analysis. Fifth question. Which media is commonly used to isolate and identify Vibrio species? Statements. Thiosulfate TCBS agar is a selective medium for Vibrio. TCBS agar helps to differentiate Vibrio species based on colony color. The high pH of TCBS agar suppresses most non-Vibrio organisms. Bile salts and thiosulfate act as pH indicator. Which is the answer? Here answer is A. Statement 1 and 2, 1, 2 and 3. TCBS agar is selective for Vibrio species, differentiates them based on sucrose fermentation and has an alkaline pH that inhibits other organisms. Bile salts and thiosulfate do not serve as pH indicators. Sixth question. Which of the following techniques is specifically associated with anaerobic culture media? Statements. Reducing agents like thioglycolate are added to the medium. Plates are incubated in a carbon dioxide rich environment. An anaerobic chamber or jar is required for culturing. Anaerobic indicators change color to confirm oxygen absence. Option A, B, C, D. Which is correct? Their answer is option A. Statement 1, 3 and 4. Explanation. Reducing agents, example thioglycate and anaerobic chambers are common for culturing anaerobes. Carbon dioxide alone does not ensure an anaerobic environment. Seventh question. Which statements are true for McConkie agar as a differential medium? Statements. It differentiates bacteria based on lactose fermentation. Lactose fermenters produce pink colonies due to acid production. McConkie agar contains bile salts and crystal violet to inhibit gram-positive bacteria. It is specifically designed to isolate Neisseria species. Option statement 1, 2, 3, statement 1 and 3, all statements, statement 2, 3 and 4. Which is the answer? Here answer is A, statement 1, 2 and 3. Explanation. McConkie agar is used for gram-negative bacteria and differentiates lactose fermenters. It is not selective for Neisseria species. Eighth question. Which statements are true for the technique of serial dilution and plating? Statements. It is used to estimate bacterial concentration in a sample. Serial dilution reduces cell concentration in a stepwise manner. This method is most useful for differentiating bacterial species based on morphology. Colonies are counted to calculate the original cell concentration. Options A, B, C and D, which is correct. Your answer is A, statement 1, 2 and 4. Explanation. Serial dilution and plating estimate the bacterial count by creating dilutions, plating them and counting colonies. Morphology differentiation is not the primary goal. Ninth question. Which of the following culture media is used specifically to promote fungal growth? Statements. SDA is formulated for fungi. SDA has a low pH that inhibits bacterial growth. SDA is enriched with chloramphenicol to selectively inhibit bacterial contaminants. SDA is used to isolate mycobacterium species. Which is correct here? Your answer is A, statement 1, 2 and 3. Saborodextrose agar is formulated for fungi with a low pH and sometimes antibiotics like chloramphenicol or to inhibit bacteria. It is not used for mycobacterium. 10th question. Which of the following is true about the use of EMB agar? Statements. EMB agar is both selective and differential. It differentiates based on lactose fermentation. E. coli produces metallic green colonies due to acid production. EMB is used to cultivate anaerobic bacteria. Which is the correct answer here? Your answer is A, statement 1, 2 and 3. EMB agar is selective for gram-negative bacteria and differentiates lactose fermenters. E. coli typically produces a metallic green sheen, and EMB is not suited for anaerobes. Which staining method is primarily used to differentiate bacteria with a high mycolic content in their cell wall? Statement. This stain uses a primary dye that is lipid soluble. It involves heating the slide to ensure dye penetration. A decolorizer is used to remove the stain from cells lacking mycolic acid. A counter stain is applied to visualize non-acid-fast cells. Option A. Gram staining. B. Acid-fast staining. C. Endosphore staining. D. Capsule staining. Here answer is B. Acid-fast staining. Acid-fast staining is used to identify bacteria with mycolic acid, primarily Mycobacteria species. Heating and lipid-soluble dye like carbol fusion are the key steps. Second question. Which technique is specifically used to identify bacterial endospores? Statements. The method uses malachite green as primary stain. Heat is applied to ensure dye penetration into the endospore. The vegetative cells retains a pink or red color after counterstaining. It is helpful for differentiating spore-forming bacteria. Option A. Gram staining. B. Negative staining. C. Endospore staining. D. Acid-fast staining. Which is the answer? Answer is C. Endospore staining. Endospore staining, often called Schaefer-Fulton method, uses malachite green for the endospores and safranin for the vegetative cells. Third question. In fluorescence staining, which of the following statements are true regarding the use of fluorochromes? Statements. Fluorochromes emit light when excited by a specific wavelength. They are primarily used to stain acid-fast bacteria. DAPI and acridine orange are examples of commonly used fluorochromes. Fluorescence staining is useful in both live and fixed cells. Options A, B, C, D. Which among the following is or are correct? Here answer is B. Statement 1, 3 and 4 are correct. Explanation. Fluorochromes like DAPI and acridine orange bind to nucleic acids, allowing for visualization of cells components. They are not specific to acid-fast bacteria. Fourth question. Which statement about gram staining is incorrect? Statements. Crystal violet is used as the primary stain. Iodine acts as mordant to fix the stain in gram-positive cells. Alcohol is used as decolorizer. Gram-negative bacteria appear purple after staining. Statement 1, 2, 3, 4. Which is the correct answer? Your correct answer is D. Statement 4. Explanation. Gram-negative bacteria appear pink or red after staining, not purple due to the counter stain saffron. Fifth question. Which staining method is used to visualize bacterial capsules? Statements. Capsules do not absorb moist or stains due to their non-ionic nature. Negative staining stains the background but leaves the capsule clear. The stain typically used is Indian ink or resin. Capsules are stained with a dark color, making them appear prominent. Options. Gram staining, B. Acid-fast staining, C. Capsule staining, D. Endospore staining. Which is the answer? Your answer is C. Capsule staining. Explanation. Capsule staining is a type of negative staining that leaves the capsule clear against a dark background. Sixth question. Which advanced technique allows for visualization of specific cellular structures by labeling with antibodies? Statements. It relies on fluorochrome labeled antibodies binding to cellular targets. It can differentiate between different bacterial strains based on antigen presence. This technique is often called immunostaining. The primary dye used is crystal violet. Option. Acid-fast staining, B. Fluorescence immunostaining, C. Gram staining, D. Endospore staining. Which is the answer? The answer is B. Fluorescence immunostaining. Explanation. Fluorescence immunostaining uses fluorochrome labeled antibodies to target specific structures within cells, not crystal violet. Seventh question. In electron microscopy staining, which of the following applies to negative staining? Statements. It enhances contrast by staining the background rather than the specimen. Uranyl acetate and phosphotungstic acid are commonly used. It is primarily used for observing bacterial spores. It allows for high resolution imaging of viruses and small particles. Options. Which among the following is correct? The answer is A. Statements 1, 2 and 4 are correct. Explanation. Negative staining in electron microscopy stains the background, not spores specifically, making it ideal for imaging viruses and nanoparticles. Eighth question. Which statement about Zeal-Nielsen staining is true? Statements. It requires the use of an acid alcohol decolorizer. Methylene blue is used as primary stain. Its primary use for identifying spore-forming bacteria. Acid-fast organisms appear red after staining. Options. Which among the following is correct? Answer A. Statement 1 and 4. Explanation. Zeal-Nielsen staining uses acid alcohol for decolorization, and acid-fast bacteria retain the red dye, carbon fusion. Ninth question. Which of the following is true about the right Giemsa stain? Statements. It is commonly used to identify blood parasites. It is a type of differential stain. Methylene blue and eosin are the primary stains. It is mainly used to identify acid-fast bacteria. Which is correct answer? Your answer is A. Statement 1, 2 and 3 are correct. Explanation. Right Giemsa stain is used for blood smears, particularly for parasites, not for acid-fast bacteria. 10th question. Which of the following applies to the use of periodic acid Schiff's paste stain? Statement. It is used to highlight polysaccharides in the cell walls of fungi. Paste staining turns polysaccharide a bright red or magenta color. This stain requires a mordant to set the dye. It is effective in staining bacterial capsules. Which is the correct answer? Here answer is option A, statement 1 and 2. Explanation. The paste stain detects polysaccharides, coloring them red or magenta, commonly used for fungal cell walls rather than bacterial capsules. 11th question. Which of the following staining methods is specifically used to distinguish between live and dead cells in sample? Statements. The technique often uses two different fluorescent dyes. Live cells generally appear green while dead cells appear red. It is commonly used in environment microbiology. It requires the use of an acid-fast decolorizer. Which is the correct answer? Here answer is B. Vital staining. Explanation. Vital staining uses fluorescent dyes like SYTO9 and propidium iodide, which helps differentiate between live, green and dead red cells in samples. 12th question. Which of the following statements is true about the concurrent staining technique? Statements. It is a negative stain often used to observe bacterial capsules. Congo red is acidic and stains bacterial cells directly. Capsules appear as clear halos around cells. This stain is removed by rinsing with water to visualize the capsule. Option. Statement A, B, C is correct answer. Here answer is D. Statement 1 and 3. Explanation. Congo red is a negative stain for capsules. Capsules don't absorb dye, making them appear as clear halos, but it is not rinsed with water as the stain binds to the background. 13th question. In the context of fluorescence microscopy, which statement is accurate about the use of DAPI as a staining agent? Statements. DAPI binds strongly to AT-rich regions in DNA. It emits a green fluorescence when bound to DNA. This stain is used to visualize bacterial spores. It can penetrate both live and dead cells, making it suitable for fixed samples. Options. Statement 1 and 4, B, C, D. Which among the following is correct? The correct answer is A. Statement 1 and 4. Explanation. DAPI binds to AT-rich regions and emit blue fluorescence. It can penetrate both live and dead cells. Ideal for fixed cell imaging. 14th question. Which of the following describes the principle behind gram staining? Statements. It differentiates bacterial based on cell wall composition. Gram staining bacteria have a thick peptidoglycan layer. Crystal violet is the primary stain that binds to gram-positive cells. The outer membrane of gram-negative bacteria impedes crystal violet uptake. Which is the answer? Here answer is B. Statement 1, 3 and 4. Explanation. Gram staining distinguishes between gram-positive and gram-negative bacteria due to cell wall differences, specifically peptidoglycan thickness and outer membrane composition. 15th question. Which type of staining technique allows for the observation of bacterial flagella? Statements. It requires a mordant to increase flagella diameter for visibility. The stain can be viewed under a light microscope. Flagella staining highlights both internal and external cell structures. Crystal violet is typically used as stain. It is the answer. Here answer is B. Statement 1 and 2. Explanation. Flagella staining requires a mordant to increase flagella diameter, making it visible under a light microscope. It is specialized for external structures only. 16th question. Which of the following statements about the hemotoxicillin eosin stain is correct? Statements. Hemotoxicillin stain acid structures such as the nucleus a blue-purple color. Eosin stains basic structures such as the cytoplasm pink. HNE staining is commonly used in bacteriology. It is especially effective for visualizing endospores. Options A, B, C, D. Which is correct? Here answer is A. Statement and 2. Explanation. HNE staining is widely used in histology, not typically in bacteriology, and it is effective for distinguishing cellular structures in eukaryotic cells. 17th question. Which of the following is a unique feature of negative staining? Statements. It highlights cell structures without penetrating the cells. India ink is commonly used as the stain. Negative staining is primarily used to observe capsules. It stains the bacterial cell, leaving the background clear. Option A, B, C, D. Which is correct? Here, correct answer is A. Statement 1, 2, 3. Explanation. Negative staining stains the background, not the cell, and is often used for observing capsules with stains like Indian ink. 18th question. Which stain is specifically used in confocal microscopy to observe biofilm structures? Statements. Chalcofluor white is a common stain for this application. The stain binds to polysaccharide in the biofilm matrix. The method can only be used on live samples. It provides 3D imaging of biofilms. Which is the answer? Here answer is B. Statement 1, 2 and 4. Explanation. Chalcofluor white binds to biofilm, polysaccharides and confocal microscopy enables 3D visualization. This technique is applicable to both live and fixed samples. 19th question. Which of the following is the characteristics of silver staining technique? Statements. It is useful for visualizing flagella. Silver nitrate is used as the primary stain. This technique is commonly used in histology for nerve fibers. It requires counterstain to enhance contrast. Which is the correct answer? Here answer is C. Statements 2 and 3. Explanation. Silver staining uses silver nitrate to enhance visibility and is often used in histology, particularly for staining nerve fibers and flagella. 20th question. Last question. Which statements accurately describe the function of iodine in gram staining? Statements. Iodine acts as mordant, helping to fix the primary stain in the cell wall. It forms a complex with crystal violet to make the stains less soluble. It is used to decolorize the slide after primary staining. It helps to distinguish between gram-positive, gram-negative bacteria. Which is correct? Here answer is A. Statement 1, 2 and 4. Iodine acts as mordant by forming a complex with crystal violet, enhancing stain retention in gram-positive gram-positive cells, and helping to differentiate between gram-positive and gram-negative bacteria. If you like this channel, please share and subscribe. Statement. A. Phase contrast microscopy is ideal for observing live unstained cells. B. Dark field microscopy enhances contrast by using scattered light. C. Transmission electron microscopy allows for three-dimensional imaging. D. Scanning electron microscopy is suited for detailed surface visualization. Here conclusion. Which of the following statements is incorrect? A only, C only, D only. All statements are correct. ഏതാ അതിന്റെ ആൻസർ? ഇതിന്റെ ആൻസർ ആണ് 2, C only. Transmission electron microscope provides detailed internal structures in 2 dimensions, while some provide surface views in 3 dimensions. Second question. A. Fluorescence microscopy uses fluorophores to visualize specific cellular components. B. Confocal microscopy provides high resolution 3D images using optical sectioning. C. Atomic force microscopy involves an electron beam to capture surface details. Differential interference contrast microscopy uses polarized light for contrast. Conclusion. Which statement is false? A only, B only, C only, D only. ഏതാ അതിന്റെ ആൻസർ? ഇതിന്റെ 3, C only. AFM uses a mechanical probe, not an electron beam, to create topographical images. Third question. First statement. Electron microscopy requires the sample to be in a vacuum environment. B. Super resolution microscopy exceeds the diffraction limit of light. C. Bright field microscopy typically requires staining to enhance visibility. D. Confocal microscopy cannot capture fluorescent images. Conclusion. Which statement is incorrect? A only, B only, C only, D only. What is the answer? The answer is fourth one, D only. Incorrect statement, D only. Explanation. Confocal microscopy is often used to capture fluorescent images with high resolution. Fourth question. Statement A. TEM samples must be ultra thin to allow electron transmission. B. STEM is capable of viewing living specimens in real time. C. Fluorophores are used in fluorescence microscopy to mark cellular structures. D. Cryo-electron microscopy is used to study biological samples at cryogenic temperatures. Conclusion. Which statement is false? A only, B only, C only, D only. What is the answer? B only. Some requires sample to be dehydrated and is not suited for live specimens. Fifth question. Which microscopy technique uses laser light and pinholes to achieve optical sectioning? TEM, SEM, confocal microscopy, dark field microscopy. What is the answer? The answer is C. Confocal microscopy. Confocal microscopy uses lasers and pinholes to eliminate out of focus light, producing high resolution images. Sixth question. The resolution limit for traditional light microscopy is approximately 10, 0.2 micrometer, 2 nanometer, 1 nanometer. Your answer is B. 0.2 micrometer. This resolution is based on their diffraction limit of visible light. Seventh question. What is the primary advantage of cryo-electron microscopy over traditional transition electron microscopy? Ability to image live cells, provides color images, allows imaging without dehydration or staining, can view samples in a vacuum. And the answer is C. Allows imaging without dehydration or staining. Explanation. Cryo-electron microscopy enables imaging of samples in near native states without dehydration. Eighth question. Which technique in super resolution microscopy uses sequential activation and imaging of individual fluorescent molecules? A. STORM, B. SEM, C. DIC, D. Phase contrast microscopy. Here answer is A. STORM. STORM means stochastic optimal reconstruction microscopy, allows imaging below the diffraction limit. Ninth question. In AFM, atomic force microscopy, the image is generated by light scattering, fluorescence, a mechanical probe scanning the surface, electron beams. Here answer is C. A mechanical probe scanning the surface. Explanation. AFM uses a cantilever with a fine tip to scan sample surfaces, creating high resolution topographic images. 10th question. What is the function of the condenser in a bright field microscope? To magnify the image, to improve contrast, to focus light onto the sample, to capture light from the sample. Which is the answer? Here answer is C. To focus light onto the sample. To focus light onto the sample, the condenser directs lights onto the specimen, enhancing visibility. Which of the following bacteria is commonly associated with undercooked chicken and can cause a foodborne illness known as Campylobacteriosis? A. E. coli, B. Campylobacter jejuni, C. Staphylococcus aureus, D. Clostridium perfringens. Answer is B. Campylobacter jejuni. Second question. Which foodborne illness is caused by a neurotoxin and is often linked to improperly canned foods? A. Botulism, B. Salmonellosis, C. Listeriosis, D. Typhoid fever. Here answer is A. Botulism. Third question. Which pathogen is most associated with hemolytic uremic syndrome (HUS), a severe complication of foodborne illness? A. Listeria monocytogenes. B. Vibrio cholerae. C. Escherichia coli O157. D. Bacillus cereus. Answer is E. coli O157. Fourth question. Which of the following foods is most commonly associated with Listeria monocytogenes contamination? A. Canned vegetables. B. Refrigerated ready-to-eat foods. Example, deli meats, soft cheeses. D. Cooked rice. D. Fresh fruits only. B. Answer is B. Refrigerated ready-to-eat foods. Fifth question. The presence of which pathogen is commonly used as indicator of fecal contamination in water supplies? A. Staphylococcus aureus. B. E. coli. C. Listeria monocytogenes. D. Vibrio cholerae. Here answer is B. E. coli. Sixth question. Which of the following pathogens can cause foodborne illness even after the food is reheated? A. Bacillus cereus. B. Clostridium botulinum. C. Campylobacter jejuni. D. Vibrio parahemolyticus. Here answer is A. Bacillus cereus. Next question. Seventh question. The term food intoxication refers to illness caused by A. Pathogens that multiply in the human body after ingestion. B. Ingestion toxins produced by pathogens in food. C. Viral infections contracted through food. D. Allergic reactions to certain food proteins. Your answer is B. Ingesting toxins produced by pathogens in food. Eighth question. Which type of hepatitis virus is most commonly transmitted through contaminated food and water? A. Hepatitis B. B. Hepatitis C. C. Hepatitis A. D. Hepatitis E. Here answer is C. Hepatitis A. Ninth question. Which of the following pathogens is associated with foodborne illness due to consumption of raw or undercooked seafood? A. Norovirus. B. Vibrio parahemolyticus. C. Clostridium perfringens. D. Salmonella typhimurium. Here answer is B. Vibrio parahemolyticus. 10th question. Last question. Staphylococcus aureus causes food poisoning due to the production of A. Endotoxins. B. Enterotoxins. C. Exotoxins. D. Mycotoxins. Here answer is B. Enterotoxins. Thank you. All the best for your exam. A. Milk pasteurization helps to reduce pathogenic bacteria. Homogenization helps in distributing fat evenly in milk. UHT treatment allows milk to be stored for long periods at room temperature. D. Lactose is the main carbohydrate in milk. Conclusion. Which of the following statements is not correct? A only, C only, B only, all statements are correct. Which is correct? A, C, B or all statements are correct? Your answer is fourth one. All statements are correct. Next statement. Casein is the primary protein found in cow's milk. Homogenization increases milk's nutrient content. Probiotic yogurt contains live beneficial bacteria. Pasteurization does not completely sterilize milk. Which statement is incorrect? വളരെ ആലോചിച്ചതിന് ശേഷം മാത്രമേ ആൻസർ ചെയ്യാൻ പാടുള്ളൂ. എ മാത്രമാണോ, ബി മാത്രമാണോ, സി മാത്രമാണോ, ഡി മാത്രമാണോ? ഏതാ ഇതിൽ കറക്റ്റ്? ഇതിന് ആൻസർ ബി ഓൺലി ആണ്. ഹോമോജൈനൈസേഷൻ increases milk's nutrient content. Then the explanation എന്താ? Homogenization does not change nutrient content but prevents fat separation. Next statement. Rennet is used to coagulate milk proteins in cheese making. ഇത് കറക്റ്റ് ആണോ? അതുപോലെ തന്നെ UHT milk does not need refrigeration if unopened. Lipase enzymes in milk can cause spoilage. Evaporation removes fat content from milk. Conclusion. Which statement is false? ഏത് സ്റ്റേറ്റ്മെന്റ് ആണ് ഇവിടെ ഫാൾസ് ആയിട്ടുള്ളത്? പറയൂ. ആൻസർ നിങ്ങൾ ആൻസർ ചെയ്തു ആദ്യം, അതിന് ശേഷം ഞാൻ പറയാം. പറഞ്ഞോളൂ. ഏതാ അതിന്റെ ആൻസർ? ആൻസർ ഈസ് D only. Evaporation removes water, not fat. What choices? Which statement is false? Na choichitullathu. Fourth question. Statement. Lactose intolerant individuals lack sufficient lactase enzyme. Vitamin A is added to milk to support vision health. Lactic acid bacteria play a role in yogurt fermentation. Casein is a carbohydrate found in milk. Which statement is incorrect? ഏത് സ്റ്റേറ്റ്മെന്റ് ആകും ഇൻകറക്റ്റ്? ഇതിലെ ഇൻകറക്റ്റ് സ്റ്റേറ്റ്മെന്റ് പറഞ്ഞോളൂ, ആൻസർ ചെയ്തോളൂ. അത് ഡി ഓൺലി ആണ് അല്ലേ? ഫോർത്ത് വൺ ഡി ഓൺലി. അതായത് കാസീൻ ഈസ് എ കാർബോഹൈഡ്രേറ്റ് ഫൗണ്ട് ഇൻ മിൽക്ക് ഈസ് ഇൻകറക്റ്റ്. Casein is a protein, not a carbohydrate. Which of the following enzymes is primarily responsible for the spoilage of milk lipids resulting in rancidity? Protease, lipase, lactase, catalase. ഇതിൽ ഏതായാലും ആൻസർ? ആൻസർ ബി, lipase. Lipase breaks down milk fat, leading to rancidity if not controlled. Sixth question. The primary component responsible for the white color of milk is lactose ആണോ, casein miscellaneous ആണോ, fat globules ആണോ, അതോ വാട്ടർ ആണോ? ഏതാ അതിൻ്റെ ആൻസർ? ഇതിൻ്റെ ആൻസർ ബി ആണ്, casein miscellaneous. Casein miscellaneous. Explanation. Casein miscellaneous scatter light, giving milk its white appearance. Next, seventh question. Which bacteria is predominantly used as a starter culture in the production of cheddar cheese? നമ്മൾ ഓൾറെഡി ഇത് ഡിസ്കസ് ചെയ്താണ്. ലാക്ടോകോക്കസ് ലാക്ടിസ് ആണോ? ഓപ്ഷൻ ബി സ്ട്രെപ്റ്റോകോക്കസ് തെർമോഫിലസ്. ഓപ്ഷൻ സി ലാക്ടോകോക്കസ് ആസിഡോഫിലസ്. ഓപ്ഷൻ ഡി സാക്കറോമൈസസ് സെർവീസിയെ. Which among the following is the answer? Here answer is A, Lactococcus lactis. Explanation. Lactococcus lactis is commonly used to produce the specific texture and flavor of cheddar. Eighth question. Which dairy product requires the use of thermophilic bacteria during its production? Cheddar cheese, cottage cheese, yogurt, sour cream. ഏതാ ആൻസർ? പിന്നെ ആൻസർ സി, യോഗർട്ട് ആണ്. Thermophilic bacteria we have already discussed in the last class. Explanation. Thermophilic bacteria like Lactobacillus bulgaricus and Streptococcus thermophilus thrive at higher temperatures and are essential for yogurt production. Oh, don't forget ഏതൊക്കെ ബാക്ടീരിയ? Thermophilic bacteria like Lactobacillus bulgaricus and Streptococcus thermophilus. Ninth question. Which type of milk has the highest protein content? A, skim milk, B, whole milk, C, evaporated milk, D, condensed milk. ഇതിലെ ആൻസർ എന്താ? ആൻസർ ചെയ്തോളൂ. ഏതാ ആൻസർ? Evaporated milk. C ആണ് കേട്ടോ, evaporated milk. Evaporated milk is concentrated, increasing protein and nutrient content. 10th question. What is the role of sodium citrate in processed cheese production? A, preservative. Is used as preservative, emulsifier, flavor enhancer, acid regulator. എന്താ ഇതിലെ ആൻസർ? ഇതിൻ്റെ ആൻസർ it is used as emulsifier. Sodium citrate acts as an emulsifying agent to help distribute fat evenly in processed cheese, giving it a smooth texture. Which of the following statements about enzymatic browning in food processing is correct? ഇതാണ് correct. First one. Enzymatic browning occurs due to the action of polyphenol oxidase enzymes on phenolic compounds. Next, the pH or adding antioxidants can inhibit enzymatic browning. Third, enzymatic browning is beneficial in processes like tea and cocoa fermentation but undesirable in fresh cut produce. Option A, only statement 1 is correct. B, option statements 1 and 2 are correct. C, statements 1, 2 and 3 are correct. D, only statement 3 is correct. ഏതാ അതിൻ്റെ ആൻസർ വരിക? ആൻസർ നോക്കൂ, അത് വായിച്ചതിന് ശേഷം ഏതാ അതിൻ്റെ ആൻസർ വരിക? ആൻസർ വരാൻ ചാൻസ് ഏതാ? ആൻസർ C, statement 1, 2 and 3 are correct. അതാണ് അതിൻ്റെ ആൻസർ. Second question. regarding the freeze drying process. Which of the following statements is accurate? First one, freeze drying removes water content through sublimation, directly turning ice into vapor. Correct or no? Second, this process preserves the texture and nutrients of food more effectively than other drying methods. 3. Foods freeze dried are shelf stable and do not require any further packing to maintain stability. ഇതിൽ ഏതൊക്കെയാണ് 1, 2, 3 ശരിയാണ്, അത് 3 തെറ്റാണോ? ഏതാ അതിൻ്റെ ആൻസർ വരിക? സ്റ്റേറ്റ്മെൻറ്റ്സ് 1 and 2 ആർ കറക്റ്റ്, സ്റ്റേറ്റ്മെൻറ്റ്സ് 1, 2, 3 ആർ കറക്റ്റ്, ഓൺലി സ്റ്റേറ്റ്മെൻറ്റ് 1 ഈസ് കറക്റ്റ്, സ്റ്റേറ്റ്മെൻറ്റ് 2 and 3 ആർ കറക്റ്റ്. ഏതാ അതിൻ്റെ ആൻസർ? അതിൻ്റെ ആൻസറാണ് സ്റ്റേറ്റ്മെൻറ്റ് 3 കറക്റ്റ് അല്ല, സ്റ്റേറ്റ്മെൻറ്റ്സ് 1 and 2 ആർ കറക്റ്റ്. Third question. In the huddle technology approach used in food preservation, which of the following statements are correct? Huddles technology statements are correct. Huddles technology combines multiple preservation techniques to inhibit microbial growth. The individual huddles in this method are optimized to maximize food quality and safety. Third one, only thermal and pH based huddles are considered effective in this technology. Either answer only statement 1 correct. Either answer B. Statements 1, 2 are correct. 3 only thermal and pH based huddles are considered effective in this technology. Fourth question. Which of the following statements about high pressure processing is true? Statements HPP preserves the sensory and nutritional qualities of food better than traditional heat treatments. It inactivates microorganisms and enzymes by applying extremely high pressures without high temperatures. HPP is ineffective against bacterial spores and cannot extend the shelf life of low acid foods. അപ്പോൾ ഇതിൽ ഏതൊക്കെ സ്റ്റേറ്റ്മെൻറ്റ്? 1 ശരിയാണോ, 2 ശരിയാണോ, 3 ശരിയാണോ, 3 ശരിയാണോ? ഏതാ അതിൻ്റെ ആൻസർ? അതിൻ്റെ ആൻസർ സി ആണ് വരിക. അതായത് സ്റ്റേറ്റ്മെൻറ്റ്സ് 1, 2 and 3 ആർ കറക്റ്റ്. 3 സ്റ്റേറ്റ്മെൻറ്റ്സും ഹൈ പ്രഷർ പ്രോസസ്സിങ്ങിൽ കറക്റ്റ് ആണ്. 5. Concerning the use of modified atmospheric packaging, MAP, which of the following statements is correct or accurate? MAP involves altering the gas composition inside the packaging to slow spoilage and microbial growth. Common gases used in MAP include nitrogen, oxygen and carbon dioxide. MAP extends shelf life but requires refrigeration to maintain food safety. ഇതിൽ ഏതാ ആൻസർ വരിക? 1, 2, 3 കറക്റ്റ് ആണോ, അല്ലെങ്കിൽ 3 മാത്രമാണോ കറക്റ്റ്, അല്ലെങ്കിൽ 1 മാത്രമാണോ കറക്റ്റ്? 3 കറക്റ്റ് ആണ്. അതായത് മാപ്പിൽ എന്ത് ചെയ്യും? 3 യൂസ് ചെയ്യുന്നു. 3 പറഞ്ഞ സ്റ്റേറ്റ്മെൻറ്റ്സ് കറക്റ്റ് ആണ്. Sixth question. Which of the following statements is true about pasteurization in food processing? Statements. Pasteurization involves heating food to temperature that kills harmful bacteria and pathogens. It completely sterilizes the product, making it shelf stable at room temperature. Pasteurization is commonly used for dairy products, juices and other perishable liquids. Only statement 1 is correct. Statement 1, 3 are correct. ഏതാ അതിൻ്റെ ആൻസർ വരിക? ആൻസർ ബി ആണ് വരിക. ബി എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ സ്റ്റേറ്റ്മെൻറ്റും 1ഉം 3ഉം ആണ് കറക്റ്റ്. രണ്ടാമത്തെ കറക്റ്റ് അല്ല. ഇറ്റ് കംപ്ലീറ്റ് സ്റ്റെറിലൈസേഷൻ നടക്കുകയില്ല. 7th question. In the extrusion process used for making food products, which of the following statement is correct? Statements. Extrusion involves forcing food ingredients through mold or die to form specific shape. High temperatures and pressures in the extruder cause gelatinization of starches and denaturation of proteins. Extrusion is unsuitable for producing nutrient-dense or shelf-stable snacks. ഏതാ അതിൻ്റെ ആൻസർ വരിക? ആൻസർ ബി ആണ് വരിക. 1 and 2 ആർ കറക്റ്റ്. മൂന്നാമത്തെ ശരിയല്ല, 3. 8th question. For canning as a method of food preservation, which of the following statements is accurate? Statement 1. Canning involves heating food in sealed containers to kill microorganisms and enzymes. Second one, it creates a vacuum seal that prevents recontamination of the food product. Third one, acid foods require higher temperatures in canning to prevent botulism. ഏതാ അതിൻ്റെ ആൻസർ വരിക? അതിൻ്റെ ആൻസർ വരിക ബി ആണ് അതിൻ്റെ ആൻസർ. 1 and 2 ആർ കറക്റ്റ്. അതായത് സി, ആസിഡ് ഫുഡ്സ് റിക്വയർ ഹയർ ടെമ്പറേച്ചർ ഇൻ കാനിങ് ടു പ്രിവെൻറ്റ് ബോട്ടുലിസം. അതെന്ത് തെറ്റാണ്. നെക്സ്റ്റ് 9th question. Which of the following statements about fermentation in food processing is true? Statement 1. Fermentation relies on naturally occurring microorganisms to convert carbohydrates into acids, gases, or alcohol. Second one, this process can extend the shelf life of foods and enhance their nutritional content. Third one, all fermented foods must be refrigerated to maintain quality and safety. ഏതാവും അതിൻ്റെ ആൻസർ? തേർഡ് ആൻസർ കറക്റ്റ് ആണോ? തേർഡ് കറക്റ്റ് ആണോ? അല്ല. അപ്പോൾ ഫസ്റ്റും സെക്കൻഡും ആണ് അതിൽ സ്റ്റേറ്റ്മെന്റ് കറക്റ്റ്. അപ്പോൾ സ്റ്റേറ്റ്മെന്റ് 1 and 2 ആർ കറക്റ്റ്. വളരെ ശ്രദ്ധിച്ച് വേണം ആൻസർ ചെയ്യാൻ, തെറ്റാൻ പാടില്ല. Regarding 10th question, regarding irradiation in food technology, which of the following statements is correct? Statement 1, irradiation uses ionizing radiation to kill pathogens and pests in food. Second one, it is widely used for all types of foods without regulatory restrictions. Third one, irradiation can slow ripening and sprouting, extending the shelf life of certain foods. Option A, only statement 1 is correct. B, statement 1 and 3 are correct. C, statements 1 and 2, 3 are correct. D, only statement 1, 3 is correct. ഏതാ ആൻസർ? ആൻസർ ബി ആണ് ഇതിൻ്റെ ആൻസർ. ഇതാണ് സ്റ്റേറ്റ്മെന്റ് 1 and 3 are correct. രണ്ടാമത്തെ കറക്റ്റ് അല്ല. രണ്ടാമത്തെ ഇറ്റ് ഈസ് വൈഡ്ലി യൂസ് ഫോർ ഓൾ ടൈപ്സ് ഓഫ് ഫുഡ്സ് വിത്തൗട്ട് റെഗുലേറ്ററി റെസ്ട്രിക്ഷൻസ്. അത് കറക്റ്റ് അല്ല. താങ്ക് യു. വിച്ച് ഈസ് നോൺ ആസ് വാറ്റ് പാസ്ചറൈസേഷൻ? LTH, STST, UHT. ഏതാ അതിൻ്റെ ആൻസർ? LTH pasteurization. LTH is known as wet pasteurization. Next question, which organism shows highest resistance? ഇത് നിങ്ങൾക്കുള്ള ക്വസ്റ്റ്യൻ ആണ്. ഇതിലുള്ള 4 ആൻസറിൽ ഏതാണ് ഇതിന്റെ ആൻസർ? വിച്ച് ഓർഗാനിസം ഷോസ് ഹൈയസ്റ്റ് റെസിസ്റ്റൻസ്? അത് നിങ്ങൾ കമന്റ് ചെയ്യുക ഇതിന്റെ ആൻസർ. നെക്സ്റ്റ് ക്വസ്റ്റ്യൻ, pasteurization is the process of heating milk above 121 degrees Celsius, above boiling point, below boiling point, above 150 degrees Celsius. നമ്മൾ നേരത്തെ കണ്ടാണ്. ബിലോ ബോയിലിംഗ് പോയിന്റ് ആണ്, നമുക്കറിയാം, ഡിസ്കസ് ചെയ്താണ്. ദ പ്രോസസ് ഓഫ് പ്രിസർവിംഗ് ഫുഡ് ബൈ റാപ്പിഡ് ഫ്രീസിംഗ് ഫോളോഡ് ബൈ ഡീഹൈഡ്രേഷൻ അണ്ടർ വാക്വം ഈസ് കോൾഡ് ലയോഫിലൈസേഷൻ ആണ്, നമുക്കറിയാം അതിന്റെ. ദ പ്രോസസ് ഓഫ് പ്രിസർവിംഗ് ഫുഡ് ബൈ റാപ്പിഡ് ഫ്രീസിംഗ് ഫോളോഡ് ബൈ ഡീഹൈഡ്രേഷൻ അണ്ടർ വാക്വം ഈസ് കോൾഡ് ലയോഫിലൈസേഷൻ. നെക്സ്റ്റ്, കോൾഡ് സ്റ്റെറിലൈസേഷൻ റിഫേഴ്സ് ദ പ്രിസർവേഷൻ ഓഫ് ഫുഡ് ബൈ റേഡിയേഷൻ ആണ് ഇത് കോൾഡ് സ്റ്റെറിലൈസേഷൻ എന്ന് പറയുന്നത്. All of the following techniques are household preservation technique except smoking, dehydration, salting, lyophilization household preservation technique alla. Next, what is the purpose of blanching, immersing food in hot water, vegetables during canning? ഏതാണ് അത്? 3 എണ്ണം കൂടിയുള്ളതാണ്. കോമ്പിനേഷൻ ടു സോഫൻ പ്രൊഡക്ട്സ്, ടു ഫിൽ ബെറ്റർ, ടു denature enzymes, to reduce microbial population. Next, who is regarded as the father of canning? Nicholas Appert. Next, the method of preventing or reducing pathogens in food products by combining many foods like high temperature during processing, low temperature during storage, increasing the acidity, etc. is called huddle technology. The combination of these techniques and huddle technology എന്ന് പറയട്ടോ. അത് മനസ്സിലാക്കാൻ വേണ്ടിയിട്ടാണ് ഇങ്ങനെ കൊടുത്തിട്ടുള്ളത്. The method of preventing or reducing pathogens in food products combining many methods. ഏതൊക്കെ? Like high temperature during processing, low temperature during storage, increased acidity, etc. are called Huddle's technology. Which of the following methods dehydrates microbial cells by plasmolysis, thereby killing them? Plasmolysis എങ്ങനെയാ വരിക? Plasmolysis high sugar, sugaring ആണ് ആൻസർ. The reason for food spoilage is growth of microorganisms like bacteria, fungus, autolysis, oxidation by air that causes rancidity to color or color changes. 3 വരും. അപ്പോൾ all of the above. The process of preserving meat by stewing in a covered earthenware jug. എന്താ പറയുക? ജഗ്ഗിങ് എന്ന് പറയട്ടോ. എന്താണ്? ജഗ്ഗിങ്. നെക്സ്റ്റ്, Clostridium botulinum grows well in high acid foods, low acid foods, above pH 4.6, D both B and C. അതായത് ലോ ആസിഡ് ഫുഡ്സിലും എബോവ് pH 4.6ലും എന്ത് ചെയ്യും? Clostridium botulinum നന്നായിട്ട് ഗ്രോ ചെയ്യുമെന്നാണ് നമുക്കറിയാൻ പറ്റുന്നത് അല്ലേ, നമുക്ക് മനസ്സിലാവുന്നത്. ആ 2 ഇതിലും എന്ത് ചെയ്യും? നന്നായിട്ട് ഗ്രോ ചെയ്യും. നെക്സ്റ്റ്, ഇൻ കേസ് ഓഫ് ഡീഹൈഡ്രേഷൻ the moisture contents of dried products should not be more than 6 to 8% for vegetables. What is the percentage range of moisture content of dried products for fruits? Dried products in 10 to 20% answer. Vegetables in 6 to 8%, fruits in 10 to 20%. Next, the efficiency of moist heat sterilization is tested with vegetative cells of Bacillus, B. Spores of Bacillus stereothermophilus, C. Spores of aerobic bacteria, D. Clostridium botulinum. Spores of Bacillus stereothermophilus. ഇപ്പോൾ അതിൻ്റെ പേര് മാറ്റിയിട്ട് ജിയോബാസിലസ് സ്റ്റീരിയോതെർമോഫിലസ് എന്നാക്കിയിട്ടുണ്ട്. അപ്പോൾ ജിയോബാസിലസ് തെർമോഫിലസ് നമ്മൾ ഓട്ടോക്ലേവിൽ എന്ത് ചെയ്യും, അത് നമ്മൾ ഓട്ടോക്ലേവിൻ്റെ എഫിഷ്യൻസി ടെസ്റ്റ് ചെയ്യാൻ വേണ്ടിയിട്ട് യൂസ് ചെയ്യുന്നതാണ്. അത് യൂസ് ചെയ്തിട്ട് അത് ടെസ്റ്റ് ചെയ്യാൻ പറ്റും. നെക്സ്റ്റ്, വിച്ച് ഓഫ് ദ ഫോളോയിങ് വാസ് ഫസ്റ്റ് വൈഡ്ലി യൂസ്ഡ് ആൻറ്റിസെപ്റ്റിക് ആൻഡ് ഡിസിൻഫെക്റ്റൻറ്റ്? ക്ലോറിൻ, ഫിനോൾ, ആൽക്കഹോൾ, മെർത്തിയോലേറ്റ്. ഇതിൽ ഏതാ അതിൻ്റെ ആൻസർ? ഇതിൻ്റെ ആൻസർ ഫിനോൾ ആണ് ഇതിൻ്റെ ആൻസർ. വിച്ച് ഓഫ് ദ ഫോളോയിങ് വാസ് ഫസ്റ്റ് വൈഡ്ലി യൂസ്ഡ് ആൻറ്റിസെപ്റ്റിക് ആൻഡ് ഡിസിൻഫെക്റ്റൻറ്റ്? ഫസ്റ്റ് യൂസ് ചെയ്ത് ഫിനോൾ ആണ്. നെക്സ്റ്റ്, ഓൾ ഓഫ് ദ ഫോളോയിങ് ആർ സ്പോറോസിഡൽ എക്സെപ്റ്റ്. എല്ലാതും സ്പോറോസിഡൽ ആണ്, പക്ഷെ എന്തല്ല, ഏതാണ് സ്പോറോസിഡൽ അല്ലാത്തത്? അതിൻ്റെ ആൻസർ എ, ആൽക്കഹോൾ സ്പോറോസിഡൽ അല്ല. ബട്ട് സ്പോറോസിഡൽ ആക്ഷൻ ഉള്ളത് ഏതൊക്കെയാണെന്നുള്ളത് ചിലപ്പോൾ എക്സാമിന് ചോദിക്കാം. ഹൈഡ്രജൻ പെറോക്സൈഡ്, എത്തിലീൻ ഓക്സൈഡ്, ഫോർമാൽഡിഹൈഡ് ആർ സ്പോറോസിഡൽ, ബട്ട് ആൽക്കഹോൾ is not സ്പോറോസിഡൽ. Next, identify the term that can describe a disinfectant that can inhibit the growth of fungi. Fungicidal, fungistatic, fungicidal again, and microbiostatic. എന്താ അതിൻ്റെ ആൻസർ? ഫംഗിസ്റ്റാറ്റിക്. ഫംഗിസ്റ്റാറ്റിക് ആണ്. ഇത് ഐഡന്റിഫൈ ദ ടേം ദാറ്റ് ക്യാൻ ഡിസ്ക്രൈബ് എ ഡിസിൻഫെക്റ്റന്റ് ദാറ്റ് ക്യാൻ ഇൻഹിബിറ്റ് ദ ഗ്രോത്ത് ഓഫ് ഫംഗൈ ആണ് ഫംഗിസ്റ്റാറ്റിക്. Each of the following statements concerning the killing of bacteria is correct except. അതായത് each of the following statement concerning the killing of bacteria is correct. Killing of bacteria എന്തെന്ന കാര്യങ്ങളും കൂടി ചെയ്യാൻ പറ്റും, പക്ഷെ ഒറ്റൊന്നും മാത്രം അതിന് എക്സെപ്ഷൻ ആണ്. അതെന്താണെന്ന് നമുക്ക് നോക്കാം. An autoclave uses steam under pressure to reach the killing temperature of 121 degrees Celsius. പറ്റുമോ? Second, the pasteurization of milk kills pathogen but allows many organisms. The pasteurization of milk kills pathogens but allows many organisms and spores to survive. C. Iodine kills by causing the formation of thymine dimers in bacterial. A 70% solution of ethanol kills more effectively than absolute 100% ethanol. So, but answer the C ആണ്. C ആണ് iodine kills by causing the formation of thymine dimers in bacteria. അതാണ് എന്ത് എക്സെപ്ഷൻ ആയിട്ടുള്ളത്. ബാക്കി മൂന്നും കറക്റ്റ് ആണ്, അതിൽ കില്ല് ചെയ്യാൻ പറ്റും. ഇതുകൊണ്ട് എന്ത് പറ്റില്ല? ബാക്ടീരിയ കില്ല് ചെയ്യാൻ പറ്റില്ല അതിനുള്ളത്. അപ്പോൾ മൂന്നെണ്ണത്തിൻ്റെ ആൻസർ കറക്റ്റ് ആണ്. അപ്പോൾ ഇവിടെ ആൻസർ ഏതാണ്? സി ആണ് ആൻസർ. നെക്സ്റ്റ്, വിച്ച് ഓഫ് ദ ഫോളോയിങ് ഈസ് യൂസ്ഡ് ടു ക്ലീൻ ഡെലിക്കേറ്റ് ഇൻസ്ട്രുമെന്റ്സ് ഓർ ഇൻസ്ട്രുമെന്റ്സ് വിത്ത് മൂവിംഗ് പാർട്സ്? മൂവിംഗ് പാർട്സ് ഉള്ള ഡെലിക്കേറ്റ് ഇൻസ്ട്രുമെന്റ്സിനെ എങ്ങനെയാണ് കില്ല് ചെയ്യുക? അതിന് മെത്തേഡ് എന്താണെന്നുള്ളതാണ്. അൾട്രാസോണിക് ക്ലീനിങ് നടക്കുമോ? അറിയില്ല, നമുക്ക് നോക്കാം. ബി. ഡിസിൻഫെക്ഷൻ നടക്കുമോ? അറിയില്ല, നമുക്ക് നോക്കാം. സി. സ്റ്റെറിലൈസേഷൻ ആണോ? സാനിറ്റൈസേഷൻ? അപ്പോൾ നിങ്ങൾക്ക് ആൻസർ അറിയാച്ചാൽ കമൻറ്റ് ചെയ്യൂ, കമൻറ്റ് ബോക്സിൽ കമൻറ്റ് ചെയ്യൂ. എന്താ ഇതിൻ്റെ ആൻസർ വരിക? ഇതിൻ്റെ ആൻസർ എ ആണ് ആൻസർ വരിക. അൾട്രാസോണിക് ക്ലീനിങ് ആണ്. വിച്ച് ഓഫ് ദ ഫോളോയിങ് ഈസ് യൂസ്ഡ് ടു ക്ലീൻ ഡെലിക്കേറ്റ് ഇൻസ്ട്രുമെന്റ്സ് ഓർ ഇൻസ്ട്രുമെന്റ്സ് വിത്ത് മൂവിംഗ് പാർട്സ്? അൾട്രാസോണിക് ക്ലീനിങ്. നെക്സ്റ്റ്, എൻഡോസ്കോപ്പ് ഈസ് സ്റ്റെറിലൈസ്ഡ് ബൈ. നമുക്കറിയാം എൻഡോസ്കോപ്പ് നിങ്ങൾക്ക് വെരി ഫെമിലിയർ ആവുമ്പോൾ എൻഡോസ്കോപ്പ് സ്റ്റെറിലൈസ് ചെയ്യുക എങ്ങനെയാണ്? ഓട്ടോക്ലേവ് ചെയ്യാൻ പറ്റുമോ? അതോ ഗ്ലൂട്ടറാൾഡിഹൈഡ് യൂസ് ചെയ്യാൻ പറ്റുമോ? അതോ ഹോട്ട് എയർ ഓവനിൽ വെക്കാൻ പറ്റുമോ? ഫോർമാൽഡിഹൈഡ് യൂസ് ചെയ്യാൻ പറ്റുമോ? എന്താ അതിന് ആൻസർ? ബി ആണ് ആൻസർ. ഗ്ലൂട്ടറാൾഡിഹൈഡ് യൂസ് ചെയ്തിട്ടാണ് എൻഡോസ്കോപ്പ് എന്ത് ചെയ്യുക? സ്റ്റെറിലൈസ് ചെയ്യുക. സ്റ്റെറിലൈസ് ചെയ്യുക എന്ന് പറഞ്ഞാൽ നമ്മൾ പറഞ്ഞു ഡിസിൻഫെക്ഷൻ, സ്റ്റെറിലൈസേഷൻ. സ്റ്റെറിലൈസ് പറഞ്ഞു കഴിഞ്ഞാൽ ഫുള്ള് അതിൻ്റെ സ്പോർട്സിനെ കില്ല് ചെയ്യാൻ പറ്റണം. നെക്സ്റ്റ്, ദ മോസ്റ്റ് സ്യൂട്ടബിൾ വേ ഓഫ് സ്റ്റെറിലൈസേഷൻ ഓഫ് ഓയിൽസ് ആൻഡ് ഫാറ്റ്സ് ഈസ്. എന്താണ് ദ മോസ്റ്റ് സ്യൂട്ടബിൾ വേ ഓഫ് സ്റ്റെറിലൈസേഷൻ ഓഫ് ഓയിൽസ് ആൻഡ് ഫാറ്റ്സ്? ഏതാണ്? ഓട്ടോക്ലേവിംഗ് പറ്റുമോ? പാസ്ചറൈസേഷൻ പറ്റുമോ? ഹോട്ട് എയർ ഓവൻ പറ്റുമോ? ടിൻഡലൈസേഷൻ പറ്റുമോ? ഏതാ അവിടെ ആൻസർ വരിക? സി ആണ് ആൻസർ. ഹോട്ട് എയർ ഓവൻ യൂസ് ചെയ്തിട്ടാണ് എന്ത് ദ മോസ്റ്റ് സ്യൂട്ടബിൾ വേ ഓഫ് സ്റ്റെറിലൈസേഷൻ ഓഫ് ഓയിൽസ് ആൻഡ് ഫാറ്റ്സ്. നെക്സ്റ്റ്, വിച്ച് ഓഫ് ദ ഫോളോയിങ് ഈസ് യൂസ്ഡ് ബോത്ത് ആസ് ഡിസിൻഫെക്റ്റന്റ് ആൻഡ് ആന്റിസെപ്റ്റിക് ബട്ട് ഇറ്റ്സ് എഫക്റ്റീവ്നെസ് ഈസ് ലിമിറ്റഡ് ബൈ ദ പ്രസൻസ് ഓഫ് ബ്ലഡ് പ്രൊഡക്ട്സ്, മ്യൂക്കസ് ഓർ സോപ്പ്? ഏതാണ്? ഏതാ ആൻസർ? ഫോർമാൽഡിഹൈഡ് ആണോ? ആസിഡ് ആണോ? ബോയിലിംഗ് വാട്ടർ ആണോ? അയോഡിൻ ആണോ? ഏതാ അതിൻ്റെ ആൻസർ വരിക? ആൻസർ ഈസ് അയോഡിൻ. Which of the following is used both as a disinfectant and antiseptic, but its effectiveness limited by presence of blood products, mucus or soap? But the answer is iodine ആണ് അതിൻ്റെ ആൻസർ. Next, any process that destroys the non-spore forming contaminants on inanimate objects is. ഏതാ അതിൻ്റെ ആൻസർ? എനി പ്രോസസ്. എന്താ ശ്രദ്ധിക്കേണ്ടത്? എനി പ്രോസസ് that destroys the non-spore forming contaminants on inanimate objects is. Antisepsis എന്നാണ് പറയുക. ആലോചിച്ച് നോക്കുക. നമ്മൾ ഡിസ്കസ് ചെയ്യുക ഡിസിൻഫെക്ഷൻ ആണോ, സ്റ്റെറിലൈസേഷൻ ആണോ, ഡീജെർമിനേഷൻ ആണോ? നമ്മൾ പഠിച്ചതാണ്. അതിൻ്റെ ആൻസർ ബി ആണ്, ഡിസിൻഫെക്ഷൻ. നെക്സ്റ്റ്, വിച്ച് ഓഫ് ദ ഫോളോയിങ് മെത്തേഡ്സ് ഈസ് യൂസ്ഫുൾ ഫോർ സ്റ്റെറിലൈസേഷൻ ഓഫ് ആൻറ്റിസീറ? ഏതാണ് ആൻറ്റിസീറ നമ്മൾ സ്റ്റെറിലൈസ് ചെയ്യാൻ വേണ്ടിയിട്ട് യൂസ് ചെയ്യുക? ടിൻഡലൈസേഷൻ, എയർ ഫിൽട്രേഷൻ, ഓട്ടോക്ലേവിംഗ് യൂസ് ചെയ്യുമോ? എന്താ അവിടെ യൂസ് ചെയ്യുക? സി, ഫിൽട്രേഷൻ ആണ് ഇവിടെ യൂസ് ചെയ്യുക. എന്താണ്? ഫിൽട്രേഷൻ ആണ് യൂസ് ചെയ്യുക. നെക്സ്റ്റ്, which of the following is most effective for sterilization of mattresses and plastic petri plates? ഓട്ടോക്ലേവ് ചെയ്യാൻ പറ്റുമോ? എഥിലിൻ ഓക്സൈഡ് യൂസ് ചെയ്യാൻ പറ്റുമോ? UV യൂസ് ചെയ്യാൻ പറ്റുമോ? ക്ലോറിൻ യൂസ് ചെയ്യാൻ പറ്റുമോ? ബ്യൂട്രാൽഡിഹൈഡ് യൂസ് ചെയ്യാൻ പറ്റുമോ? നിങ്ങളുടെ അഭിപ്രായം എന്ത് യൂസ് ചെയ്ത് കഴിഞ്ഞാൽ നമുക്ക് എന്ത് ചെയ്യാൻ പറ്റുക? മാട്രസസും അതുപോലെ തന്നെ പ്ലാസ്റ്റിക് പെട്രി പ്ലേറ്റ്സും സ്റ്റെറിലൈസ് ചെയ്യാൻ പറ്റുക. എന്താ ഡി ആണ് അതിൻ്റെ ആൻസർ. എഥിലിൻ ഓക്സൈഡ് യൂസ് ചെയ്തിട്ടാണ് എന്ത് ചെയ്യാൻ പറ്റുക? എന്ത് ചെയ്യുക? സാധാരണ സ്റ്റെറിലൈസിങ് മാട്രസസ് ആൻഡ് പ്ലാസ്റ്റിക് പെട്രി പ്ലേറ്റ്സ്. നെക്സ്റ്റ്, ടിൻഡലൈസേഷൻ ഈസ് എ ടൈപ്പ് ഓഫ് ബോയിലിംഗ് ആണോ, ഇന്റർമീഡിയറ്റ് സ്റ്റെറിലൈസേഷൻ ആണോ, പാസ്ചറൈസേഷൻ ആണോ, ഡ്രൈ ഹീറ്റ് ആണോ? ഏതാ അതിൻ്റെ ആൻസർ? എന്താ? ടിൻഡലൈസേഷൻ. നമുക്കറിയാം ഇന്റർമീഡിയറ്റ് സ്റ്റെറിലൈസേഷൻ ഈസ് നോൺ ആസ് ടിൻഡലൈസേഷൻ. നെക്സ്റ്റ്, if a 1:600 dilution of a test compound kills a standard population of Staphylococcus aureus in 10 minutes but not 5 minutes, while a 1:60 dilution of phenols kills the population the same time, what is the phenol coefficient of the test compound? ഇത് ഫീനോൾ കോഎഫിഷ്യന്റ് ഡെഫിനിഷൻ വരുന്നതാണ്. അപ്പോൾ ഇതിൽ ഫീനോൾ കോഎഫിഷ്യന്റ് എത്രയാണ് വരിക? ഫീനോൾ കോഎഫിഷ്യന്റ് ബി 10 ആണ് വരിക കേട്ടോ. 10. ഇങ്ങനത്തെ ഒരു ക്വസ്റ്റ്യൻ വന്നു കഴിഞ്ഞാൽ 1:600 dilution ടെസ്റ്റ് കോമ്പൗണ്ട് കിൽസ് എ സ്റ്റാൻഡേർഡ് പോപ്പുലേഷൻ ഓഫ് Staphylococcus in 10 minutes but not in 5 minutes. അതാണ് ഫീനോൾ കോഎഫിഷ്യന്റ്. അപ്പോൾ വാട്ട് ഈസ് ദ ഫീനോൾ കോഎഫിഷ്യന്റ് ഓഫ് ദ ടെസ്റ്റ് കോമ്പൗണ്ട്? ഇവിടെ ഇതിൽ ടെസ്റ്റ് കോമ്പൗണ്ടിൽ എന്താണ് അതിന്റെ ഫീനോൾ കോഎഫിഷ്യന്റ് വരിക? 10 ആണ് വരിക. 10. നെക്സ്റ്റ്, എ ഡിസ്അഡ്വാൻറ്റേജ് ഓഫ് ഗ്യാസ് സ്റ്റെറിലൈസേഷൻ ഈസ്. എന്താണ് ഗ്യാസ് സ്റ്റെറിലൈസേഷന്റെ ഡിസ്അഡ്വാൻറ്റേജ്? The instruments must be placed in a small chamber. It takes up many hours to complete the process. It uses ethylene oxide, which is hazardous to humans and to the environment. It raises the temperature in the unit to 270 degrees Celsius Fahrenheit. ഏതാ അതിൻ്റെ ആൻസർ? ഇതിൻ്റെ ആൻസർ സി ആണ് കേട്ടോ. ഇറ്റ് യൂസസ് എഥിലീൻ ഓക്സൈഡ് വിച്ച് ഈസ് ഹസാർഡസ് ടു ഹ്യൂമൻസ് ആൻഡ് ടു ദ എൻവയോൺമെന്റ്. അതാണ് അതിൻ്റെ ഒരു ഡിസ്അഡ്വാൻറ്റേജ് ഉള്ളത്. വേൾഡ് ഫുഡ് ഡേ ഈസ് സെലിബ്രേറ്റഡ് ഓൺ. World Food Day is celebrated on 7th June, 16th October, 5th June, 22nd May. Which is the answer? The answer is B. 16th October is celebrated as World Food Day. Second question. Forastero is. What is Forastero? It's a variety of cocoa, it's a variety of tea, it's a variety of coffee, it's a variety of none of these. Which is correct? The answer is A. Variety of cocoa. Forastero is a variety of cocoa. Third question. Anti-nutritional factor in tea is. Tea contains an anti-nutritional factor. First one, caffeine or tannin. C, phytates, D, oxalates. Which is the anti-nutritional factor in tea? Is tannin. B is the answer. Anti-nutritional factor in tea is tannin. Fourth question. Thermophilic anaerobe or TA spoilage is caused by D-sulfotumaculum nigrificans, Clostridium nigrificans, Clostridium bifermentans, Clostridium thermosaccharolyticum. Which is the answer? Here answer is D. Clostridium thermosaccharolyticum. Fifth question. Spoilage of honey is due to dash. Spoilage of honey is due to first choice A, Zygosaccharomyces mellis, B, Zygosaccharomyces roxy, C, Candida, D, both Zygosaccharomyces mellis and Zygosaccharomyces roxy. Which is the answer? Here answer is D, both A and B. Sixth question. Caffeine in coffee is present in the range of 5 to 6%, 1 to 1.8%, 10 to 12%, 12 to 15%. Which is the answer? Here answer is B. 1 to 1.8%. Caffeine in coffee is present in the range of 1 to 1.8%. 7. Prolamine in rye is known as dash. Prolamine in rye is known as ordine, secalin, zein, gliadin. Which is the answer? Here is the answer is B. Secalin. Secalin. Prolamine in rye is known as secalin.
Eighth question. Colostrum is very good as it is rich in protein and carbohydrate, proteins and antibodies, minerals and vitamins. D. All of the above. Colostrum is the first milk from mother and which consists of protein and carbohydrate, proteins and antibodies, minerals and vitamins. So here answer is D. All of the above. Ninth question. AR enamel cans are used for. This is a question in canning. Enamel cans are used for acidless fruits, acid-rich fruits, acid-rich fruits with soluble colors, acid-less fruits with soluble colors. Which is the answer? Here answer is C. Acid-rich fruits with soluble colors. AR enamels are used for acid-rich fruits with soluble colors. 10th question. Carmoisin is a food additive used as a dash. Used as a liquefying agent or moisturizing agent, flavor or color. Here which one is the answer? Here answer is D. Carmoisin is a food additive used as a color. 11th question. Cow meat is produced from. Cow meat is produced from mare milk, cow milk, camel milk, sheep milk. Which is the answer? Here answer is mare milk. Cow meat is produced from mare milk. A. 12th question. Synthetic food color which is used to impart red color. Points you for are carmoisin, tetrazine, D both A and B. Which is the answer? Here answer is D both A and B. Points you for are carmoisin that are used to impart red color. 13th question. Cans treated with acid resistant lacquer are coated with SR enamel, AR enamel, C enamel, none of this. Which is the answer? Here answer is B, AR enamel. Cans treated with acid resistant lacquer are coated with AR enamel. 14th. BOG certificates are issued by. What is meant by BOG certificate? We will discuss in the next slide. These BOG certificates are issued by B, FSSAI. What is BOG? BOG means blissful hygienic offering to God. So it is to encourage places of worship to adopt and maintain food safety and hygiene in preparation, serving and sale of earth, along with training of food handlers for basic food safety and hygiene. This is BOG means blissful hygiene offering to God. 15th question is the last question of this session. Gluten proteins are rich in lysine, tryptophan, methionine or glutamine. Which is the answer? Here answer is D, glutamine. Gluten proteins are rich in glutamine. What is the importance of monosodium glutamate or MSG in canned food items? For what purpose we are using MSG in canned foods? MSG, it contains glutamate. It is a derivative of glutamic acid. So derivative of glutamic acid, the compound glutamate, which inhibits the development of spores or it inhibits spore formation. This inhibition can limit a cell's ability to form spores, which are essential for survival under adverse conditions. So MSG is used in canned food for or to inhibit the growth of spores. Second question. An outbreak of 15 cases, including one death of dash disease, a particular disease or foodborne illness, occurred in Bordeaux, France in September 2023 during the Rugby World Cup. Which is that particular disease? The disease is foodborne botulism. Foodborne botulism is caused by Clostridium botulinum. Third question. The most important chemical spoilage of canned food is dash. Which one? Which is the most important chemical spoilage of canned food? That is the hydrogen swelling. The hydrogen gas is formed by the action of acid of food on the iron base of the can. The can is made of iron-based cans. In iron-based cans, what will happen? The hydrogen swelling will happen. The spoilage usually occurs in high acid canned foods. It is usually happening in high acid foods such as apples, cedars and strawberries. So the most important chemical spoilage of canned food is the hydrogen swelling. Next question. What is the meaning of F value in canning? I have already asked you. This is the question for you. You have to comment. You have to give the answer. What is the answer? So F value is used to designate the time necessary to destroy what? The time necessary to destroy a given number of microorganisms at a reference temperature, usually 121 degrees Celsius autoclave temperature for spores or 60 degrees Celsius for vegetative cells. So the F value is used to designate the time necessary to destroy a given number of microorganisms at reference temperatures like usually 121 degrees Celsius in the case of spores or 60 degrees Celsius for vegetative cells. Next question, what is D value or decimal reduction time? What is D value or decimal reduction time? And so the time it takes to reduce a microbial population by one logarithm or 90% of reduction or 90% of its initial value under specified conditions like stirring concentration, exposure temperature, relative humidity, package configuration. This is known as D value. Fifth question, what is the 12D concept in canning? What is 12D concept in canning? The term 12D treatment means or it refers to the application of heat to kill 10 raised to 12 Clostridium botulinum endospores. So, to kill the application, it refers to the application of heat to kill 12 raised to 10 raised to 12 Clostridium botulinum endospores. This Clostridium botulinum, a heat resistance for forming pathogen. It is majorly found in low acid foods and it is extremely harmful and it is anaerobic and has the ability to survive and multiply in a sealed container. So that is the 12D concept of canning. Next question, sixth question. What is atmospheric steam canning? It is the latest method approved for home food preservation. Research conducted by the University of Wisconsin shows that steam in an atmospheric steam canner reaches the temperature of boiling water 212 degree Fahrenheit and is safe for processing high acid foods, same as boiling water. Next question, seventh question. What additives are used in canning? A number of preservatives such as citric acid, ascorbic acid, sodium benzoate, vinegar, salt, calcium propionate, sodium sorbate, potassium sorbate, benzoic acid, etc. are used in various foods and beverages in order to prevent spoilage and also to increase the shelf value of food during storage. That are the additives used in canning. Eighth question, flat sore. You might have heard about flat sore. Flat sore is primarily caused by the activity of heat-resistant bacteria, particularly strains of thermophilic bacteria such as dash and dash. What are that particular bacteria? Examples. The first one is Bacillus coagulans and the second one is Geobacillus stereothermophilus. These thermophilic bacteria are more resistant to heat and chlorine than thermoduric bacteria. What are thermoduric bacteria? The examples for thermoduric bacteria, Streptococcus, Lactobacillus, Corynebacterium, Microbacterium, Micrococcus, alkaligens, and spore-forming bacteria. The thermoduric bacteria cannot withstand the heat like thermophilic bacteria. Next, the ninth question, sulfur stinger spoilage is caused by. What is sulfur stinger spoilage or the causative agent of sulfur stinger spoilage? Clostridium nigricans. Clostridium nigricans. Mostly found in low acid foods like peas and corn. Next question, 10th question. Dash is also an important chemical used to preserve ham, sausage, bacon, and other cured meats by inhibiting growth of Clostridium botulinum. This protects against botulism and reduces the rate of spoilage. Normally, sodium nitrate is used. Sodium nitrate is used to prevent the growth of Clostridium botulinum spores. Next question, 11th question. Dash is used in the can lining of Campbell's soup, Conagra and General Mills, ravioli, pasta, vegetables and juices can cause oxidative stress to kidney tissues and reduces the filtration capacity of kidney. Which is a chemical? That is BPA, bisphenol A. It will also cause albuminuria. 12th question. Canned food gastroenteritis infection is caused by is Campylobacterium jejuni and Clostridium perfringens. 13th question. Dash is a natural antimicrobial peptide produced by strains of Lactococcus lactis subspecies lactis that effectively inhibits gram-positive and gram-negative bacteria and also the outgrowth of spores of bacilli and Clostridia. So what is the answer? The answer is nisin. Nisin is the answer. Nisin is a particular bacteriocin, antimicrobial peptide or bacteriocin which is produced by gram-negative bacteria. Then 14th question. What is Botox? Botox is a botulinum toxin. A minor dose is used to reduce wrinkleness in our skin.